You already know we always start on time. So I have Shubha, HV Suresh, Sarvanan, Prasanna Bhagwan, Vel Murugan. What? Shyam Prasad, please come. Where? Shyam Prasad, can I have you as a panelist today? Mr. Krupakaran, Navneeta, Lakshmi, and Brinda Devi. Keynote speakers. I have a small request today. My team is always here, but I need those people who can stay back for the talent search. 1. Yes, sir? 3 o'clock. 2. 3. Prasanna Bhagwan, sir. Will you be here for the talent search? 4. So 1, 2, 3, 4. Ipo idu pakkam. Yes. No. Yes. Yes. No. Okay. No. Yes. Yes. Navneet. Will you be there for the valedictory and for the talent search?
At what time is your train?
Navneeta, will you be here?
They have a train to catch up.
All 3 of you are together? Okay, so I need 2 of you. 10 o'clock only, you have a train. This will finish before 6 o'clock. You saw me, no? I finish everything before 6. Okay, so you will be there for talent search. What we are doing... Huh? You will be here. Say, excellent. So I have 4 people on this side. Bhuvna, give me a paper pen, please.
Yes, ma'am.
What we are doing now is we are putting talent search in between. Thank you. Poor thing, the children have all prepared and come, and we don't have. So I need the 6 judges. Wherever you're sitting, you will be marking for singer, dancer, and whatever else you feel like giving. Don't worry about what awards. Please give awards freely. No problem. Madam, start. Prasanna Bhagwan, Shyam Prasad. Hey, hands up, judges. Shubha, Bhuvneshwari, Brinda Devi, Navneeta.
They are also willing, let them. Youngsters.
Done. I have 8 judges. All 3 are willing. Hey, madam.
Give me, ma'am.
All 3 over there. I need their names. Okay, done. Okay, done. Thanks. I have 8 judges for the talent search. What is your duties? Whole day you're doing it. Okay? I'm going to give you very limited time even in lunch break. Half an hour poet vandarunga. Lunch break also we are putting talent search. Okay? So we are starting with Ganesh Vandana and one dance which is part of the talent search. And we are lighting the lamp. Then we are starting the session.
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि
एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि
गानचतुराय गानप्राणाय गानान्तरात्मने गानोत्सुकाय गानमत्ताय गानोत्सुकमनसे गुरुपूजिताय गुरुदैवताय गुरुकुलस्थायिने गुरुविक्रमाय गुह्यप्रवराय गुरवे गुणगुरवे गुरुदैत्यकलक्षेत्रे गुरुधर्मसदाराध्याय गुरुपुत्र परित्रात्रे गुरुपाखण्डखण्डकाय गीतसाराय गीततत्त्वाय गीतगोत्राय धीमहि गूढगुल्फाय गन्धमत्ताय गोजयप्रदाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशाय
What's your name?
Smiling. Smiling.
Smiley.
Sia. Sia.
And which school?
Sri Bhavani Group of Institution.
Thank you, beta. Please, let her outside. Come, please. Bring this here, no? Come, come. Let's light the candle. Thank you. Come on, Goma. Bring it to the center stage. Camera angle here. I know, I'm so sorry. More technology. Now, Krupakaran is going to do the honors. First, introduce yourself because our judges have to write your name down.
Hi everyone, this is Varshini and I'm from Biotechnology third year. So I'm doing RVC.
So,
Varshini PS.
Varshini from RV College.
I'm doing a Kuchipudi Tarangam. It's called as Shankara Shrigiri.
Kuchipudi Tarangam. Kuchipudi Tarangam. Judges for talent search. She's doing Kuchipudi Tarangam and her name is Varshini from RV College. Bhuvneshwari, Shubha, you got it? Thank you. Yes, ma'am, start.
कृपासमुद्रम् 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 सुमुखम् त्रिनेत्रम् जटाधरम् पार्वती वामभागम् पार्वती वामभागम् सदा शिवम् रुद्रम् अनन्तरूपम् सदा शिवं चिदम्बरेशं हृदि भावयामि। शंकर श्री गिरिनाथ प्रभु शंकर श्री गिरिनाथ प्रभु शंकर श्री गिरिनाथ प्रभु शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु झनन 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 घुंघरू बाजे झनन 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 घुंघरू बाजे झनन झनन झन घुंघरू बाजे देव मुनि सब गगन विराजे शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु अधैध कीटतक श्रुति गति रागे अधैध कीटतक श्रुति गति रागे अधैध कीटतक श्रुति गति रागे
Tangidataka tarikataka tum tak jum Tangidataka tarikataka ti Tangidataka tarikataka tati Tangidataka tarikataka tum tak jum tak jum taka tarikataka tom taka tarikataka tom taka tarikataka tom tak jum taka tarikataka tom tak jum taka tarikataka tom tak jum taka tarikataka tom tak
अधैध कीटतक श्रुति गति रागे अधैध कीटतक श्रुति गति रागे अधैध कीटतक श्रुति गति रागे अधैध कीटतक श्रुति गति राजे अधैध कीटतक श्रुति गति राजे पद्मनाभवन कमल विराजे पद्मनाभवन कमल विराजे शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु के नृत्य विराजित चित्र सभा में शंकर श्री गिरिनाथ प्रभु के श्रीगिरि शङ्कर श्रीगिरि शङ्कर श्रीगिरि नाथ प्रभु
Thank you very much, Varshini. That was a wonderful performance. And I'm now leaving the stage to Bhuvna. I think everybody knows all our judges of here, Dr. Bhuvneshwari, Dr. Sarvanan, Dr. Shubha, Dr. Shyam Prasad, and Dr. Prasanna Bhagwan. But we are starting our first keynote speakers, who will be Dr. Kripakaran, Dr. Brinda Devi, and Dr. Navneeta Lakshmi. Please, Dr. Kripakaran, please introduce him.
Good morning, everyone. Again, I'm just inviting everyone for the second day of our conference. Our first keynote address here is Dr. Kripakaran. The small introduction about him is a leader in the high-growth business management studies, biopharma domain at domestic and international markets. He has a postgraduate in applied microbiology and international business management with 23 years of experience in the field of in vitro diagnostics, biopharma, genomics, molecular biology, and cell biology. And he is an expert in WAC under leadership, commercial excellence, strategic planning, startup experience, P&L responsibility, partnership in the Lean and Matrix organization. I would like to invite Kripakaran onto the stage, please.
Good morning, everyone. Is that the energy? How many of you hear the good morning from the audience? Good morning, everyone. Good morning. That's the energy. Can I call you as bio legends, all of you? Yes? Are you all bio legends? I'm not hearing anything. Are you all bio legends? Yes or no? Come on. Those who are bio legends, let the auditorium blast with your yes. Are you all the bio legends? Again. See the energy. See the energy. Have you seen the energy of Dr. Sunita Rana? Throughout, starting from morning till the night, round the clock. Don't you think energy is infectious? So can we say a big thank you for Madam? Yeah? For giving this opportunity, especially to Dr. Pankaj and Dr. Sunita Rana. Can we give a big thank you? Thank you with me. Wonderful. As you all agreed that you are all bio legends today, I'm going to tell you a strongest secret. I'm not going to give you a lecture. I'm not going to give you any advices. But I'm going to give you a strongest and a strangest secret which nobody identified so far in the world of biological sciences. Is that exciting? Yes? Wonderful. Before that, how many of you know me? I'm sure most of you don't know me. No? No? Look at that. That's the connection. See, according to me, the word stranger is the world's shortest valid word. You know why? The moment we met as strangers and we start a connection and communication, the word stranger have no meaning. How many of you agree with me? Because we are already connected. We are no more strangers. You can interact with me. I can interact with all of you. Yes or no? Wonderful. Let me tell you who I am. In my entire journey of the past 24 years, people call me Krupakaran. Even after 24 years, people call me Krupakaran. No doubt about it. But in these past 24 years, I've climbed so many mountains. You know, before 2000, I climbed my first mountain of academics. The second mountain I climbed was not Tosho. The first mountain I climbed, the second mountain I climbed as a lecturer in a private college. I was teaching microbiology, medical microbiology, immunology, and molecular biology for students like you. Yesterday, you saw some of my beloved students have become the world-renowned entrepreneurs in the industry today. Of course, Bhuvana and me, we both studied together in the same college. The second mountain I climbed is a company called Lilac Medicare. It's an immunoassay company. Today, it's called as Tosho. The third mountain I climbed is Lab India, where I had an opportunity to work with various world-renowned brands like ABI, PerkinElmer, and Invitrogen. Today, these brands are in the name of Invitrogen, Thermo Fisher. The third largest mountain I climbed for 13 years in different positions is Roche, which is one of the world's largest biotech company, where I had opportunity to introduce these brands in the market. And here, my expansion started because I had a dream. My dream is to travel the world. And that opportunity started from my fourth mountain, Roche. So while I introduced these brands in India, I had an immense opportunity to replicate these successes in various overseas markets. And that's how I traveled to Australia, Taiwan, various other countries to establish these brands. My fourth largest mountain, where is that? Yeah. Fourth largest mountain is Single Support, and that was my previous organization. Today, combining all these 24 years, I decided to serve the world. And now, if you ask me why I am saying this story, because this podium given by Dr. Rana, Dr. Sunita Rana, and Dr. Pankaj is helping me to announce myself. And I utilize these podiums to talk about what is the strangest secret today. You will know the secret until you stay with me in the end. Will you? Will you stay with me? Not in the room. The connection. Are you all listening me? Wonderful. Going 2024, there is something evolved. And that's called Wealthy Needs Movement. And what is Wealthy Needs Movement in this podium is to make technologies like Dr. Sunita and Dr. Pankaj evolved over a period of time has already reached certain amount of people, 15,000 people. But what makes these kind of technologies to reach millions and billions of people? That's what Wealthy Needs does. Enough is enough. Let me move ahead. What is in it for you going forward? As I said, international market coverage is my strength. Now, after knowing this information, how many of you wanted to connect with me? How many of you wanted to connect with me? Raise your hands. Those who don't raise your hands, no need to connect with me. You lost the opportunity. You lost the opportunity. I'm seeing very few hands. So it's always the success of 1% remains in the world. The top successful people remains in the top 1% of the world. So now those who have not raised their hands, are you the 1% or the 99% of successful people? I'll give you one more chance. How many of you wanted to connect with me at the end of the session? Raise your hands. Wonderful. Let me tell you more exciting news. This book is coming up in the month of November. And this is called Untold Corporate Stories. Who wrote this? Who wrote this? And who's that? I'm here. Thank you so much. Well, I said I'm not going to bore you with lectures. I said I'm not going to give you any advices. What I'm going to do, I'm going to give you 3 unique stories. 3 unique emotional stories, and one I happen to work inside of the story. How many of you know this girl? This good-looking, charming girl? Anybody? Nobody knows? Okay. This girl is called as Emily Whitehead. A very happy childhood for the first 5 years, a wonderful family. But at the end of the 5th year, there's something happened. Something serious happened. Emily was diagnosed with ALL, acute lymphoblastic leukemia. Once their parents diagnosed this, the doctors started treating her, giving her confidence that this is an easily curable cancer. Some cancers are curable, isn't it? Most of the cancers are not. And you will connect why I called you BioLegends at the end. Remember that. This was said to be a curable cancer, and the parents remains very happy. And it cured. But after a few weeks, there was a relapse. It got relapsed. And every time it relapsed, Emily's physical condition and mental condition deteriorated. And at some point of time, they have no other choice that this can't be cured. The final statement given by the doctors, we are losing Emily. And at this age, Emily have to live her terminal illness in the hospice, the death home. The people who go to hospice will never return back home. That was the decision. But there was a miracle. You know what was the miracle? And you found that miracle? I'll tell you the secret later. The miracle was the opportunities given to you and everybody in the field of biological sciences to work on cell and gene therapies, especially in Emily's case. So those days, I'm talking about way back in 2012, the therapy which they found is from the Children's Hospital of Philadelphia. They call it as CHORD. And that clinical trial, the research on CAR-T, gave a relief, but then they were not very sure. When doctors injected the clinical trial samples in the body of Emily, Emily started progression, a positive progression. Isn't that happy for their parents? Isn't that happy for the doctors? The day when they found Emily is absolutely cancer-free, the world's first CAR-T therapy evolved. So that day, they call it as Emily has become the first child in the world to receive CAR-T immunotherapy. This was the world story. When I was talking about Emily's stories, today at the age of 17, Emily is still cancer-free, and Emily Whiteson's foundation is educating people about CAR-T therapy. What's happening in India? When I was talking about this story with one of our application support scientists who are working under me, he said, "Sir, I know this story. Because there are similar research and work is going on in IIT Bombay and Tata Memorial Center. But at the same time, when there was announcements coming up from India, the first CAR-T cell therapy was given to the country in the year 2023, there is a group of people, there is an innovative group of people who are fighting to reduce the cost of this therapy. Yesterday, how many of you heard these therapies are extremely high cost? Can you afford this? If any of your connections, networks, relatives, friends have cancer, are they able to afford these therapies? Keeping that in mind, there is a set of scientists. There are two amazing ocular scientists working on this in Bangalore. And who are they? Who are they? Who are they? It's none other than Dr. Rana Agarwal. Dr. Sunita Rana Agarwal and Pankaj were working on this when this was going on. And the world brought a therapy worth of 17 crores, saved Emily. Can we all afford it? And what Dr. Sunita and Dr. Pankaj is making and giving to the world? How many of you follow Dr. Sunita Rana Agarwal? Raise your hands. How many of you follow this YouTube page? She's an athlete. She's a swimming athlete. How many of you know this? She is world 6. How many of you know this? Today you got this news. This is the power of information and communication sharing. Now, from whom you are listening this on the podium is what the world is listening. Can I have your commitment that you will constantly follow the page of Dr. Sunita? Yes? Everyone, pick up your phone. Type Dr. Sunita Rana Agarwal in YouTube. Look at their journey, the patient journey which she covers. But it remains in the YouTube. The people who view this are just viewing this as a fact of information. How many of you who happen to see these kind of information just keep this within yourself? Then how come the world comes to know about it? So that's called the power of being vocal about your knowledge, being vocal about your known information. And that's what I do. Now tell me who wanted to connect. How many of you wanted to connect with me to know the art of communication and make the world to know about your skills? Moving forward, it's a breakthrough cell therapy and gene therapy. There are so many scientific steps behind it. As an academic microbiologist, I'm not going to cover scientific steps behind it because you people made it. The doctors, the nurses, and the healthcare management people helped patients like Emily and patients who are in the Agarwal, help them. Is it right to call them as biolegends? Because the innovation starts from you, students and researchers. Everybody in this room, including the panel members, speakers, every audience here, we all emerge from the zone of students, isn't it? So the storyline connects from the stories, the students and researchers. Imagine the people who involved in bringing this therapy in the clinical trials, testing them and making it as a commercial product. The people who promotes it, the people who make the world aware of it, and the people who supplies the products, the people who injects the products, the people who takes care of those who are in need. Are we all not biolegends? Yes or no? We are biolegends. That's the reason I said we are biolegends, which includes policymakers, government people as well. Yesterday we had experts from Drug Control Office. Because unless they approve, these drugs do not go away in the market. So now, let me come to the real secret. And I think I'm going to cover this in the next 3 to 4 minutes. The real secret remains in the world. Because cell and gene therapy is not new in India. It's one of the oldest technologies in the therapeutic Indian world. The problem is the world is speaking about it, but we are not. So who has the responsibility to speak about the cutting edge therapies and innovations? If you feel I am a student, I am a professor, I am a scientist, my job is to innovate, then the innovation remains in your labs. The innovation just remains in your organization. Let me reveal the strongest and strangest secret in the bioscience world. The biggest secret is the disparity in the world, including India, where people keep their innovations and knowledge within them. Let me go into details. Before that, I have a question to all of you. Yesterday, Dr. Sunita was talking about the MANA technology in the video. How many of you watched that? How many of you saw that? Only this much. Seriously? In the evening? Raise your hands. How many of you saw that? More than the time the video was running, Dr. Sunita was running around everybody. Any doubts, any questions, any doubts, any questions? Yes or no? You see, there are some opportunities people talk about. I am not given an opportunity to talk. I am not given an opportunity to convey what I wanted. When Dr. Sunita asked, anybody have a doubt, ask me for more than 3 minutes, isn't that not an opportunity to talk? Yes? Then what pulled us back? Asking questions. If somebody don't understand, ask again. Nothing wrong in asking again. If you don't understand, nothing wrong in asking again, right? Until you understand. And that's exactly what the secret behind the power of communications, which is lacking in the world. And I categorize, this is when I travel around the world, when I see different technologies, different innovations lies within them. I'm not talking about all the innovations. There are platforms like CCamp, which brings innovations together and makes the world aware of it. But if I tell you, that opportunity reaches to only 5% of the industry. Only 5%. Remaining 95% of the industry are working in silos. They work within themselves, within their organizations. They don't talk out. They keep everything within them. The large organizations where the innovation belongs to the innovators, they don't come out and talk because that's their profit. But it's okay as long as it is available. Let the result reaches the right people who are in need. The strategic blind spots. This is something, the knowledge which is insufficient in the art of positioning innovation in science. We all talk science. We all talk innovation. But when you talk science, the moment you get bored, when this person will stop talking, when this session will get over. How many of you feel this? How many of you feel this when lectures are going on, when speeches are going on? It's called the art of talking science to the world. The third biggest challenge, nuance. This is very common. This is across the world. It's all about one country has an amazing capability. For example, India have all the capabilities except positioning, communicating to the right audience. Australia, they have amazing innovation capabilities, but they don't have production facilities. Cost of manufacturing in Australia is too high. Can't they ask India to make it? And who can facilitate that? If somebody who can come along with me, you will be a legend of making such worldwide connections, and that's what I enjoy to do. Fourth biggest problem, branding deficiency. We are all seeing commercial brands, commodity brands, advertisements in TV, YouTube's, shorts. How many of you have seen a science shorts, branding shorts? And today, 5,000 bio-innovations, bio-founders, and commercial organizations exist in India. And there are more than 3 lakh biotech organizations are incepted and functioning in the world. But those who start, especially those who are scientists who are starting these facilities, has this deficiency of branding their products. The fifth largest problem, commercialization. If profit is the only aim, then countries like India cannot afford. But service is the only aim, then every country can afford it. Now, combining together, what can bring all these challenges addressed? And that's exactly what I'm talking about. It's a movement. It's not an organization. It's a movement. And this movement is called as Wealthinis movement. And the movement talks about make the world aware of who are you, what your organization is, what are your skill sets in the field of bioscience. How does that sound? Isn't it amazing? Isn't it amazing? Yes? Now tell me, when I say connect with me is not just by words, by actions. I'm a man of actions. As long as your proximity needs to be close with me, I'm not going to ask you to scan this. It's absolutely your interest to stay with me. Because the moment you stay with me, you get opportunities. Global opportunities. You get opportunities to take scientific innovations from Sunita to the world and various other founders to the world. If you really wanted to stay in a close proximity with Kripakaran, founder of Wealthinis movement, you have 2 QR codes here. Stay in this community. Automatically, you will go where you wanted to go. I stay back again with the QR code. If you're not able to scan it, reach out to me. I'm available. It's absolutely for making the world know about Indian bioscience capabilities. Thank you so much. And I take all your gratitude and go back to my seat.
Thank you, Kripakaran. Thank you for your excellent talk. Any questions, students, to Dr. Kripakaran? Okay. I can then invite our next keynote speaker, Dr. Brinda Devi. She is Associate Professor at Queen Mary's College, Chennai, and she has over 13 years of experience in fishery biology and biotechnology. She applies modern biotechnical techniques to promote sustainability in the fishery biology, and her qualifications are MSc, MPhil, B.Ed, PhD, and so on. And she has an advanced research and interdisciplinary studies in zoology. And Dr. Brinda Devi is our long-term association with ISGCT. Can I invite? Oh, madam, you're here. Yes.
Thank you, Bona. Good morning. I'm a teacher. Good morning, students. That's it. Okay. Let me start like this. Kripakaran sir was asking you, are you all bio-legends? I could see only a few hands raising, right? Now I'm going to call you. Are you all biryani eaters? See? Such a josh, like what Lalit ji sir said yesterday. I'm seriously asking you, are you all biryani eaters? Yes? Do you know what is seasoned there in biryani? Your mom does, isn't it? When seasoning, she puts oil in the frying pan and or the cooker, and she puts something inside that. So I'm sorry. What's it? Can you? Anyone? That's my research topic here, and I've done some molecular docking with that biryani material, something inside that biryani, and you'll see next. Can you pass on the slide? I'll concentrate on my. Sorry, thank you. Now, so you're all interested in what is going to be next, isn't it? Now, next slide, please. So my topic is molecular docking, and as the engineering students, you might be knowing what is docking. That is, it's a binding interaction between the target and the ligand, which makes a complex. It might be a protein, or it might be a nucleic acid or ligand. And this is particularly used for drug development that is in pharmaceuticals. And the hero of the research is going to come in the next slide, please. Can you see what it is? Yes? Have you all seen it? Yes, it is star anise. You put a masala or some spicy substance inside the biryani material, isn't it? So this is star anise, and we took it for our research actually. The scientific name of star anise is Illicium verum, and I did some docking studies with this Illicium verum. It is a very, very, very flavorable substance, and essential oil is being prepared out of it. And they have predicted some structural conformation. That is the next part of this slide. Next, please. And have you all known that there is a very interesting material in this star anise, that is, it is called as alpha bergo. What is it? Alpha bergamotine. It's an anti-cancer agent, and it is not only an anti-cancer agent, it is also a highly it is an antibiotic against the pathogenic organism. And here our pathogenic organism is Aeromonas hydrophila, and we did some studies, biochemical studies, and we found out that there are certain compounds, out of which there is one compound, I've highlighted it here. It is sesquit. Can I have the hand mic? It is sesquiterpenoids, and it has alpha bergamotine, and we did some docking studies with that, and we found that that it is against some pathogens, as well as it has that is, it protects us from the cancer. And we interacted this alpha bergamotine and some that is the medicine which we use, like doxycycline as well as tamoxifen, which goes against the breast cancer, and we did this docking studies. Next, please. And our docking studies are here. And this is a part of my research work with my PhD student, Dr. Mogna Sundari, who's a colleague of those invitees here. She's my student. Yes, next slide, please. And we did some in silico docking process here, and the data was retrieved using the SwissProt database, which is relevant to both Aeromonas hydrophila as well as the breast cancer protein. And the structure was predicted using the Swiss model. 2D as well as 3D structures were predicted. Next slide. Now, for the ligand preparation, we took two ligand molecules. One is tamoxifen. That's a small selective estrogen receptor modulator, which is used to prevent the breast cancer, which is a medicine which is used for breast cancer. And then the doxycycline, as you all know, you take it for, it's an antibiotic. It is against the pathogen, it is used for the pathogenic studies. And we retrieved a 2D structure as well as we drew some 3D structure using the ChemDraw. And the protein ligand docking was performed using the autodock method. And the autodock method was, it was a semi-flexible docking approach where we retained the protein as rigids and then the ligands are flexible. Next slide, please. Now, you could see the, that is, I told you we drew the 3D structure using the ChemDraw, and the 3D structure of doxycycline is here, tamoxifen, as well as alpha-bergamottin is here, and it's there. See, it's you can see the 3D structure and the functional groups of these all 3D structures like doxycycline, tamoxifen, as well as alpha-bergamottin. Next slide, please. Coming to the main area of the research, we did the protein ligand mechanism of these, that is the protein which was chosen by us, and as well as those two ligand complexes using the Autodoc 4.2, and the allowed degrees of freedom for the ligand molecules. I told you the ligands are flexible and the protein is kept rigid. So only the side chains can move, side chain residues can move in the protein, otherwise the ligands are very flexible and the degrees of freedom are 10. And we used two types, that is box type as well as grid box parameters. And to predict the active site was also depicted out of it. And we made an exhaustiveness of 100 so that we could get the best output out of it. Next slide, please. Okay. Now, there are two more interesting things which was identified by us, like, see, actually we made an interaction of this tamoxifen as well as the doxycycline with two more proteins. That is, there was a transport protein and structure. I did not mention it here. That is the second point. Transport protein as well as a structural protein, which was used to combat the Aeromonas infections with doxycycline. And the docking result was also compared, the binding modes were analyzed with caspase-3 as well as BCL. BCL is breast cancer lymphoma 2 proteins. And we found out the potential synergistic effects of these proteins using the docking studies. And you can see the ribbon-like structure of these two proteins, that is the structural protein and the transport protein of Aeromonas hydrophila, which is a pathogenic organism. So you could see that the transport proteins in Aeromonas hydrophila, it facilitates the movement of the molecules like ions, atoms, everything across the cell membrane. And from the protein data bank, we retrieved this model. And then we also had the secretion protein. That is, it is type 3 secretion system. You call it as T3SS complex from the Aeromonas hydrophila. It is a structural protein. It also enables, that is, it injects the, with the help of only this T3SSS, Aeromonas hydrophila, it injects proteins directly into the host cells and establishes infections. Next slide, please. And, okay, there comes the docking studies. And we performed some docking studies using PyMol. And the docking scores we got was this one, that is minus 6.2 for alpha-bergamottin, which is docked against transport protein, and the structure is there. You could see the side chains with the amino acid residues. I'll explain it to you next. The next slide, please. Docking score of alpha-bergamottin against the structural protein of Aeromonas hydrophila, it is 4.8, and you could see the docking between the alpha-bergamottin as well as the structural protein. Next slide, please. And the next is the docking between the doxycycline against the transport protein, that is 6.9, and you could see the docking structure there. Yes, next. I'll quickly just move on. The docking score between the doxycycline and the structural protein. I've been telling about the transport protein and the structural protein, and we have been doing docking against alpha-bergamottin as well as structural as well as transport proteins, and we are also comparatively doing studies against doxycycline with structural protein as well as the transport proteins. Next. Now, we made the docking processes. I'll explain later to what happened about the docking processes. And next study of us is we did docking against caspase-3 and the BCL2 protein with the MCF7 cells. And caspase-3, you can also call that as Cas329306. It is a group of enzymes which plays a role in apoptosis. And it has a name in protein data bank, that is 2XYG. It is an unique identifier. And as well, there is B cell 2, B cell lymphoma 2. It is, 2 is also a protein that plays a very good role in apoptosis. And you know, apoptosis, it is a programmed cell death. Next, please. And now, like doxycycline, which is compared to alpha-bergamottin, we are also comparing alpha-bergamottin with caspase-3 protein, as well as against caspase-3 protein, as well as BCL2 protein, and these are the docking scores. And you could just see the docking score, that is 6.6, and how it is docked here. Next. The docking score and H bond interaction of alpha-bergamottin with BCL2 protein is here, and it is 4.43. Yes. Then I told you about the ligand, that is we are using against the protein. So that is tamoxifen against the caspase-3 protein. It is 6.98, the docking score. The next one is tamoxifen against the breast cancer lymphoma protein 2 protein. The docking score is 6.5. I hope you will remember all the docking scores now, and then we'll be going for the explanation. Next, please. Now, so comparing the docking score between the alpha-bergamottin and the doxycycline with aromonazide, we are going for only infectious proteins. And alpha-bergamottin against the transport protein, we saw the docking score was 6.2 kilocalories per mole. And you could have seen those hydrogen bonds there. I told you hydrogen bond interaction. So 3 hydrogen bonds were there with specific amino acids. So the amino acids are named there: tyrosine, isoleucine, tyrosine, as well as valine. And then where alpha-bergamottin was also docked against the structural protein, you could have seen the docking score of 4.8 kilocalories per mole. And 1 hydrogen bond was formed with the specific amino acids, that is tyrosine, arginine, and valine. And doxycycline was also docked against the transport protein, and the docking score was 6.9. And 2 hydrogen bonds were formed. Then the doxycycline was also docked against the structural protein, and docking score was 7.28. 2 hydrogen bonds formed with specific amino acids. So that was for the pathogenic studies, and we did a separate study with tamoxifen for the breast cancer cells, and MCF7 was for breast cancer cell that we took for our study. And we docked it, and alpha-bergamottin against the caspase-3 protein, the docking score was 6.6 kilocalories, and it formed 3 hydrogen bonds. You should note that the hydrogen bond interaction is very, very important in predicting the docking scores. So 3 hydrogen bonds formed with specific amino acids. Then the alpha-bergamottin also against BCL2 protein, the docking score was 4.43 kilocalories per mole. 2 hydrogen bonds were formed. And tamoxifen against the caspase-3 protein, as well as tamoxifen against the BCL2 protein. You could see the docking score as well as the amino acids involved in it. Next. So we made a comparative docking analysis of this alpha-bergamottin, tamoxifen, as well as the doxycycline. And the alpha-bergamottin exhibited superior docking potential among the compounds which are tested. So it had the highest docking score of 6.12 kilocalories per mole against the transport protein, as well as 4.8 kilocalories per mole against the structural protein. And as I told you, hydrogen bond interaction is very, very important in this docking process. So alpha-bergamottin forms 3 hydrogen bonds, whereas tamoxifen as well as doxycycline demonstrated only a few hydrogen bonds. So tamoxifen formed only 1 hydrogen bond with caspase-3 protein and 2 hydrogen bonds with BCL2 protein, whereas doxycycline formed 2 hydrogen bonds with both the target proteins. So variations in this docking potential as well as the hydrogen bond formation was attributed to the chemical structures, as well as we did the functional groups, and we also found out the molecular properties of these compounds. And these docking results, we also maintain that is specific amino acids were also, that is I told you about the amino acids within the side chain residues. It was also involved in the hydrogen bond formation, and it contributed to the binding efficacy, and it maintained the stability of the ligand protein complexes. So these docking results which we performed was insightful on computational simulations, and it also formed potential interactions. Next slide, please. So we made some GCMS characterization of the methanol extract of Illicium verum, wherein the alpha-bergamottin is understood to be therapeutic. It had therapeutic properties, and it was used for docking studies. And the protein-ligand interaction and the docking scores were evaluated. And from this computational docking studies, we ascertained the antibacterial as well as the anti-cancer properties of this particular compound alpha-bergamottin, which was characterized by GCMS in the extracts of the Elysium verum. Next. So I think you didn't see this. So I asked you in the first, before asking my doing my presentation, are you all biryani eaters? Yes? Yes. So you will never forget this. What compound? Alpha-bergamottin. Alpha-bergamottin, which is there is in your biryani, right? It is an anti-cancer agent and as well as it is a agent against the, it has an antibiotic property. It is an agent against the pathogenic organism, Aeromonas hydrophila. So put your knowledge into this and you can also do more research on this, right? Thank you. And next slide. There's a paper, this is a prestigious paper we published in Fish and Shellfish in Immunology, me and my student. It's an Elsevier journal. And we made all the studies. Apart from this docking studies, other studies were also done in this. Thank you.
Thank you very much, Dr. Binda Devi. Thank you very much. Wonderful talk.
I thank Dr. Sunita ma'am, Dr. Pankaj sir, and the organizers of the RV College of Institutions for this opportunity. Thank you, ma'am. We are honored to be with you always.
Thank you. Thank you so much, ma'am.
Thank you, Brinda Devi ma'am. Our next keynote speaker is Dr. Navanitha Lakshmi from Ithiraj College, Chennai, Department of Zoology. Madam, your stage.
Sorry, can you hear me? I heard from some of my speakers, Puratasi Masathla biryani kamchi emathitinga.
Good morning to all. On the outset, my heartfelt thanks to Dr. Sunita and Dr. Pankaj for having me here in this esteemed gathering of knowledgeable academicians. How many of you got to watch the movie Matrix? Matrix. Movie 3 decades ago, no? Even in between, yeah. So that was a blockbuster then, three decades ago. And the movie got us, it had many series and it got us to the tip of the seed. And that is when our knowledge into artificial intelligence was budding off. And from then on, I think we have moved down the lane a long way. And so my presentation is going to be on a small insight. Of course, I'm not going to take much of your time. It's going to be on a short insight into where AI and DL could be integrated in future research. So what is artificial intelligence? I know it's a big jargon for, at least for me, because we are basically zoologists. But artificial intelligence in its truest sense says intelligence artificial, something that is going to be generated by a fabricated machine, a human fabricated machine, which is designed to perform cognitive tasks like to think, to solve problem, to perceive, and above all, to make decisions. So with the generation of artificial intelligence and DL, that is deep learning, which involves a convolution of neural networks, I think this innovations is revolutionizing the field of regenerative medicine. So regenerative medicine, which involves basically healthcare. So that was a topic that was given to me, AI and ML in healthcare. Right? So what are the areas that AI and ML could be integrated in for making a better life? So that's what humankind is aiming at. So we are trying to revolutionize healthcare with AI and ML. So this slide will show you where and how genomics or proteomics or transcriptomics, metabolomics. So all these integrated fields are working together for a betterment. And the first major use of AI is in tissue engineering, where one of the main feature of AI and ML and DL or whatever, it is for personalizing the outcomes. So here, AI, the algorithms that are framed is so that they can collect the data for an individual, fabricate the materials, and then also predict the outcome. So AI and ML, deep learning, is where the machine is able to fabricate, right? And this is one more area where AI and ML is gaining more momentum, which is your cell therapy, where the cells are designed and what is the environment that can be given. All these are algorithms, programmed materials, information, which is going to collect the information from wide areas of data sets, put it into gathering in, and create environments for better growing or culturing of the cells, and how these cells can be infused, what are the problems that will be faced by the individual if these the cells are going to be infused, and how those problems can be overcome and create a better result. This is an overview. Gene editing, I'm not going to go much into it because my fellow speaker will be talking about the CRISPR-Cas system. So I'm just going to say that AI, it's not only your CRISPR-Cas9. There are variations of the endonucleases that are used for gene editing, which is being framed and formulated, which can be predicted based on your AI tool. And immunotherapy, sir has given a very wide picture about your CAR T cells, which is your chimeric antigen receptor T cells, where the T cells are gathered, and the T cell receptor, we know that T cells play a very important role in immunology, your lymphocyte, your T lymphocyte, which generates these receptors. And along with your NK cells, it is your T cells that blocks the advancement of cancer. So with a deficient immune system, this is a boon. Your CAR T cells is a boon in the field of cancer treatment. Challenges and limitations. Any machine learning we take will have the challenges. One of the most biggest challenge that is faced is bias. So it is said that the programmer, the person who is writing the algorithm, should not be biased. So if there is a bias, there is a possibility of a failure in the system. So that is one of the major issues that should be looked onto. And of course comes your regulatory issues. Here we are, you know that the recent talk is more projected to the killer bots, right? Of course I'm not talking about our Terminator, where we had cyborgs, turning against mankind itself. So there, the regulatory issue is one of the limitations that is faced by AI and ML. So there again, we should know how we have to bring out the algorithms that is going to bring about the betterment of mankind. And in future, apart from tissue engineering, cell therapy, and immunotherapy, AI is to be having its influence on personalized treatments, 3D bioprinting, drug discovery, cell manufacturing and surgery. That is, what is the scaffolding that can be used for the better infusion of the cell and for the localization of the cell in your tissues, predictive modeling and simulation. As I told you, here we are not talking about the Terminator or the very bad cyborg. So the future, so this, the platform is to give you and to project areas where the young minds, as he said, the bio legends, the future bio legends, to kindle your knowledge in these areas where you can exploit maybe a decade from now or a couple of decades from now. So this is going to be the major thrust area towards which you people will have to work. Thank you for giving me this opportunity. Thank you, the team.
So now I invite the next speaker, Dr. Leelavati, for her presentation.
Very good morning, students. And I stand in the midst of a learned community. I feel very proud to be a part of this conference. And I take this privileged opportunity to thank the organizing committee members. And before getting into my topic, that is CRISPR-Cas and genome therapy. That was my topic. Hope this won't bore you, because in this current scenario, in each and every house, you will have patients suffering from diabetic or cancer. For this, instead of going for so many complicated therapies, CRISPR-Cas, that is a new technology which made it ease, and it also less the pain in the patients. Let's see what is the technology it speaks about. One minute. What was it then? Okay, children. Meanwhile, let me tell what is CRISPR-Cas and why it is so important. For these 2 days, it's the second day of our conference, right? The first day, so many speakers, they gave you insight about, so what is the therapy and what you have to do, what is the cost of, that is for the treatment and all the things. Whereas CRISPR-Cas, that is the newly emerged technology, where you need not to go for pain-going therapies and all. So that it is the process of editing your genome. Hope you know the genome. So each single cell of the body, it have the entire genome of your body cells. I think you have the basic knowledge of the genome. Am I right, dear students? You have the basic knowledge. We have 23 pairs of chromosomes. Do you agree with me? Yes? Yes? So each chromosome have its own functions. Do you agree with me or not? Yes? Whereas what is the function of chromosome number 5? Anyone in the crowd? So you might be heard about 5p minus. It's in 5th chromosome, the p-arm. I hope you know about the centromere, and you have a p-arm and a q-arm. The chromosome have the p-arm and q-arm. Right, children? Right? Whereas if there is deletion in the p-arm, it causes a disease in the children's. Anyone, any guesses? Any guesses? It causes cat-cry syndrome. The child will cry like a mewing of a cat. You imagine how it will be. If a baby, it cries like mewing of the cat, how it will be. So that is called as cat-cry syndrome. I hope you know chromosome number 21. If any defect in this, what it will cause? Anyone? Anyone? It's a trisomy. Trisomy 21? Yeah, very good. It's a Down syndrome. In case 18, chromosome 18 defect, it causes Edwards syndrome. Like that, each chromosome has its function. Hope, my dear children, you might hear about sickle cell. Yesterday also, so many speakers, they have quoted about sickle cell diseases. So how the sickle cell is being caused? How it is caused? What's the reason for sickle cell? What is the therapy for that? the person who is having the sickle cell, that is, you imagine the RBC is a biconvex in the shape. If it is a sickle shape, while it is passing through the blood vessel, it will cause much pain for the patient. It is caused because of that is mutation which happened in chromosome number 1. That's what I said in the beginning. Each chromosome has its own function. So if this mutation occurs in chromosome 1, the gene for the sickle cell is BCL11A. BCL11A, that is the gene for sickle cell. So this gene is present in chromosome 1. Which one is this? So how I have to know? So that's what I'm talking. So we are in the era where we have much development in the field of biochemistry and molecular biology. Do you agree with me, children? Because the molecular biology and biochemistry is not the same as our ancestors as we studied. It is not like that. Much of improvement is made in the field or in the platform of biochemistry and molecular biology. That's what I said. Because of this increase in the knowledge, it paved the way for treatment purpose. The sickle cell, I said just before, it is present in chromosome 1, and the gene is for the sickle cell is present in chromosome 1, and the gene is
दग्निरजायतः ॐ सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामया सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् दीपो ज्योति नमोस्तुते ॐ स्वस्ति न इन्द्रो वृद्धश्रवाः स्वस्ति नः पूषा विश्ववेदाः स्वस्ति नस्तार्क्ष्यो आरिष्टानेमिः स्वस्ति नो बृहस्पतिर्दधातु ॐ नमो।
So I'm going to ask you, please, thank you. Please. Yeah, because my hand only had a little problem catching. Oh, thank you, thank you, thank you, thank you. Thanks, sir. Thanks, sir. I'd like to ask our inaugural address from our beloved minister. Please, Dr. Dinesh Gundu Rao, please give us your inaugural address. He has been an Indian politician for a long time. I think a second or third generation of politicians.
No, it's fine. Thank you, Dr. Sunita Agarwal. It gives me great pleasure to be here
to find the solution for the other. So next for this, instead of this is a second example for CRISPR-Cas, and yesterday I hope Bhuwana ma'am was talking about RPE65. Ma'am, do you remember RPE? Yeah. So this RPE, that is the gene that is 65, that is present in chromosome 1. Actually, this RPE mutation occurred means the people will have defect in visual cycle. Visual cycle. They won't be able to see the image properly. For this, instead of injecting into all the things, that's the old therapy, we have CRISPR-Cas gene therapy. Now it has been evolved a much, and you can edit the gene. The mutation part can be deleted, and the corrected gene can be infused in that tool, and you can inject to the person, and easily the treatment is also available for RPE. This is a successive rate of CRISPR-Cas. The next slide is, I will tell you how this CRISPR-Cas works. What is CRISPR-Cas? I said about Gelsinger, I said about RPE, I said about sickle cell. For all these things are important disease where the community is facing nowadays. For this, only solution is CRISPR-Cas9. Now there will be a doubt within you. What is this CRISPR-Cas? CRISPR-Cas is clustered. CRISPR is clustered regularly interspaced short palindromic repeat. I'll show you the image. So this is the CRISPR-Cas. So this is CRISPR is clustered, short, regularly interspaced. You can see the space that is regularly interspaced. The repeated unit will be of 21 to 40 base pairs. BP is a base pair, and for spacer will be 20 to 58 base pair. Where is found? This CRISPR under the enga where it is found? Any idea, dear students? Where you can find the CRISPR? What it is? I said the acronym for CRISPR, but where it is found? Where this CRISPR is found? Anyone? Anyone? So the CRISPR is it is a prokaryotic. It is present in prokaryote, not in a eukaryote, but it is widely used for eukaryotic treatment. Kindly listen, dear children, because as someone said, the speaker said, you are the bio-legends. So you have to know what's happening, the current modern technology. So instead of reading in the printed books and reading the past treatments, come to the newer treatment, what is available to you. So the theme of our conference is from the lab to market. This CRISPR is still in the lab, and you have to bring to the market. That is there in the hands of you. Okay. So here, CRISPR is a clustered, regularly interspaced, short palindromic repeat. So that is the CRISPR. The palindromic repeat, anyone, what is a palindromic? Anyone, what is a palindromic? Yeah, very good, my dear, very good. So from where, you can see the TC, GC, GC, CG. So if you read from up to down or down to up, it will be the same. It won't change. That is a palindromic repeat. Okay. So here, so the CRISPR, as I said, it is a prokaryote. It is not found in eukaryote. And Cas, it is the endonuclease. The CRISPR and Cas, they will bind together. So this is the immune deficiency, that is immune modulation where the bacteria will act. So if we target any disease like a fever or viral infections, our immune system will get activated, right? Am I right, girls, boys, students, am I right? Am I right? Yeah, whereas in bacteria, so what happens, they have modulated to form this CRISPR. So this CRISPR, it is a very clever technology. So it will eliminate the virus. Always the bacteria has been infected by virus. To eliminate the virus from the bacteria have made this CRISPR-Cas. So that's what we are using for human genome therapy. That was amazing and fantasy technology. The scientists and the doctors were see how it works, and they chosen this technology to infuse in human treatment. Okay, so here that is gRNA1 and gRNA2. Kindly listen, this is the technique how the CRISPR works. So the CRISPR gRNA1 and RNA2. The gRNA1, it will form the sequence, the DNA sequence, which is similar to the virus. So, okay, so here the gRNA, it will go and bind with the virus, and gRNA2, it will bind with the Cas, and together they will form a complex. You can find here that is this gRNA1, RNA2, and Cas9. Together, they will attack the target, that is a mutation site. So it will act as a molecular scissor. It will cut where the mutation has been occurred. Mutation, there will cut the gene, the nucleotide sequence. If for cutting to act as a molecular gene, it needs the PAM, PAM is the protospacer motif. Protospacer adjacent motif. If there is no protospacer adjacent motif, that is the CRISPR-Cas, it won't be able to function or it won't be able to act as a molecular scissors. Because molecular scissor is needed to delete or to cut the mutated site. Mutated site cut panadhan, you will be able to infuse the correct edited gene. And you can make the regulations and you can delete any diseases or whatever mutated site if you delete. So then the correct gene will get infused so you can treat the diseases. So if PAM is not there, the Cas, it won't act. PAM is essential for this Cas and this complex to act as a molecular scissor. PAM is a palindromic adjacent motif. It is also very essential. So in this, the laws that I said, DSB is a double-strand break. You can make a double-stranded DNA, you can make a cut, and you can delete any mutation like deletion. I said 5p minus. In case 5p minus is a cat-eye syndrome. That is, in chromosome number 5, the P arm, there will be deletion. So if the deletion is there, the child view will cry like a mewing of a cat. That's what I said in the beginning, right? So to remove that, you can insert a new gene for chromosome number 5, and you can edit. So that's what CRISPR-Cas has done. And any doubt in this? Any doubt? Any doubt? You can ask me now. And finally, so this CRISPR-Cas is an innovative development in medical science and in the field of research and development. These innovations improve the accuracy and effectiveness of gene editing methods, opening the door for creation of individualizing therapies. Not only thus, this tool can be used in the field of agriculture. How it can be used in agriculture? Whereas for treating human disease, this tool has been used. That's what I was talking so far. This can be used to treat in the field of agriculture. How it can be used? The same technique can be used in the field of agriculture. Anyone? It's the same thing, you know very well. The crops will be infected with viral diseases and the productivity and disease ability can be increased using this CRISPR-Cas pathway. Any doubts in this? Thank you, dear children. Thank you. Thanks a lot.
I thank Dr. Leelavati from Etraj College, Chennai for a nice presentation on CRISPR-Cas technology. Now I call upon the next speaker, Dr. L. Shakila, for a presentation.
No, I'm just inviting Tanai. Yeah. I would like to invite Dr. Tanai Bhatt for the next presentation. Tanai is a co-founder and CEO of Koshki Biosciences, and he is my colleague in National Center for Biological Sciences, and now he's working as a startup focusing on the antimicrobial resistant AMR and wound healing solution. Tanai, the stage is yours.
Thank you.
You want to?
Sorry to disturb. At this juncture, I take this opportunity to thank Dr. Brinda Devi ma'am from Queen Mary's College. She's associate professor for introducing me. Thanks a lot, ma'am. Thank you.
Thank you, ma'am. Yeah, the presentation is already there. It has come. You can use this.
Okay. Thank you very much, organizers and Bhuvna, for inviting me over here. As she mentioned, my name is Tanai Bhatt, and I co-founded Koshki Sciences along with Professor Colin Jamora about 2.5 years ago. In Koshki, the Kosh means in Sanskrit a cell, a biological cells. We are all made up of multiple billions of cells. A single cell is called Kosh, and key is to unlock the potential of that cell. So the vision behind Koshki is that can we use our body's own cells and unlock their potential to become disease-free? But it's, of course, easier said than done. So what we are trying to do is bring in novel RNA interference therapies for various kinds of diseases. And in this presentation, I'll take you through how we are doing it. But to begin with, let us understand what is the problem. The problem is that there are more than 10,000 diseases for which there is no treatment. These diseases are the ones which are rare and genetic in nature. And because they are rare, many of these diseases, we are not hearing them every day. What we hear every day is probably cancer, diabetes, and various kinds of resistant infections. And worldwide, they are the reasons behind, hundreds of deaths. However, the other rare genetic disorders which are not treatable are mostly treated, I would say, only for the symptoms. The current medicine is not treating the root cause of it, and hence patients are often frustrated because they have to keep on taking these medicines lifelong, and eventually, if you consider the cost they are spending for these therapies, it's a lot. So most of these diseases, as I mentioned, are genetic in nature, and if you want to treat a genetic disease, what would you do? And as the previous talk suggested, you would have to go for the genetic therapy. CRISPR-Cas9 is one of the ways you can do it, but in the umbrella of gene therapy, when you want to change a DNA code, and that too in each and every cell of your body, it's a very, very huge task, very difficult task. And traditional gene therapy that's why very rarely we see in the market, very rarely we see developed within India and given to the Indian patients. And that's why we need a new solution. What are the problems with the genetic therapy currently? It's a very high cost of investment. At least $2 billion you have to spend to bring out one therapy to the market. Second problem, high risk of failure. Only 28% chance that whatever you are starting today, after 5 years, may reach to the market if it is into this genetic therapy domain. And the third is, because you are spending so much time, energy, and investment of various investors, you'll have to charge patients a lot. And that's why you might have heard that a single injections for the DMD, Duchenne muscular dystrophy, which is one of the rare genetic disorder, cost more than $3 million per patient. Of course, that is a very, very small population of patients which can afford that. And hence, these kind of therapies are not becoming very popular, not only in India, but elsewhere in the world as well. So what is the solution? We are bringing in RNA interference therapy, which addresses key obstacles faced by the traditional gene therapy. What is the meaning of RNAi therapy? It's very simple. We are interfering with mRNA of a particular gene. It will lead to gene silencing of that particular gene which was causing the disease. And it's a solution for the long-term disease resolution at an affordable cost. So very briefly, I'll go through how this entire therapy works. And it will be a sort of an oversimplification because I'm considering that, most of the students right now might not know everything in detail. But as I mentioned, that it all starts with a mutation in your DNA. Because your DNA has a mutation, your RNA gets a mutation, and because of that, that unwanted protein gets produced, which causes the disease. Now, what you are seeing in the red color is antisense oligo, which is precisely tailor-made for that mRNA. We are not right now altering the DNA, but rather designing antisense oligo, which will bind only to that mutated mRNA and degrade it. This antisense oligo will result in the silencing of that gene, no protein production, and hence no disease. When we compare these RNA interference therapies with the traditional gene therapies, there are several advantages as well as it's comparable. It has, you can target, in fact, any gene in the body because each gene and each mRNA has its own sequence, its own signature. It has high efficacy and it is long-lasting. You can administer this therapy not only through the veins or subcutaneously, you can also inhale it. It can be applied across multiple areas, not only rare genetic, but nowadays these therapies are coming up for various commonly occurring diseases as well, such as hypercholesterolemia, heart diseases, kidney diseases, brain diseases. The cost of development, that's the most important thing for any pharmaceutical company. The cost of development is low, hence the cost of therapy to the patient is low. And I would say on the humanitarian side, the safety of this therapy is also very high. There are very low safety issues you would face in RNAi therapies. Why? Because if in the gene therapy something goes wrong, that something goes wrong will be embedded in your DNA for the rest of your life. But in the mRNA therapy, your DNA remains as it is. You are only targeting the mRNA, which is continuously being produced. And this therapy is, something which is not going to be integrated in your DNA. Hence, it is having very low safety issues. So our mission is that can we make this indigenously affordable, low regulatory hurdle RNAi therapies for diseases with high unmet need, for which often they are called orphan diseases. No drug is existing. and can we support research for the same for various other companies as well, right? So what we have done over here. So of the many things which are in the pipeline, I will be explaining just one of the therapies, which is targeting the AMR. Why AMR? Because if you look at various other diseases, AMR currently might look small, but by 2050, we will be seeing that every year, 10 million people will be dying because of the antibiotic resistance. Why is that happening? Of course, because we are, particularly in India, antibiotic use is uncontrolled. Bacteria continuously are evolving. So today, whatever antibiotic is working for a bacteria, tomorrow it might not work because that bacteria has evolved against it. The number of deaths you see might become higher than the number of deaths which is happening because of the cancer. What we have also observed that the antibiotic resistance does not affect all of us equally, rather certain population, those who are undergoing chemotherapy, those who are having HIV, or those who are diabetic are more likely to get these kind of infections. So there is a strong correlation between those population having weaker immunity are also going to get various kinds of infection. And if you think about AMR infections, they are often hospital-borne in nature. People with lower immunity get it, and people who are having non-healing wounds, those wounds often get it. So we know that if you are a healthy individual, if you get any injury, you do not worry much. Injuries are part and parcel of the life. There is nobody who can say that I have never got injured. And we don't worry much because it often gets healed nicely. But if we are a diabetic individual, our skin cells, particularly called skin keratinocytes, do not sense this injury, and it leads to progressive diabetic foot ulcers. This kind of foot ulcers, after a certain stage, also attracts various kinds of infections, necrosis, and after certain points, it's non-returnable. It leads to either limb amputation or sepsis-mediated death of that individual. So how can we intervene in this particular program? So what we discovered as part of my PhD program, I was working on it, that every skin cells have a pool of AMPs, which are called antimicrobial peptides. So if I take any bacteria and put on the skin of a healthy individual, mostly nothing happens because our antimicrobial peptides take care of it. However, in the diabetic individual, there is a break on the release of these antimicrobial peptides. We will call it for now a break gene. And when the break gene is highly expressed, the pathogen cannot be neutralized and it will continue to grow on your skin. So what do? We design a therapy, siRNA, small interfering RNA. You make it a paste or ointment out of it and deliver this topically near to the diabetic wound. This siRNA goes inside, silences that break gene, and once the break gene is gone, automatically antimicrobial peptide secretion starts. These antimicrobial peptides will also kill your pathogen neutralization as well as it will kickstart the wound healing program. So in one stone, you are killing two birds. You are also neutralizing the pathogens which are AMR in nature. You are also increasing the speed of wound healing, a perfect solution for a diabetic non-healing wound. So this is what I summarize, that if a patient is coming up with a chronic non-healing wound, the Koshki's therapy KK03 will be applied topically, which can bring back the healthy healing. It can resolve the infection as well as bring back healthy healing. This is how the therapy would look like in future. It would be topical in nature, antimicrobial peptide mediated neutralization of pathogen as well as faster wound healing. How did we come up with this thing? This is not just a hypothesis. We had to work on it at in vitro level. We took the cells of a diabetic population, we grew them in the culture, we observed that this was happening. We tried these in vitro conditions on the human cells, as well as we developed a mouse model of diabetes. This diabetes mouse model, just like a human patient, also shows this defect. And we tried to treat these diabetic mice with this particular formulation. So what you are seeing is that in this particular graph, if your mouse is wild type, which is no mutation, just like a healthy human being, a blue line shows that a particular size of a wound will heal in about 12 days. But if the mouse is diabetic, just like diabetic human being, it will take 24 days. If you convert that 24 days into the human population, it would turn into some 3 months or more. But now when you're applying RNAi, the green line, you would see that 24 days becomes 14 days. And we have to understand that what it means for the human diabetic population. It would mean that it's a 40% faster wound healing, 12 times more effective against infections, 30 days reduction in the follow-up period, and 25% reduction in the total treatment expenditure and opportunity cost. So moving forward, I just want to introduce you that when you are interested in developing any new drug, it is not a simple stages of moving from idea all the way to market. There are multiple stages you will have to cross. For this particular drug, what I am describing, it all started with the basic research, where we identified that one break gene was the gene which was causing issues in the diabetic population. That's where it's called idea stage, a target discovery stage. We worked out that in Institute for Stem Cell Science and Regenerative Medicine, as well as part of the research happened in Milan, IFOM Molecular Oncology Institute. Then we did the target validation, that whatever we are thinking that this medicine will work, will it really work? So we had to try it out in vitro condition in the human cells, explants, as well as the diabetic mouse model. That's target validation and lead generation. And currently we are in the efficacy and safety stage. You will have to convince the regulatory agencies in any country that this particular drug is also safe enough to be applied in the human. After that, you get early phase clinical trial approval. That's called phase 1 trial, which will be followed by phase 2 and 3. And then if you are all lucky, everything efficacious and safe in the human as well, then you reach the market. So we are currently in the preclinical development stage. So with that, I'm thankful for all of you. And if there are any questions, maybe now or later on, we can meet and discuss. Thank you.
Thanks, Tanay, for your very good presentation. Next, I would like to invite Dr. Divya, as a senior application scientist in Altum Technologies, Karnataka. She has delivered on the title, Unraveling the Role of AI and ML in Developing the Toxic-Free Compound Integrated Approach in a Sustainable Chemistry, and pursued a software development in Biova Discovery Studio. Dr. Divya, please carry on with it.
Very good morning to everyone. I think Kripakaran sir waked up everyone. So I think you all in a wake up stage, and followed by Brinda ma'am, and everyone has given you insight about what is going on in current industries. It is going to be very simple. Everything starts with the food. Food is something which you are consuming which should be toxic free. In case if your food is toxic, then whatever we are going to take inside our system is going to be toxic. And moreover, it makes me so pleasure to stand in front of Dr. Vel Murugan sir, who encouraged, as like you people, I will go and attend his session wherever he is giving in University of Madras or Bardar University. So it gives me so pleasure to stand in front of mentor, Dr. Vel Murugan sir. So to start it, agrochemical screening. Now we all are speaking about a screening of molecules and everything. But first we have to understand where we are starting. It's from the food. If your food is pure, then your system is pure, then everything what you want to do, it's going to be perfect. If there is a toxic is going to raise over there, it definitely affect your whole system. Then you have to get into gene therapies and treatments and everything. So I'm going to start in this route, how we have to make toxic-free materials for your agrochemicals, and through that how we can live so healthier for more than 100 years. Let us start it. So I would like to introduce my company, Altum Technologies, who are the leading partners in 3D printing technology and 3D scanners. And one of the wing in BioVia is Dassault system is BioVia Discovery Studio, Material Studio, Pipeline Pilot, and we are now more into an electronic laboratory notebooks, lab networks, and one labs and so on. So today I'm going to project about this combination of techniques, how it could benefit for everybody in screening of materials, including AI and NML. So I'm just skipping this. Agrochemical screening. Any idea? Why we need screening of agrochemicals? So for example, you are going for taking some kind of an ice creams. Would you like to see the formulations? Or any cosmetics? Let me come very easily to cosmetics. Everybody use cosmetics, right? All girls and boys? Okay, have you seen that what are the materials are inside it, how much mgs or things are there? No? Only if it is giving shiny and then good look, it's good to move on. Am I right? Come on, what are the products you will use? Definitely would have been come out with many kind of products, especially L'Oreal. Right? Not it, you don't use any cosmetics? Okay, I am tired, ma'am, so we need a food to fulfill ourselves. Is it so? Okay, I'll keep as simple as possible. So today is going to be like this. First, we are going to screen it. How we have a different materials to screen it. Then how to make a formulation recipe. That recipe should be as simple as possible and make you more effective as possible. That is going to be my talk. So agrochemical screening is very important. If your agriculture land is pure and perfect, then whatever you are going to take, it is going to be perfect. So we have to develop your chemicals in such a way, it should not affect your soils, it should not affect your plants, and as an outcome or consumers, we should be free from the chemicals. So we need a biological based materials which could degrade all these things. That's why we need agrochemical screening. So need of why biogenic? Everywhere you are seeing a biomaterials, biogeneratives, and sir told biolegends. So bio is something which you make you always that can be removed. And you can have a sustainability. Without having a sustainability, if you are developing some products, it is going to end up with a toxicity. So that's a reason we need a biological based materials. Say for example, it could be taken from microorganisms and or from the plants or different sources. You have to extract their biomolecular things and screen and formulate, and finally, without product, any research, it's just a paper again. We have to bring out the product formulations. So build the product in such a way, it should have a lower impact and reduce the risk to human health. How we are going to do this? So there are two different ways. One is a conventional method, which we are already utilizing, and one is a computational method. So conventional method, you will take for laboratory experiments, field trials, so it will take a time-consuming process, and you have the regulatory challenging, and you have to make a less trials because cost is more important actually. But however, you come to the computational techniques, like virtual screening. 101 times you do mistakes, it is not going to affect anybody. And if I build the models and I'm going to screen with that, it is not going to affect anybody. So that means I am preserving myself, I'm not affecting harmful to any organisms also. So this will be a less time consuming, highly cost effective. So this kind of technique will boost you like anything, and you don't have to worry about a regulatory, it comes in the further stages, and you can do more trials with the multiple formulations. So bioprospecting, how this BioVia prospecting will be supporting your agriculture, allochemical screening, precision farming, antagonism for plant pathogens, chemical priming, plant disease pathogenism, pheromones to control, and agrochemical screening. So these all are the prospecting that BioVia will help you to address these issues. So if you see these techniques, so we can have a different way of handling this technique. One is numerical based, another one is a pharmacophore and docking. Dr. Brinda ma'am has explained very good and information about by prospecting the biryani informations about arsenics and told about how this chemical will be benefit. But to deep dive into it, we are adding a pharmacophore to that. And along with that, we have a QSR and machine learning techniques could benefit more. This is one of the example which we perform with one of the customers, especially how the insecticides for chemical screening. Instead of working off all the chemicals, we have come out with various steps and optimize the libraries, and through that we obtain the specific similar molecules that is toxic free and it is not having any irritation to the skins and ocular retention is not there, and it is environmentally protected, and those kind of things we have designed in that chemical formulations. So whenever people use, it is not going to affect anybody. And how to perform this, just a small video for you all. So we are trying to work out on a similarity of molecules. Any kind of systems you can select and you can screen. I am taking some kind of natural products libraries. I try to screen for that. And I want most perfect similarities. And trying to find out with that. And I'm using a different distance method. You can choose any of the distance method, because anything if you see practically, it will be more to reach you people. That's why I have prepared this kind of videos. So I'm selecting it and trying to run it. And if in case the chemical is very less, it is not available, you can always have an alternatives by using these kind of techniques. So you can find the similarity of the things and you can process further. The another one method is very important in agrochemical screening is the chemicals that we are taking, it should not have any toxic to your cells, and you should be free from ADMET properties also. Using AI and ML based model, we can able to predict even all those things, will help you to understand whether that particular molecule is crossing a blood-brain barrier or it's causing hepatotoxicity. So all this information you can able to gather from this kind of AI and ML models. If you see at the back end, it's all of biostatistics. Everything is a max. Even biology, if you have put it across with the mathematics or statistics, that's where the boom of AI and ML is beginning actually. So any models of any systems you can develop with the help of AI and ML using this kind of technology. So this is an example of hepatotoxicity modeling of systems where we collected a molecule. Data mining is very important in AI and ML. Then we tested for screenings, which are the molecules are making hepatotoxicity, and we rolled out those molecules and taken only the non-toxic molecules. And we can able to test this for all these animal models, which is a part of a BioVR Discovery Studio software. And you can also test for ocular retention, deafenia, fat amino, aerobic biodegradability. All this with a single systems, you can able to analyze all these informations. All are based on AI and ML. And toxicity predictions, these all are, I told, this is based on Bayesian model classification. I told which kind of an algorithm. Almost we go for supervised learning technique, which will help you a lot to more understand these informations. So then regressions. This is one example how it works on. So for example, I'm giving some chemical molecules. I want to understand, is it toxic or non-toxic? I already trained the model. The model will know which is having a toxic fingerprints or non-toxic fingerprints. When I give my molecule, it will go and scan and predict and tell me whether it is toxic or non-toxic. So it scores, again mathematics, and it reads, runs the system, and finally you get a biomathematics. This is how we'll be running the things. And you come to know with good and bad fingerprints. And you come to analyze whether it's mutagen or non-mutagen also. So I'm just moving on to the next one. So once you do all these things, we will do even the multiple approaches also. Instead of screening with one target, we go for multiple target analysis. So single agrochemical, instead of it, we'll take thousands of chemicals for formulations. And instead of going for classical modeling, profiling model, having all the systems together with all the chemicals, you can screen and identify which is toxic free. So this is low cost and high yield, that is high cost and low yield. So this is a profiling technique of any organism. You can put it across and you can screen and you can get it. So this is in a case studies. Done. So after that, this is final one. I would like to see what are the chemicals you have taken, it is toxic free. First, next you have to make a recipe formulations. So we use a BioVR formulation design, will help you to understand which formulations virtually you can screen before going for your techniques, wet lab technique, and you can bring the perfect formulations, which will help you to go more on. How do you do this? Just going with this. First, you select the formulation, select the multiple formulas to simultaneously check it, and then seamlessly integrate your management of formulations with integrants, and integrate with a scientific notebook to understand more information, and then transfer to lab operation. Instead of 100 experiments, you will do only 3 experiments, will save your time. Then, of course, finally the product should come. If you see this, any products, you should more confirmers. Any customers, you should expect to have a less toxic, and it should not affect your living systems. Any cells is going to interpret and bring a genotoxicity, those products should be avoided, and any recipes that should come to the market in the shortest span of time with a higher efficacy. That's why we will be regulatory and constraints. It's not only agrochemicals, cosmetics, foods and everything, even in ice creams also. Henceforth, look at the recipes which makes toxic free, and also it should be benefit to your systems that will definitely help you to bring a good product in the market in a shorter span of time. And advantages of this, and our life sciences and academic partners. And thank you so much for this organizing committee for giving wonderful opportunity. Thank you so much.
Thank you very much, Divya. That was a wonderful talk. I really like to thank all our judges and all our speakers. And as the judges are going through the tallying, we are going to involve...
No, ma'am.
Sorry, ma. 11 o'clock, time out. Sorry, ma. We'll push again. Okay, Arpita and group.
Bhavani Group of Institutions. Can I have a great applause for the Bhavani Group of Institution?
Yes.
Yeah.
Fantastic.
Please.
Fantastic. Yes, Arpita and group. You got your music. Arpita and group music, please. This is part of the talent search. Talent search judges. Talent search judges, this is part of the talent search.
दो नैन सितारे है चांद सा मुखड़ा, क्या कहना उसका आफरीन। दावत में जैसे हो शाही टुकड़ा, उसके जैसी ना कोई नाज़नीन। शाही जोड़ा पहन के आई जो बंधन के, वही तो मेरी स्वीट हार्ट है। Drop the beat.
दो तोले की मुंदरी ला दे, 3 तोले का कंगना, मुंह दिखाई दे दे फिर तू ले जा अपने अंगना। ना समझ तू चीज पुरानी, चेहरा मेरा पीस नूरानी, देखेगा तो खा जाएगा 100 झटके। पल्लू लटके रे मारो पल्लू लटके, पल्लू लटके रे मारो पल्लू लटके। जरा सा टेढ़ो हो जा के आवागी तन पढ़ के जावागी, तुझ पे नजरों ब्लॉक हो गई मेरी। हद हो गई डिस्टेंस की, गल हो गई अर्जेंसी की, बहुत हुई डिस्टेंस तेरी मेरी। कदी ते क्लोज है आ, मैं तेरे हार्ट नु टच करना, ये डटनी पीछे नहीं हटनी, तू एक दिन
गुलाब जइसन खिलल बारू, तू भंवरा से मिलल बारू, करत दिल के कतल बारू, के कर मेहनत के फल बेथलपुरमा सुन ले ओ छोरिया, झूमे नगरिया जब जब ये घूमे। कमर यार तारी कमरिया, कमर यार तारी कमरिया। बेथलपुरमा सुन ले ओ छोरिया, झूमे नगरिया जब जब ये घूमे। कमर यार तारी कमरिया, कमर यार तारी कमरिया।
Superb, superb. I request Dr. Vidya to please give an award to Arpita in group. Arpita in group, talent search. Yes, Arpita in group, please. Thank you very much. That was absolutely wonderful. Thank you, thank you. Wait, you, come on, come on. Thank you. Now do we have the fashion show ready? Now we are ending this session with the fashion show, and after that our judges will give out the best paper of session. Ready? Music ready? Chalo.
नजर जो तेरी लागी मैं दीवानी हो गई दीवानी हाँ दीवानी दीवानी हो गई मशहूर मेरे इश्क की कहानी हो गई जो जग ने न मानी तो मैंने भी ठानी कहा थी न देखो कहाँ चली आई कहते हैं ये दीवानी मस्तानी हो गई मशहूर मेरे इश्क की कहानी हो गई जो जब ने न मानी तो मैंने भी ठानी कहा थी न देखो कहाँ चली आई कोमल काया की मोह माया पुनव चांदन नाले सोन्यात सजले रुप्यात भिजले रत्न प्रभात नुल्याले नटली थटली जशी उमटली चांदणी रंग महाली मी यवन बिजली पाहून थिजली इंद्र सभा भवताली अप्सरा छबेदार सूरत देखनी जनु हिरकनी नार गुलजार छबेदार सूरत देखनी जनु हिरकनी नार गुलजार सांगते उमर कंचुके बोबुड़ी मुके सोसते भार शेलटी खुड़ावे कटी तशी हनुवटी नयन तलवार
Wow, RV's College. Really done us proud. Please come back. RV College, please come back. I request Dr. Dayanand from NCBS to come and give the award to our beautiful dancers. Please.
Sir.
Judges, are you ready? Okay. Vidya. Bhuvna. Thank you all very much. Once again, one picture, please. Come forward. Thank you very much, RV College. Wonderful, wonderful performance. Thank you. And now I request, don't go away. Sir, don't. I request our judges, please, to come forward and all our speakers to come forward. Yes, please bring the best paper of session.
So thank you for the opportunity. And for this session, the track 4 session, we have adjudged 2 faculties for Best Faculty Award. So it's Dr. Kripakaran and Dr. G.V. Brinda Devi. So congrats for both of them.
I have 4 faculty awards, Vidya. I have 4 faculty awards to be given to members who have not yet received their faculty awards.
From the
I
Etiraj
think
College.
you didn't receive, right? Please, faculty award for Dr. Saravanan.
From Ambedkar College from Chennai all the way.
Superb, superb. And to Dr. Kripakaran. Bhuvna, go there to Shakila and here.
From the
Shakila
people from Etiraj College, I think so.
Madam, this does not end our performance. Come.
You, once again.
I didn't know. Prettiest Lady Award goes to Dr. Vidya. Okay, come. Ready?
Just a minute.
Manguma?
Yes.
All ready? So this is our signature style of ISGCT to say thank you. Yes, sir, Dr. Dayanand, come on. Ready? Thank you, everybody. And bow to you. Thank you. Now we go for a little tea and coffee break. Faculty, your tea and coffee is here. Students, your tea and coffee is in the shed. But please come back in 10 minutes. We will start the program ASAP. Thank you.
Good afternoon, everyone. And now I request all the judges, chair and co-chair to take the dais. May I request Dr. Kripakaran, Dr. Vijayata Gunamali, Dr. Brinda Devi, Dr. Shakila and Dr. Dayanand to take up the stage for chairing, co-chairing, convener and judges. And I would also like to invite our speakers, keynote speaker Dr. Bhuvaneshwari Meghanathan, Dr. Krishna Kumar Sugumaran, Dr. Leelavati, Dr. Srilata, Dr. Shyam Prasad, and Prasanna Bhagavan. Please take up the dais. Let me introduce to you about our judges, chair and co-chair, the team who is going to judge the best speaker session. Dr. Kripakaran is a leader in high growth business management sciences, biopharma domain at domestic and international markets. Welcome.
She has been.
Excuse me, ma'am. Just before I take present,
Yeah,
my
yeah.
name is there.
Leelavati. Instead of Leelavati, Shubha has been shifted here.
Okay.
Kindly
You have to call.
Okay. Instead of Leelavati, make Shubha. And we have Dr. Vijayta Gunamali, Chief Operating Officer at Oro Biotechnology, Dr. Brinda Devi, Associate Professor at Queen Mary's College, Dr. Shakila, who has 17 years in toxicology and biochemical analysis, has developed a strong research environment in zoology department. She is from Ethiraj College for Women. And Dr. Dayananda is associate with the Campus Medical Center at the National Center of Biological Sciences, Bangalore. Now, let me invite Dr. Bhuvaneshwari Meganathan. Madam, you... Yeah, Dr. Krishna Kumar Sukumaran. So there is one change. Instead of Dr. Leelavati, we will be having Shubha on the stage. Dr. Srilata, Dr. Shyam Prasad, Dr. Shubha, and Prasanna Bhagwan. Now let me invite the keynote speakers. I would love to invite Madam Dr. Bhuvaneshwari Meganathan, and I kindly request you to meet up the time of 10 minutes.
I have given the presentation, AML. So, good morning to one and all present here. I want students to come over here. The students who are standing outside. You had tea? So, good morning. Those who are sitting in the top, good morning. Okay, thank you for your presence. I thank wholeheartedly for the opportunity given by Dr. Sunita Rana Agarwal and Dr. Pankaj, that is from Agarwal's Hospitals. And I was associated with them for the past 5 years, maybe 6 years. And every time I come here, I can see the students who are interestingly participating in the conference. So kudos to the whole organizing team. And let me start with my presentation. So my presentation deals with some basics and some advanced techniques. Hope you will enjoy. So I have just chosen this emerging trends in biotechnology as my topic, in which the next slide, I can have it. How to operate it? Next, okay. Thank you. So emerging trends in biotechnology for human healthcare, in which I am going to concentrate in this AI and ML, that is in gene therapy. So before getting into it, I just want to have some brush up knowledge of gene therapy, how it is happening before, and then we can shift our talk to AI and ML in gene therapy. So myself, Dr. M. Bhuvaneshwari from Sona College of Arts and Science, all the way from Salem, Tamil Nadu, to look all your young minds and to interact with you people. So getting into gene therapy, for the past 1.5 days, everybody is saying gene therapy, stem cells and cell therapy. So gene therapy as it's an everlasting field, because from the birth of the child who has been affected by genetic disorders, people are thinking about gene therapy, how to rectify it. So it's an emerging field of biotechnology, which is used to give ultimate cure for the patients from the dreadful diseases. Okay, so normally, for example, no medicine has been discovered, like an infectious disease or any other diseases. We do have medicines, but for this gene therapy, no medicines has been prescribed. Okay, so there's a question for this gene therapy, that is, are we treating the cause or the symptom? Okay, so if we treat the cause, then there will be a medicine, right? But we are always treating a symptom. For example, a child born with type 1 diabetes, she has to be administered with the insulin hormone for her lifetime. But just hopefully, if our gene is corrected, then automatically there is no need for the medicine. But how this can be done? That is a big question mark, right? So, in this accent, the gene therapy is a technique or approach which is used to administer useful gene or to rectify the error in the gene. So gene therapy aims to fix the correct gene or to rectify the errors in the gene, and this can be done in two methods: germline gene therapy or somatic cell gene therapy. We don't prefer germline gene therapy because we have to do the changes or to rectify the errors in the embryonic or in the reproductive cells. If there is any change in the gene therapy, then automatically what happens, the whole generation is affected. So people are opposing for this germline gene therapy. So everyone is concentrating in this somatic cell gene therapy, which is possible, but not to a greater extent. So three main methods of administering the gene therapy can be ex vivo, in situ, and in vivo gene transfer. So we are going to see how it happens. So ex vivo gene therapy is a technique in which the target cells are removed. I want students to listen. Excuse me. So target cells are removed and they are cultured in the laboratory, and then we are going to mix the gene along with the vectors and administer to the target cells, and those target cells can be replaced into the individual. So this is ex vivo, taking the cells out, treating and administering the gene of correction and then giving it back. This is ex vivo. Whereas there is in situ gene therapy is administering the gene correction of whatever, maybe CRISPR-Cas9, whatever it may be, even RNAi, everything can be mixed as a drug and it can be injected on the spot, that is to the correct organ itself. That is in situ, which is very, very impossible. Okay. And another technique, they can go for in vivo gene therapy, in which as a medicine, if we have fever or headache, we just inject medicine into our blood and it goes and correct the illness, right? The same way, scientists are attempting to go for in vivo gene therapy. You develop a gene corrected or whatever the material, you inject them, it go and rectify on the spot. So these are the three main types in which you can just see the comparison of in vivo gene transduction and ex vivo gene transduction. So inject the therapeutic gene inside, whereas ex vivo, you take the cells out, do all the changes, and then you can inject it back. That's the thing. So there are many modes of gene transfer. Everyone has repeatedly saying. So they are having some vectors, viral vectors, or they have some materials which can transfer the genes to this therapeutic measure, ex vivo or in vivo or whatever it may be. Even we have this CRISPR-Cas9, the latest technology, which can correct a gene, which can cut the error gene, and then it can be rectifying the genetic disorder. So all these things can be done with either of the methods, that is ex vivo or in situ or in vivo. So now coming to this main topic, AI and ML and gene therapy. That is, actually I have given this as my topic, in which we can have a game like this. So everyone would have received a message like this or in a Facebook or in WhatsApp. Just identify the error. That is, I have given a letter of 8 and we have to identify the 6 among that list. I have magnified it so that everyone can see, but it is very tough to identify. And here you can see it clearly. Where is 6? It's there? Yes. So it will be, as you know, it will be a very tough process if you have more lines of 8 and you have to find out the error, that is 6 or whatever may be the digits, 9 or etc. Okay. So just imagine, what is it? What is it? See, if we are given a box, we have numbers and we have to find out the error. That can be easily done. So can you imagine what is this? It is the DNA sequence which has been done by sequencer. Can you find anything in this from this image? No, nothing. We can't say anything. We have no idea of what it is. So what is this then? The same thing in an elaborated way. So, as you all know, our DNA is made up of 3 billion bases, 3 billion letters, if I want to say. So, 3 billion letters are there in our DNA, and genetic disorder is the one in which there will be only one base change or one error base incorporated in 3 billion bases. So, how to identify it? Is it possible manually? Can you imagine a scenario? We have 3 billion bases, and just look at this. Can you imagine to find out you have so many frames, which I have shown only one of the frame with 8, whereas you have so many frames, and each frame is so much complicated with the sequence. Can you easily identify which gene there is one base problem? Can you identify with this? Manually, if you reveal this 3 billion bases to find out a single base change, it will take almost years together to find out where exactly the base changes. But this is simplified with our AI and ML. So what is AI? As everyone was speaking, artificial intelligence, which mimics our brain, which acts very, very faster than our brain. So in this case, if you have a DNA sequence which has been impregnated with the bases, and AI can easily find out which chromosome and in which exon the exact base change is there. Within few minutes, this AI can reveal this sequence, this exon number 3 or this exon number 12 is having your base change. So AI is simplifying our identification process. So AI and ML is a boon for biotech and for gene therapy, as per my idea. I'm myself an academician in biotech. I'm very passionate towards the biotech research also. So I consider this AI and ML as a boon for the biotech field, or exclusively for the healthcare in the gene therapy field, because analyzing and getting the right place is very, very useful because of this AI and ML. So this I have taken as an example. I just mentioned a single base change, in which here you can see that individual C must be there, but instead of C, T has been incorporated in a genome, in a sequence. But how to find out which gene is having this change? So as I said, this AI and machine learning can simplify the process. It can easily find out exactly which gene is having this change. And it can give us, with the algorithm, it can easily find out, it can give the results. We can easily follow the results, and we can do it in the laboratory process. So this kind of thing can be done easily with the AI and ML. So even everyone is telling us CRISPR-Cas9, you can decide which regulatory CRISPR-Cas9 can be easily used, can be used effectively otherwise, for the gene therapy. That can be also done with this AI and ML. So you can find out which RNA, guided RNA can go and bind to it, which CRISPR-Cas9 protease can go and cut the sequence. Everything can be analyzed in silico with the AI and ML. So many gene therapy examples are there. So here I want to give a short Kuti story, in which I said, no, I met this team of Agarwals, Sunita ma'am and Pankaj sir, 6 years before. And we have a meeting, and in that meeting, Sunita ma'am was asking, Bhuvana, I want a program in which a gene sequencing has to be done. I'll be giving you the result of a gene sequence, and you have to tell me 50 prone genetic disorders in future for the patient. I said, what is that, ma'am? Like giving a gene sequence, and I have to get you the diseases which they can be getting after 20 or 30 years. Is it possible? I was asking. And she said, no, Bhuvana, note my words. It's going to rock the whole world with this detection. And she just wanted us to have a gene sequencing in a cheaper model and to find out, to diagnose. It is called not diagnosing, it pre-diagnosis, to find out whether we are prone to any of the genetic disorders or any of the diseases. So that time I was wondering, but now within 6 years of time, this AI and ML is giving a prospect to find out whether we can get this kind of disease in near future. So this kind of diagnosis can be also done with the AI and ML. And Sunita ma'am is a very visionary, I can call her as a visionary person, because she was asking me before 6 years, I want an algorithm. You have a IT team, no? So you build an algorithm so that I can find out what are the changes this person will be getting after 10 years or 15 years. And she wants this diagnosis to be done in a very cheaper manner, that is in within 4,000 or 5,000, which is very, very expensive in the other countries, ranging from 1 lakh to 5 lakhs. So these are the various aspects of AI and ML which can accelerate our sequencing. It shortens the time, and then it reduces the error. Okay, manually we do errors, but machine, they very rarely do some errors in findings. And AI swiftly and correctly pinpoints the genetic variations so that it makes our task very easily in the healthcare sector. And as everyone is talking, personalized medicine, okay, which is possible for each and everyone in near future because of this AI and ML. So these things I just want to say. So the integration of artificial intelligence and biotechnology is the need for the hour. But what happens is AI has very broad impacts in healthcare, as well as the machine learning is accelerating the biology processes. But technology is advancing faster than the policies. So we must have some regulatory policies to fit this technology so that it can be used properly and it can be used for the people. But this is missing so far in India. So from yesterday onwards, we are talking about regulatory affairs. So I need these policies can be incorporated. We must frame a policy, set of policy. Otherwise, to incorporate an AI/ML in developing gene therapeutic studies. So thank you. Thank you very much for your patient listening. Any questions? Any questions?
Any questions? The panelists? Okay. Thank you. Next, I request Dr. Chris to take up the dais.
Madam.
And, yeah. Sir, get ready.
Hello.
Yes.
You are focusing on artificial intelligence in detecting the defect or already developed?
No,
it's in the process.
It's in the process, developing process, because a sequence analysis and all is there, which takes so much of time. Whereas if artificial intelligence is involved in it, we can easily detect exact position of the gene, that base, which can be done within a few hours. That can be done. Because even though we have sequence analysis, it takes time to unearth that sequence and to find out, to compare, and manually it is taking so much of time.
Check.
So if it is incorporated with this NGS, next generation sequencing, then definitely it can be a very time frame. Within a time frame, we can identify it, and that can be used for the analysis purpose.
And we have a lot of challenges when we come across with infertility treatment. Most of the time, the pregnancy will take place in the elderly priming.
Yes.
So they try with the natural method, and end of the day, they conceive. The marriages are late, 30 plus. So in these cases, generally these genetic defects and all more common. We are trying to identify as early as possible with the ultrasound and amniocentesis and all this. So in these cases, if this artificial intelligence helps to detect genetic disorder in the very early stage of the embryo, So that will be a boon for the parents, whether to
So
go for
they
terminal.
are in the way because the hormonal imbalances and why it is not producing the particular amount of hormone, it's all in the same category. There is some defects like insulin. Some people are not at all producing insulin in the diabetic. The same way, some people are infertile because of that hormonal amount is very less. So that kind of detection can be done with AI and ML. And it ease down the process. The only thing is people has to trust this technology and they have to develop it and they have to be framed under a policy. Otherwise, everyone will be having a stick and doing all part of things. So it must come under regulatory affairs so that it can be managed properly and it must be in a safer hands. Otherwise, it develop a risk for the patients as well as for the nation. So definitely, sir, within another 2 or 3 years, this AI/ML is going to rule the world. As I said, Sunita ma'am asking, I give a sequence, you have to detect whether this person is going to get the disease. The same way we can detect for the infertility also. If we know that exact sequence, normal sequence, we can do it with the AI and ML.
Yeah. Already we are doing cervical cancer screening with HPV kit, human papilloma virus.
Yeah, okay.
So what they claim is when we do a routine pap smear, we identify after the malignancy.
Okay.
So in HPV kit, in advance of the 6 months or 1 year in advance, we are detecting the defect genes, which a patient may get the infection. Anyway, it's a good challenge, and Madam has come out with a very good idea of artificial intelligence in detecting the future defects in the embryo. So we will wish her a good success. Students, for you also, it's a beautiful opportunity to go ahead. Thank you.
Thank you, sir.
Vanakkam, Namaskara, Namaste. A very good morning to one and all. Respected judges, my fellow speakers, and first of all, I would like to begin my talk by giving my thanks to first Dr. Sunita Rana Agarwal, who is the chairperson of the organizer for this conference. Second to Dr. Pankaj Ji, Dr. Vidya Niranjan Madam, Bhuvaneshwari Madam, and the whole RVCE team. I happen to remember Anirudh's name and the whole team. And without the audience who are present here, I appreciate each and every student, who are behind this organizing this conference, and my fellow speakers and one of my gurus here, Professor D.V. sir. He's there, I'd like to take his blessings and nice crowd of people. See, I'm going to talk on a different topic. It is about agriculture, okay? But I'll show you some part of genomics work which is related. And I am a molecular biologist by profession, and I work at the Kuwait Institute for Scientific Research, which is a scientific organization in Kuwait. Kuwait is a country which has the highest currency in the world. 275 rupees is one Kuwaiti dinar. We only have oil. It is a desert country. I'll be talking about that later. And Assalamu alaikum to you guys there, it's very, very, I happen to be one of the project director for a very famous project wherein we've designed a prototype to grow plants in space. Sunita Williams is currently working on that prototype because she's just idle there. She has nothing to do over there. And this is the only possible hope for her till February 2025. So I'll be talking about that, and I'll be talking about innovative strategies we follow in how agriculture is done, okay? Again, thanks to the organizers of this seminar first, and this is I can have a mic. Yeah, I think this is better. I'm audible, right? So, probably, I work for the Desert Agriculture and Ecosystems Program, and this is my research. We are a purely research organization. And our ultimate goal is to produce food and feed by adopting international best practices. Imagine I am doing agriculture in the desert, okay? You can see pictures. I'm growing in soilless I don't use any pesticides. Zero pesticides. It's only 100%, I won't say it's organic. We use only what is needed for the plant. And it's all biological pest management. And we recently even touched 61 degrees Celsius, July 17, 2024, 61 degrees Celsius, you know. That is the temperature conditions that I work in. And importance, so half of my research is involved in greenhouse, protected agriculture, anything under protected structures. And the other half is on vertical farming, wherein we use high-tech technology for growing plants in a vertical mode in very extreme conditions. For example, if you take, we tested a lot in the North Pole and the South Pole before we designed this prototype and worked on it, and we sent it to the space in 2019. Okay, so this machine called Veggie is lying in the International Space Research Station for more than 5 years now. And the astronauts first wanted food. Now, so we grow leafy vegetables and all this, strawberries, and now they're demanding they want flowers for them, you know. That's the stage that we are in now. So this is a pilot seawater greenhouse integrated production of, see, you use the seawater to generate fresh water using solar energy, and you grow crops inside the greenhouse in this structure. Then you have, you grow algae, you grow seafood, we grow prawns, everything. So it's an integrated system which is happening. And this is my research facility, which is available there. And you can see the whole controlled environment. See, I have greenhouses. This is a 50,000 square meter greenhouse area, wherein I have different structures, different technologies to work inside my greenhouse. And Kuwait, as I told you, it's a very, very small country in the Middle East. It's called the Pearl of the Gulf. I told you the richest currency in the world. 1 Kuwaiti dinar is equal to 273 rupees. And it's keeping high and high. It's a semi-democratic monarchy. Okay, it's ruled by the king, you know, and the crown prince. And we are a research organization, which is called Kuwait Institute for Scientific Research. And we do only research, no academic things. I'm here, and again, I would thank both Sunita madam and Pankaj and Vidya madam. I want to get into collaboration. I also have a small startup in Chennai, which deals with innovative agricultural technologies. So I'm trying to meet people and recruit, because we have lots of analysis work to be done, which we outsource to people, you know. So I would like to collaborate with organizations and get into people, have youngsters into my team, as well as provide an opportunity for them. So harsh climates, I told you, so we get dust storms. Imagine you can't even grow vegetables at 25, 30 degrees here. But imagine we are talking about 60 degrees. 1 liter of water costs me around 200 rupees in Kuwait. 1 liter of petrol costs me only 22 rupees. So that is the difference. So for me, petrol is not important. For me, electricity is damn cheap. I pay hardly, maybe I pay just maybe 5 paisa per unit. But I pay for 1 unit of water, I might be paying 200 to 300 rupees per, I'm talking about in here. So water is very precious for me. And we have very, very poor soil. The ppm of the soil is going to be very, very high. It's going to be very salty. And partial self-sufficiency, that is why we are talking into protected agriculture. Okay, so how did I land in NASA from Kuwait this was like 10 years back. We went on the base station because Kuwait has the money. So Kuwait holds a 40% stake in that prototype which NASA has developed. So myself, I don't talk, we talk as a team. My team, and we have 5 other teams from Germany, Norway, US, and Canada who collaborate together, and we design this prototype in different aspects. So these are the different kinds of greenhouse structures you're talking. These are all low-tech structures, you know. You have only, you'll have a covering, a plastic sheet, and you'll have a cooling pad. So air is pumped, a fan pumped, and water flows through this and comes out. This is soilless method, so you grow in the soil, you grow plants. Greenhouse with high structures. Then you talk about polyethylene tunnels. So these are protected structures to help you people understand. These are protected structures wherein I use to grow plants. So you give the plant what it requires. You know, it's same like a human being. You give the plant the correct temperature, the correct conditions, the correct nutrients, the plant is going to give you more than what you expect about it. And main crops that we grow is tomato, cucumbers, capsicum, beans, eggplant, lettuce, and strawberry. And these are my developmental research options that I've been working on. Majorly, you know, it would be, these are the research strategy ideas that we're working on. These are the different methods. Again, these are all containerized. See, what I'm able to grow in 1 acre, I will be able to do it in just 1,000 square meters of space. That is what is my policy. My calculation is per square meter, how much of tomato I'm going to get. We have achieved 26 kilograms in 3 harvests, the same in 1 square meter. 1 square meter is just this. I'm able to get 26 kilograms of tomato from a single plant. Just imagine. So that is the rate of technology that we're talking about. So these are cucumbers. We use slabs. These are slabs where we fill them with a mixture of vermiculate, compost, and peat moss, perlite, and other things. Again, soilless culture is, I don't use soil at all, zero soil. Okay, I use a medium which supports my plant for growth. So these are the advantages. So sustainability. Sustainability, we are talking about sustainability, which is the core thing of this conference where they've introduced this. And my research priorities, improvements in cooling efficiency, greenhouse automation, improvements in structure. Then we use solar energy to capture energy because we have sun most of the day, right? But again, there are problems with that. So we are trying to use seawater, use solar energy, convert it into drinking water, and then use it for your plants. This is the greenhouses. This is the advantages of greenhouses. So this is my greenhouse, the initial greenhouse facility, which is in the heart of a desert. Maybe this is in the heart of the desert. You can see we have constructed a greenhouse, and this is how we grow. So you have a fan on this side, you have a pad on the side. So this fan will keep running, so water will keep flowing, and this fan will suck the air. So the temperature inside is controlled. It gives you a cooler temperature. And we have the latest, it's all automated, Every plant will receive 10 a.m., 10 ml of water. I need this plant number 1 to be done, it will be done. It's all highly automated systems. And you can see these are the covers. So whenever I get a sandstorm, I don't want my cooling pad, so I just lower these and it automatically lowers down. So these are the fertilizer tanks. It's computer controlled. It's not like you on a motor, nothing. I just do a computer programming over here. This machine will take care of everything. Okay, I will mix whatever nutrients I want, I'll just give it, which is my formulation, which we scientists develop, because every crop requires a specific temperature, a specific nutrition. It is not that I cannot give the same nutrition that I'm giving to tomato, I cannot give it to strawberry. It depends. So this is how the benches are. Then this is how, this is when the plants are not there. This is when the plants are in full, okay? So this benches hold, this 300 square meter facility holds around, I believe around 2,850 pots like this. So these are different varieties of lettuce, basil, strawberry, all these plants we grow. Okay, see, these are strawberry plants. This is kale. Then you have lettuce varieties, different kinds of varieties. See, over here you see, you have these small tubes, okay? This will give the nutrition, everything for the whole. We call this spaghetti type of irrigation. Spaghetti, like your noodle. It likes like a noodle. So this is again a 300 square meter greenhouse. I don't use soil, okay? Zero soil, I told you. See, again, this is, I have 50 plants. So totally in this space, I have 500 plants. 50 on each row, 5 rows. So each plant will have a water being given to it. Water or nutrition or whatever it is. Then I have monitors, sensors, and other things to control my temperature. Then I have screening levels also. This is when, these are tomato plants kept. This is how we grow. We grow each plant, this is how tomato grows. We use grow boxes. Then, again, you can see tomatoes coming out. These are grow boxes we use. You get these milk packets coming, right? So we fill them with a particular medium for holding the plants and just give it. Then these are different types of, vegetables that I got from, these are products of your own Karnataka. Sri Sri Ashram actually provided this particular thing with these. So these are all bell pepper varieties, chili varieties. Okay, these are all, none of them are tomatoes or something. Then you have red color okra, then cauliflower, and these are different kinds of tomatoes, 8 different varieties of tomatoes. Okay, why am I showing you this? See, it's going to give me the same type of energy, the same amount of whatever I'm going to spend. But this tomato over here costs me 1 kilogram 50 rupees. But this tomato over here, which is yellow pear variety, costs around 300 to 400 rupees per 500 grams. So I'm going to use the same thing. For example, if you go for blueberry variety or if you go for cherry tomatoes, they're very, very expensive. You know, each kilogram costs like around 700, 800, even 1,000 rupees. So you use the same thing, you use the technology, you try to develop a variety which is required by most. And this is what I showed you, you know, different varieties of tomatoes that we grow. Okay, these are all plants which have been tested, trialed in Kuwait, as well as in Canada, as well as the NASA Florida base station, as well as they have been grown using veggie at the International Space Station. That is why I'm showing you these pictures. Different kinds of soilless production systems. I have to run through my slides, you know. I've taken permission from Pankaj for an extra 5 minutes because I cannot complete it. That's why. It's a huge talk. Sorry for that. And relevance of the topic to this area is, you know, I'm into sustainable agriculture and genomics. Again, I'm telling you, you know, I'm in this session, you know. Anyways, hope my talk is interesting to you. You know, this one sips, you know, you have nothing inside this. It's a box where you'll have only water inside. See, you see how tomatoes are growing. See, this is again the sips where cucumbers are grown. These are, we grow flowers and lettuce. So, using aeroponic systems. So these are grow bags, which used to grow tomatoes. These are grow box, okay? Then these are tomato, just a close-up view of thing. This is how the flowers will look when you take it out, okay? No soil, see, nothing is this. This is how we grow. And different systems, you know, all containerized pot system. Then see, these are multi-level stacked layer system. See, these are, again, these are tower system, vertigrow system, which we have developed for strawberry. See, normally you can keep just one plant, right? But here you see there are around 40 plants in the same space that you grow one plant. That is the advantage of it. So I am talking about, I have a company which deals with manufacturing and growing of such systems. We have designed, prototyped, and have made available systems for home, office, and farming purposes. This is a plant factory. And this is greenhouse, you know, these are cherry tomatoes being grown, you can see. This is a very, very small space. This is not even, I think, maybe not even 30 square meters. Just see how many plants. I have around 60 plants inside this. See how the tomatoes come. So this is my greenhouse inner view when we use cocoa slabs. And computerized. I will know which plant over where, what is it doing. You know, I'll know every hair, I have a monitoring system. In and out, it will tell me completely over here. That is where computerization and, life is no longer, you cannot be only computers or biology or whatever it is. You have to have a mix and match of different subjects which will enable you to grow well in the future. Yes, we are all here for a purpose. So live life to the fullest, enjoy, but keep a goal and set it where you're leaving, you know. Quickly, I'll run through. This is my research baby. This is the mother prototype of what we have kept in space. So we call this MAPS. It's Modular Agricultural Production System. And it was initially between us, Kisar, and the University of Guelph in Canada. So it was same. We used this technology concept for life on space. So we thought, if Mars or space is a very, very higher climate, even Kuwait has a very, very different higher climate. So why don't we test such systems at very high climates? So it's completely, I don't do any genetic modifications. I let the plant naturally react. You know, if you give something different, it gives you something different. And my specialization over here is how light and how nutrition affects plants' growth. Okay, for example, if you give only blue light to a plant will grow like this. If you give only red light, it will grow tall. So plants have different characteristics. So this is the research tool I'm talking about, wherein we have 3 layers. So it's a 2 by 1 meter. That's it, this unit, okay? And it's 8 meters in height. If you see this particular prototype, I can grow 162 plants. So these are my specific objectives, you know, look for light recipes, nutrient recipes, and all this. So this is how it looks. 3 layers, 3 different lights I'm giving, and I'm trying to test what is happening. So this is my lab where this mother prototype lies. This is the brain. It's completely, again, computerized, controlled. I can click off my mobile phone. I will know what is happening over there in Kuwait. So that is how I have a firewall to access my system. and know how we do this particular type of, how are my plants responding, what is the conditions that I'm going over there. And just when the lettuce seeds are done, then, so these were all the things that we looked into. Another 3-4 minutes, please. That's it. I finished off first. Yeah, soon, madam, soon. Okay, so these are some things. So this was what made us do this. If it can be done there, it can be done in the space also, right? So if it's done there, space, so that is why we got this technology into place. And our research unit at Florida Base Station, and you can see how the lab over there looks. And this is the prototype that was designed, because in space, you don't have pressure, gravity, gravitational force is not there. So we had to work in hyperbaric chambers where you completely remove the pressure out of it and you see how the plants react over there. So these are the chambers, bars, and then you see, this is the unit. So the plant will go inside that, and it comes, the studies that we did. This is Jamie, the guy who does the light manufacturing recipes for us. For my research, the former Canadian Prime Minister showcasing our unit first during 2019 at the NASA Space Station a bigger facility. Then these are some light studies we did with genomics, okay? And what we did was, see, I sow the seed comes next, I take it and put it inside this. See, this is how it will look when I lift up the tray, and I do my experiments using different nutrition or light or whatsoever it is. So at the end, I come to know that I've given the same light, everything is the same except the light quality. See how the light quality changes the coloration of the leaves. So this coloration is involved again with various biochemical pathways involved with this. And there was a significant difference. These are the results. And some more just pictures of what all studies we do at the root level, at the shoot level and other things. So we did strawberry. These are strawberry plants. Again, experiments with strawberry. We did an RNA sequencing work with IBAB Bangalore over here. In the future, we have some very good UN projects. I would like to interact with Dr. Vidya Madam and her students over here to get into, we want to outsource certain work, contractual research work. So any other person who's open to doing analysis work for us at the genomic level, biochemical level, or whatsoever it is, I'm open to collaboration with you guys. So we had this paper of our work published in Nature at 2022, December, which was very, very unique paper because there was not much work done on how light affects different biochemical pathways in plants. So during Corona, was getting bored, so started growing whatever I could in that particular facility. And I'd like to thank the management of Kisar, then my colleague and friend, Dr. Vinod, my team and staff. There are nearly 57 people reporting to me, and I would like to thank each and every member of my team. Without them, nothing would have happened here. And I'm grateful to BGI Hong Kong and IBAB Bangalore. This is my team. He's my super boss, okay, Dr. Mohammed Al-Baho, and my team of immediate reporting team of people. And thank you. I'd like to take any questions if you have, and thank you so much.
Any questions?
And it was a nice presentation, sir.
Thank you, ma'am.
And it was rather colorful, and it was tempting our taste buds, right? All of us.
Probably you can
we
get
were admiring the colorful
if you want
vegetables.
to taste, probably I can send you some of my products from Chennai, for sure.
And I would like to ask you, you were saying about the remotely accessible Argus system. Were you planning to go into growing plants in the space by remotely accessing to it? Do you mean that?
Yeah, we access the systems which are available there.
Okay. So you will be remotely accessing.
Yeah. See, the system that I have in Kuwait, I showed you that system, right?
Yes.
Anybody who has an access to the firewall over there, to the system, can access what is happening over there.
Oh, that's great.
So awesome. We had people from the NASA space station from Florida, then people from University of Guelph in Canada, and us. We were the 3 people who will be able to access this. So I will be able to use it. I have an app on my phone, specific app, so I'll be able to use it. And what happens, the same thing applies to the system that is kept there also. So probably it is daytime for me when it's night for Canada. So usually they monitor, and whenever any small mistake happens, when you enter these parameters, it sends you an alarm. If nobody responds, it will send you an SMS. Or it will send a call, distress call to the SPOC, the single point of contact, who's in charge of the system. Okay. So if something happens, something is going wrong, and any parameter which I have set is not working, suppose the electricity fails or something, it will send an alarm, it will send a SMS, last final distress call, it will call me. Because I am in charge of that particular unit. Same way, different labs, different people. And we have to get it corrected, make sure that it does not falter. Because always remember this technology and everything, it's all colorful to watch, nice. It's a never forgiving machine, okay? It's a never forgiving machine. It will give me everything I want. But day 2, day 3, if I don't monitor my pH levels or my EC levels in my nutrient solution, or if I don't monitor my temperature levels, that's it. Again, I'll have to go to stage 1 back. So this technology of hydroponics and soilless cultivation is a never forgiving technique. Always keep that in mind. I would suggest, not advise, all students here, write what you do. I still write. I go to my lab and I write. Morning I went there, I saw this, because that will give you the strength to later on go back to your problems and see what happened. So always, if you are a science person, write and record your observations. You have a strong observation book. That is what I would say. Thank you. Hope I answered your question, ma'am.
So I got a question for you.
Yes, ma'am.
It's not by a natural means the plant is not growing there. It's all that your man-made things.
No, madam, it's natural.
Soil and
everything. It's purely natural.
It's purely
nothing.
natural.
I'm not even doing any genetic modification.
It's growing in a very compact manner.
Yes.
In a concealed area where it is not being gained by the exact sun and the soil.
What
you are putting them into the
plant. What I am doing is I'm giving them an artificial environment.
Yes.
I'm giving them an artificial light.
Okay.
I'm giving them a medium to support. Fine. And I'm giving the required nutrients what the plants needs.
But what will be?
Those are all small weed-like plants.
My question is...
I'm talking about 60 degrees in summer, and it will go to 3, 4 degrees, 0 degrees in December. The same happens. And we get frequent dust storms, madam. It's very difficult. Humidity will be 0.
So what will be the nutrient content of it? If the humankind is going to take up that tomatoes which has been growing here, and it's all about the globalization where you can get tomatoes and it can be exported also. But still you are growing there and doing some work on it. Will the nutrition be affected?
What my work is?
Yes.
My work, I have to enhance the quality, which you talk about, and quantity. Okay, because I'm going to give, it's same. Granite is a big stone, but not big. Diamond is a very small stone, but it has lots of value. Same thing. We have a strawberry plant, which we do using light and nutrients. We enhance the vitamin C content and give it there. So we pack it, we create using artificial means, but you make the plant respond naturally and enhance the quality of it. We have 100, not 200% very strict quality policy because we check for the nitrate content, we check for the oxalate content, everything, any other heavy metals, anything which gets usually absorbed or whatever it is. And I told you, I don't use 0 pesticides, no pesticides at all enters my greenhouse right from the time, whether it's a greenhouse or in the vertical farm as well.
A small addition, sir. You can try vermicompost also to it. That would be a better suggestion.
We do actually do vermicompost.
Fine.
I told you, compost is a very component. I don't use soil, but I use vermicompost.
Thank you, sir. All the best for your good work.
I think Israel Technology is the same one.
Yes, sir. Exactly.
They are using the system, and the system is fed with the nutrient availability to the plant. How much zinc should be supplied, how much they detect the deficiency. As per the deficiency, water and nutrients will be supplied to the plant.
Exactly, sir.
So that's how the nutrition value of the plant will be retained. Am I right?
Yes, sir.
Yes.
So, he was asking about disease management. You get sometimes we get tip burn in lettuce, then you might get blossom and rot, for example, tomatoes, which you use using calcium. Correct time you give the calcium. You have to do. But I use stringent even if I'm going to use a pesticide or something, it will be a biological method. I use another insect to eat another insect.
And,
that is how we use.
Dr. Krishna Kumar, sir.
Thank you.
Speakers are waiting here. We'll have this panel discussion after the...
I think...
Thank you so much, sir. That was a wonderful presentation.
Let me introduce Dr. Srilata, Professor and Head at ESI, President for Indian Society of Blood Transfusion and Immunotherapy. She is a COVID warrior and Lifetime Achievement Award from ISBTI. And she also worked as Professor and Head of Immunotherapy and Blood Transfusion at Victoria Hospital previously. Now I invite Dr. Srilata to give her speech. And please, we'll stick to 10 minutes this time. Sure.
Thank you very much, Mrs. Vidya, for giving the good introduction. This is the one, right? This is not audible, I think. This is the right. This is the one, right? Good morning, everyone. Here I come from, I think I should thank Dr. Sunita Agrawal and Mr. Pankaj. I'm here from the first conference which took up, and this is the platform which they have created, wherein all the agriculture, the medical people, and also the engineering, this is the only platform I feel that comes all together for the betterment of the future of you, future citizens. So let us give a big, loud applause for the organizers who have taken this stall every year to bring us all together all over the world, not only here, all over the world. So my talk is something different from nanotechnology to the very basic one. So I come from the background of immunohematology and blood transfusion. Of course, it has reached to the extent of the nano, wherein the regenerative therapy and all these things, it comes under my umbrella, the study. But without the basic root, which has to be very strong, we cannot reach to the extent, we cannot reach the sky. So that is how I have come across. And you all know, can you please tell me which is at present, which is a disease which instantly kills us in today's world? The topic is there with that of the cardiac arrest. So it is not the cancer, it is not something else, it is the cardiac arrest. Many of us, I think, last 2 weeks, we have lost one of the medical students, just 19 years old. So with that of the heart attack. Is that the way that we need to lose them at that younger age? No, it is not. It is at the present status, we are losing. It is not taking the age, it is not seeing the differences between the gender, nothing of that sort. So in this world, I thought this is the topic which gave insight and opened my eyes to. That is what I'm going to share with you. And it is whether you are, I'm seeing in the audience, whether you are a medical student, the nursing students or the engineering students or otherwise the other BCom students are also here. I do find them here. I have interacted with you people yesterday. I'm glad that you are all here. But the basic thing is, without having the good health, how can you proceed with your research work? So to have that, I have come here. So this is what is the building. And this is the study which opened up my eyes to, and that is how I started my, of course, being healthy. So what do you think that my age could be? Yes? Yes? Take a guess. Which number do you like? Sorry, 80? Yes? Yes, so please tell. There's nothing wrong in it. This is not, I'm not giving you any marks or something like that. You can go ahead and tell the wrong thing also. Yes, 30. Something is showing 3, 4, 40. Okay, no, I'm sweet 60. 60, not 16. Sweet 60. So I have just recently retired from BMCRI, the Bangalore Medical College and Victoria Hospital, and joined to the ESI. So how am I healthy? So how I adopted and why I adopted. At the age of 50, I took up this blood donation. So why not you people? So this is the article which shows that, which gave me an insight, which opened up my eyes. And this is what now the cardiac arrest is more, which is causing the 85% of the death, which is the cause for the death in today's world. So should you succumb for that? So that is a research that tells you. So if we go on to this article, so it says why and what. It is not there in any of the curriculum, but it is there in this, the original, the article, which was been studied. We being in the medical field and then the college will be going through what is the latest which is happening around the world. And that is how I found this is very interesting. So this is, in this article, it says that the cause is there within us. Okay, the one, all of you must have read about the red blood cells. So the red blood cells, the lifespan of the red blood cells is just 120 days. So this 120 days, will it differ from that of the person who is in US, the African or the we Indians or Chinese or anything? Or is it taking a difference from that of the gender difference? Or is it taking a difference from that of the, whether you be being as a B negative or O negative or A positive, B positive? Nothing of that difference. So for everyone, it is 120 days itself. This 120 days is the cause for the heart attack. So this 120 days, the cell is biconcave. One of the speakers spoke on the sickle cell also. So the sickle cell, yes. So all these things, it will have the cytoskeleton, like how we have the skeletal structure. The cells, each of the cells in our body have the cell, the cytoskeleton. This, the 120 days, which is growing old, it cannot squeeze in into the very tiny blood vessels, which are there as thin as your single hair strand is. So here is the pathology where what happens. It cannot squeeze in. So I have my restrictions. When I go into the, if I happen to go into the cave, I cannot crawl and go. So if you, the people who are behind me also get stuck. So if you allow the 7 years boy or someone, he'll crawl and make the way for the others. So in the similar way, the pathology happens at the vasa vasorum. So this vasa vasorums are the blood vessel to a blood vessel, bigger blood vessels. They are in millions in our body. So this is where, when the blood stucks there, it cannot have the spontaneous blood flow. When the spontaneous blood flow cannot be there in the very sensitive organs, that is heart and the brain, so it brings about the, in the form of the heart attack. So do you want this or do you want to avoid? So the study went on to say, yes, what is the solution? So the solution is nothing but having the blood, the new blood cells often. So according to this study, so by donating twice a year our blood, so twice a year, we can reduce the incidences to 80 to 88% of this heart attack. Do you want or not? You think about, you're all grown up, you're all 18 years and above. Of course, that is what the criteria I'll be coming across now. So how to reach? Just if you say, yes, come, we can draw, no. You should come to some criteria also. So anybody who has donated blood here in this, so big, 2 hands I can see. Yes, oh, good, good, good. Give them a good, big, loud of applause here. So thank you, everybody. At least a few hands, about 5% of the audience are there who have donated the blood and experienced it. It is not painless. It is as sweet as an ant bite. Is that so, sir? So don't you fear for that, that there is a needle and it will be a painful procedure. It is just 5 minutes it just takes, and not all the blood. In our body, just 6 liters of blood will be there. Out of that, we'll be drawing just 350 to 450 ml. That means to say your lassi glass of blood itself. Okay? So the criteria stands as whoever is 18 years and above till 65 years. I have already donated twice, so I'm planning here now again on October 1st, which is coming up as a National Blood Donation Day. So the weight should be 45 kgs and above. The hemoglobin, irrespective of whoever it might be, it should be 12.5 grams and above. Not all the females of India will be reaching to this hemoglobin level. We can improve the weight and as well as the hemoglobin value. So blood group, everyone, all of us, whether we are negative blood group or positive, we should donate twice a year. Then only we can get the effect of reducing the incidences of heart attack and stroke also. So this is where it helps all. This is, okay, I'll just skip this. The criteria goes on. The BP also will be checked, which should all be normal. The sleep, at least 6 hours of normal sleep one should have. And of course, you should avoid the tablets and the habits like smoking and alcohol for 24 hours. Yes, the heart attack, you can see that it is 88%, it will get reduced. And cholesterol, of course, it will be 44%, it will be taken out from the blood if you are becoming a blood donor. And blood loss and others like liver diseases and cancer also gets detected. Like early days, it is not so. Like earlier days, the blood used to go from one person to the other person. The older movies, have you watched the older movies? Amar Akbar Anthony, if you like. If you watch it, earlier movies, one person used to donate, the other person used to receive and become bhai. Like that, it is not so. We need to take this blood, the donor's blood, and detect for the other diseases, major diseases like HIV, HPSAG, HCV. It takes nearly 1 complete day to detect the diseases. It's not like give it away instantly to the patient. So it gets checked up there, that 1% of the donors also get checked up whether they are in the precancerous stage. So is it not a benefit? If they were to be a precancerous state, their treatment and all these things, it becomes very easy. The patient will recover fast and his lifespan will be probably from 15 years to 20 years, he can stay with us. So this is that video. Of course, you must have watched that video. It never it never exempts us from any profession, whether you are a journalist or a singer or whoever it might be, it's all the same. So that was about that. And the effect you can see, yes, what is the effect of this blood donation? How many lives could it save from what we donate 1 unit? What can you say about that? How many lives can we save? Yes? Take a chance again. 1, 2, 3, 3. Okay, you stand for 3. So it is not so. In today's world where the technology is ahead, it is saving at one time of donation, it is saving nearly 10 to 12 lives at one given time. Don't you feel happy about it? Can we save so many lives at one time when we donate any other parts of our body? Can we donate any other parts of our body while we are alive? That's the main thing. Yes? Can we donate? Yes. Kidney we can donate, but we'll have a lot of after effects about that, BP, sugar and all these things. I think it will shoot up since one kidney has to take up that load. So you will get to have all the BP and sugar, all these things, problems within a short time. So such sort of side effects is not at all there in this blood donation. So you'll have all the benefits only, not just saving somebody else's life, nearly 10 to 12 lives. You will have nearly about 18 days, 18 diseases. Your body says, if your blood has gone to the other person, that means to say your body is free from 18 diseases, free of cost. Do you want or not? If you get tested, you'll have to spend nearly 5,000 rupees. So this is free of cost. You will have the gratitude. Today is a International Day of Gratitude. So you will have that gratitude also. You will have this is Peace Day also today, and Alzheimer's Day also today, International Day of Alzheimer's, International Day of Peace, International Day of Gratitude. So all these things you will find it once you start becoming a good donor. So do you want or not, you're all grown up. So whatever you might take up your profession, future profession, you will have to be strong enough. So once you are strong enough, then only you can go ahead with your dreams. So this is what I say. The need for the blood is, of course, too much. Nearly 16 to 18% in today's world, but it is happening just 1 to 2%. Of course, the sickle cell anemia and other thalassemia, they need it daily. Okay, it's a genetic encountering disease. So here is a solution. So one who have, what I did was, that's what I'm sharing with you. So you can see my thyroid, I'm having a thyroid problem also. I am a thyroid patient also. So my hemoglobin was fluctuating between 9 to 10 most of the time. So what I did to become a donor and what I chose and why I chose, like how you can also adopt it, that's what I'm going to share. So I chose this beetroot. All of you must have tasted beetroot. Do you all like the beetroot? Whether you like or not, it is not a bitter ground. It's a tasty. You can make it still more tasty by putting the lemon juice, which is ascorbic acid, which helps in absorption, more absorption. You can put a little bit of salt, sprinkle salt and pepper to it. 5 minutes, you can eat it. If you like sweet jaggery water, you can sprinkle it and leave it for 5 minutes and consume it. So the next one is, I did not chose the ones which are there, that side that...
Ma'am, you may have to wind. Fine.
Oh, it is going back. So why I did not choose the other which are there in the market or which we can consume. We all need to cook. We are not carnivorous. We are not herbivorous, totally. We are evolved. We are evolved to that extent that we need all masalas and all these things, fries and all these things. We cannot go ahead with anything else. So that is why I chose this part. So the next one is ragi porridge. This next alternative you can give us, the ragi porridge. Ragi balls will not help much. So next one is the dry fruits. This I took up for the MNCs. MNCs, we used to go for the camps where every alternatively, but they were not able to donate the blood. So I suggested, of course, before Corona, they were all being picked up by the cabs. So at the midnight, we cannot suggest them ragi porridge or the peel of the beetroot. Raw beetroot you should consume 100 grams. So this is what
Dr. Srilata ma'am, you may have
1
to
minute. The main thing.
Yeah.
Main thing I'm coming up. So
please wind up in the interest of time.
Yes, this is the last slide.
Okay, thank you.
So you don't want the mass to be healthy?
No, just
so
want to
you
give
want
it.
the healthy, yes.
Just want
I'm also there, I can understand. So this is the last one. So the dry fruits, when I took up, I do not pick up the pista or the badam, which are rich in the fat. So I chose in the formula of 5:15:15. So 5 is the anjir, the dry fig. Another 5 is the dates. 15 number of dry resins and 15 number of the almonds. So all put together, when I gave them this solution, they were ready to donate the blood by the time we went next time. So this is what I chose, and you can choose one of this or in combination of this to be healthy, and for the future, your endeavors, you can go ahead with it. So this is what the myths and facts. It is not painful at all. It is sweet as an ant bite. So this is how the processing happens. It is not just the color we give it. So it needs a lot of efforts to be done. So this particular doctor intern, she came to the level of blood donation within 19 days. So you can also do it. So camps we do regularly, not all the, even temples, mosques and churches everywhere we do go. So you can see the physically challenging person on the right side who has also donated. So this is the athlete. He is a football athlete who has donated. So this is how we celebrate World Blood Donors Day. So thank you everyone. So you can have a good health.
Thanks for understanding, ma'am. And in the interest of time, we are not going to take any questions. And now I request Dr. Shyam Prasad to give the next.
Thank you, ma'am. I thank the organizers for giving me this opportunity.
Excuse me, sir.
Yeah. So my presentation is about pigment, microbacterial pigment. My presentation is divided into 3 parts. One is isolation of bacteria and identification, production of pigment and its applications. So I'll be taking 5 minutes. No, I have to show that. Okay. So today textile industry is making a business of around 9 billion US dollars with an average dye consumption of 8 lakh tons. You know, these dyes are toxic, they are carcinogenic, and they are released into water bodies. So they pollute groundwater as well as they contaminate water bodies, and they accumulate in aquatic life. From aquatic life, they indirectly enter into human beings and pose health issues. So in this way, textile waste is discharged into the environment. So here, the industrial people are looking into natural colors because natural colors are eco-friendly, they are stable, and among natural colors, microbial pigments are very stable as well as they grow on cheap media and their generation time is very less, and also ease of genetic manipulation. So here we can see some of the examples of pigments already available in the market. So these are the pigments and these are the applications. So in our study I have selected prodigiosin. Prodigiosin is a red pigment which has many biological activities like anti-cancer, anti-malarial, the list goes on. So despite many biological activities, so the application is very limited because of high production cost. So researchers are working to reduce the production cost. So this slide shows the suppliers of prodigiosin. So on average, the price of prodigiosin is 25,000 per 1 milligram. Okay. So here we have isolated an organism capable of producing prodigiosin. So this is the slide which shows isolation. And this is the prodigiosin pigment. So this is prodigiosin when plated. So the bacteria which we have isolated, identified as Serratia surfactans. So we have deposited in gene bank with accession number. So then we have taken some synthetic media from literature and we have gone for production of prodigiosin. So we could get peptone glycerol broth is giving very good results of 5098.5 units per cell. So this is the yield of prodigiosin in synthetic media. Then we have used agro waste to reduce the cost. So when we have used agro waste, this waste we got from oil mills after oil extraction. This is no use for anyone. So here, by using solid state fermentation, we have gone for production of prodigiosin. So among different agro waste we used, so this black sesame is giving very good results. So peptone glycerol broth is giving 5098 units, whereas black sesame is giving 69,000. So this is the difference of using agro waste. So here we can cut down the cost. So here we have gone for mass production. We didn't go for scale up. We gone for mass production. So here this is the pigment after extraction. So after purification and all, we got the pigment. So then we went for structural elucidation by using different spectra. So this is structure we got. Prodigiosin has got different derivatives also. So we got, this is the structure. Then we have also used for synthesis of nanoparticles. So by increasing the concentration, we got nanoparticles also here. So this is the UV spectra of that. So then we went for application. So here we went for dyeing of different fabrics here. So in the first row, we have selected different fabrics. First row is application of only prodigiosin, and second, third, fourth, fifth row is we have used different binders. So when we went for washing, so without binder, we lost the color. And where binders are applied, the color is retained here. So we also tested antimicrobial activity of this dyed fabric. So we can see, against clinical isolates. So we got almost 90% reduction in bacterial growth. Okay. So this is antifungal activity, 50% reduction in fungal growth. So these are some pictures. And also we have studied in silico study, anti-eczema activity by using in silico studies. So this is atopic dermatitis. Proteoglycan is showing good results against atopic dermatitis also in silico studies. So by using this proteoglycan, we can also use for designing of antimicrobial shirts, underwears, moss for controlling of skin infections. Not only for dyeing, not only can be used as a dyeing agent, but also can be used for designing of antimicrobial fabrics. Then we also designed antimicrobial soap by using only proteoglycan. And when we have tested this, it is also giving very good results against bacteria as well as fungus. So these are the results. Then we also gone for Band-Aid. We also prepared Band-Aid by using proteoglycan silver nanocomposite Band-Aid. So by using nanoparticle proteoglycan nanocomposite, we have prepared Band-Aid. This Band-Aid is also working good against clinical isolates, bacteria as well as fungi. So control Band-Aid has not given any inhibition, whereas our prepared Band-Aid is giving very good results. So this is a picture. So we also prepared proteoglycan nanocomposite ointment. So this is ointment. So this is also giving very good results against clinical isolates of bacteria as well as fungus. So we also prepared Band-Aid. So this can be used for infections of the wound. So scope of proteoglycan is we can prepare antimicrobial fabrics, we can prepare Band-Aids, bandage, ointment, proteoglycan soap. So we recently have seen proteoglycan is also inhibiting viral replication, that is herpes simplex virus. So we can also prepare eye drops to control this virus also. So we are collaborating with this micro-tech company for further studies. Thank you.
Thank you so much. Any questions? Yeah. Any questions? So if not, let me invite the next speaker, Prasanna Bhagavan.
Hello everyone.
कहते हैं लोग पर मोम सा है मेरा जानू नश्तर है वो यही दूर दूर गूंजे फितूर पर बादशाह है मेरा जानू.
Since I could grab your attention, thank you so much. I won't take more than a minute. My topic was on patenting and how to think. But I know you people are tired, so am I. Thank you so much for coming and listening to all our lectures. If somebody is interested in learning about patents, I will be available, I will be around. Let's talk about it. Thank you so much for the opportunity. If somebody wants to listen to the complete song, probably I'll play it.
Yo, boys, I am singing sound, shoot sound, flop sound. Why this cola very deep? Why this cola very deep?
Coke Studio, Season 14.
Can anybody guess this song?
हाँ तेरे जावा मैं पूरी नू आना जियो नहीं आया दिल बांग बांग मेरा टकराया कागा बोल के दस जावे मामा क्यों दी चू.
Thank you so much.
Thank you very much, Prasanna Bhagwan. Wonderful talk. I'm requesting now our judges to give us the best paper of session. In the meantime, we are calling Rumki Group, correct? This is part of the talent search and talent search judges. Thank you. Talent search judges. Rumki Group to perform.
झिम सन 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 हवा टिप 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 बूंदे गुर्राते बिजलियां
भीगी भीगी साड़ी में यूं ठुमके लगाती तू जैसा फिल्मों में होता है हो रहा है हूबहू.
आई 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 जुबी डूबी डूबी पंपारा जुबी डूबी परांपा
जुबी डूबी नाचे क्यों पागल स्टूपिड man.
लड़के और लड़के कहाँ से आया है रे तू प्यारा है शक्ल से अकल का मारा है रे तू लड़के और लड़के कहाँ से आया है रे तू प्यारा है शक्ल से अकल का मारा है रे तू.
हाँ चल गई मेरी बातों से तू चल गई हाँ खल गई तुझे खल गई मेरी बेपरवाही खल गई ओ धर्मा तू किस खेत की मूली है जरा पता.
टीवी पे ब्रेकिंग न्यूज हर मेरा घाघरा है बगदाद से लेके दिल्ली आगरा टीवी पे ब्रेकिंग न्यूज हर मेरा घाघरा है बगदाद से लेके दिल्ली आगरा. साया जब से मिला तेरा साया भाया मुझे हर बार तू भाया लाया सपने हजार तू लाया ऐसे ऐसे हाँ ऐसे ऐसे
Wonderful. Once again. Rumki, who's Rumki here? Thank you very much, Rumki. Here is so this all is part of talent search. Lovely. Wait. Take a bow. I'll ask Mavia to come on stage as this is the next performance. Yes? Madam. Yes, Mavia, please come.
Good afternoon, everyone. Check. Good afternoon, everyone. My name is Maya Ashraf from Shri Bhavani Group of Institutions, and I am delighted to share a special song with you all today. This melody is one that holds a dear part in my heart, and I hope it will resonate you as well. Please sit back, relax, and enjoy. हमारी कहानी लिखी हुई है. ये दुनिया क्या, दुनिया के लोग क्या, हमें कोई अलग नहीं कर सकता. It's all about a Hindi song, so please enjoy it. तुम नजर में रहो, खबर किसी को न हो, आंखें बोले वो लब पे खामोशी, आंखें बोले वो लब पे खामोशी. तुम चलो जिस डगर, मेरी राह गुजर, आंखें बोले वो लब पे खामोशी, आंखें बोले वो लब पे खामोशी. दूरियां कम कर दे, प्यार का अंबर दे, जुल्फों का वो आसमां, बस आँखों पे हो तेरी तमा. सुबह रात हो, नजर मिले जरा बात हो, खुले मौसम तुम नजर में रहो, खबर किसी को न हो, आंखें बोले पे खामोशी, आंखें बोले हो लब पे खामोशी.
Ready? This is all part of the talent search. So we've already finished these two, and now this is the third. And then we all break for lunch. But please get back here by 2 o'clock for the next and last session, where we have free papers as well. Come. Music not ready. Music.
இந்த மண்ணு மணக்கிற மல்லிகைப்பூ நம்ம மனச எடுத்து சொல்லும் வந்து நின்னு ரசிக்கிற ஊரு சனம் இந்த தேர இழுது செல்லும் மதுரை குழுங்க குழுங்க நீ நையாண்டி பாட்டு பாடி குழுதி பறக்க பறக்க நீ போடாத ஆட்டம் போடு இந்த மண்ணு மணக்கிற மல்லிகைப்பூ நம்ம மனச எடுத்து சொல்லும் வந்து நின்னு ரசிக்கிற ஊரு சனம் இந்த தேர இழுது செல்லும் தன்னாரே தன்னாரே
তোমরা দেখো গো আসিয়া কমলা হেনে তো করে থমকিয়া থমকিয়া তোমরা দেখো গো আসিয়া কমলা হেনে তো করে থমকিয়া থমকিয়া এই গো কমলা হেনে তো করে থমকিয়া থমকিয়া এই গো কমলা হেনে তো করে থমকিয়া থমকিয়া রে কমলা হেনে তো করে থমকিয়া থমকিয়া
అరే దేవుడా పిల్లడాట దగ్గరై నన్ను చేరదీస్తివే బుట్టబొమ్మ బుట్టబొమ్మ నన్ను సున్నుకుంటివే జిందగీకే అడ్డబొమ్మై జంట కట్టుకుంటివే బుట్టబొమ్మ బుట్టబొమ్మ నన్ను సున్నుకుంటివే జిందగీకే అడ్డబొమ్మై జంట కట్టుకుంటివే
तुझे यार दिल में बजी गिटार देखा तो तुझे यार दिल में बजी गिटार छलका आंखों से प्यार दिल में बजी गिटार आ रहा कैसा ये नशा रे आ रहा जीने का मजा रे अरे दिल में तो गया दिल भी गया रे अरे दिल में तो गया दिल भी गया रे
Thank you, thank you. 1 minute, 1 minute, 1 minute. And now I'll ask our judges to come forward. Yes, madam. Please, get up, get up. Yes, thank you very much. There was a wonder. Thank you, thank you, thank you. Please. Thank you, girls. Thank you very much. Wonderful, you just changed the platform of the convention. Look at that, the whole audience was dancing with you. Thank you. Please, I request our speakers also to come up. And now is our time to give the best paper of session. Bhuvna, please. Ama, correct. You give one and he'll give one. Yes, Dayanand, please. Yeah, you announce first. Okay. Yo.
So here for this session, we are announcing the best speaker of this session, Dr. Krishna Kumar Sugumaran and Dr. M. Bhuvaneshwari. Thank you.
Thank you. Yeah, we are taking. Oh, okay. Thank you. Aja.
Madam, Bhuvaneshwari, madam.
Bhuvaneshwari, madam. Thank you so much. Thank you so much, madam. Of course. We are taking snap right now with everybody. I have just 2 more awards to give out. The faculty, Dr. Tippa Reddy. Thank you. Sir, did you get your delegate kit? Okay. And Dr. Dayanand, did you get your delegate kit, sir? Thank you so much. Thank you. Now it's our signature movement. So let us all join hands. Yes? Come. What is there? Inna solluma? Vama. So thank you all very much for being such a lovely audience. Thank you. Lunch is ready and please come back at 2 o'clock. Thank you. Thank you so much. Ayo, thank you, ma. Thank you.
I have to give it to...
I don't have the pen. Dr. Trilok Chandra, Dr. Srilata, Dr. Akshay, Dr. Chandrasekhar, Dr. Navneet, and to come back as judges for the last session. And I request Dr. Vel Murugan, Dr. Mahesh Sahare, and Dr. Subha to come in as keynote speakers, after which we will have the free paper sessions. You will get them lined up. Yeah, go. Everybody, go for lunch, but please come back by 2 o'clock. Dr. Mahesh Sahare, Dr. Shakila, Dr. Shubha, and Dr. Vel Murugan. Are anybody over here right now? Sir, we will start with you, sir. How nice. The professor of our professor. Please come. Dr. Vinod is chairing our session, and he's a fantastic anesthetist and is working with the Sankara Hospital, where he gives anesthesia for the cancer patients, and he has been my anesthetist for more than 20 years. So he's absolutely fantastic, especially when it comes to pain management. We have Dr. Srilata, and where is Dr. Srilata? Dr. Srilata, madam. And Dr. Srilata is Dr. Trilok Chandra, Dr. Akshay, and Dr. Chandrasekhar are from RV College. So we are very, very proud to have this very close association with you all. Thank you, sir. Thank you, Subha. Can I ask Dr. Mahesh Sahare and Dr. Shakila to please come up on the dais. Now I would also like to take this opportunity to invite all our free papers which has been mastered by Dr. Vidya Niranjan. So I like Dr. Bharat, Farhan, Shruti, Murla, Nitya, Bindu, Falkya, Arnaga, Advait, Leah and Divyani. Please come up on stage. Thank you. Over to you.
I invite Dr. Vel Murugan. He is from Chennai. I invite him to start his first lecture, please.
Is he projecting?
Audio, video. AV, Kurpa. Thank you, sir.
No, it's Prashant. Good afternoon to all of you. After listening to your interest in the morning, I decided to change the way of my presentation. This was supposed to be for 30 minutes. Definitely, if we continue, nobody will be there in the audience. So, I've been asked to cut down to 10 minutes. Even in the 10 minutes, I'm going to show you the PPTs only for 5 minutes. So 5 minutes, I'm just going to tell you what it contains. So most of you know that by using Pan Parak and Ganja, many people are getting neurodegenerative diseases. And it is going to be million in another 20 years. So we have been looking what are the neurodegenerative diseases, and main things are Alzheimer's diseases, Parkinson's disease, Huntington disease and so on. Of course, Alzheimer's disease is coming because of the plaque in the brain, and Parkinson is due to motor problem, and both the things they have only limited medicine. Then we thought, will there be any medicine from nature? So these are called phytochemicals. So that I've been working. In the past 5 years I've been working on the marine natural products like seaweeds and so on. We identified some compounds and we did how the work on how these molecules bind with the targets like acetylcholinesterase, butyrylcholinesterase, beta-secretase and so on, about which you don't know anything. Okay. So I will tell you about our field. There is a field called X-ray crystallography, which has produced more than 40 Nobel Prize winners. So that is the only unique field which has produced so many Nobel Prize winners, and most of them are for the novel structures obtained using single crystal X-ray diffraction. If you want to understand the function of a molecule, or if you want to modulate the function of a molecule by binding with a small molecule that is known as, which is known as structure-based drug design, the knowledge of the three-dimensional structure is important. If you look into the database called protein database, we have more than 1.5 lakhs these structures deposited. And whenever a structure is doing a malfunction, you are supposed to attack the active site with a small molecule to control the function. So that is the trick. There are automated programs for doing that. That is known as docking. So in the audience, I have my friend, one Raghu, who was a former global vice president of a module called Schrodinger. So now he is the proprietor of one more thing, Molecular Sciences Corporation, which will be started in December. So I have been actively using this thing for more than 10 years. So that's what I'm going to present to you. So as I told you, every molecule has atoms. If the number of atoms are less than 100, we call it a small molecule. If you look at proteins or enzymes, they are made up of amino acids or peptides, which contain thousands of atoms. Those are called macromolecules. Every molecule does a function. Somebody told you about sickle cell anemia, no? There's only one mutation of amino acid, the function changes. Like so many cases are there. So to understand the remedy for the diseases, you must first of all know what is the three-dimensional structure of the protein related to that disease. So that you have to design molecules to bind. That is known as structure-based drug design. The knowledge of the three-dimensional structure is required. That is why crystallography plays important role. If I ask you one question, nobody is going to answer. Who is the person Indian, who shares the Nobel Prize for solving the biggest molecule? Not nod your head. Who is the person? Itiraj College people are asking so many questions. No, I'm also from Chennai. I must ask, no? There's an Indian who solved the biggest structure. And he shared Nobel Prize with 3 people. No. Because he's not in the syllabus. That's what. When Vidya was doing MSc, I used to take 5 hours classes at this stage, quantum mechanics, classical mechanics. Suppose I do this in these colleges, present days, people will get TC and go away. Right? Because continuously 2 hours you are unable to listen to science because you are in between break dance. Huh? So that is the level. Because now everything is fast food, no? So that is why I am not showing PPT at all. Fast, no? I am also a fast food person, no? So now you know there are molecules which are doing some function. If they do some malfunction, you must bind a small molecule at the active site to control the function, to modulate. Right? To whatever level, suppose you have 100 molecules to modulate, which is the best one? How to take? Suppose you have headache, no? So many tablets are there. Most of them contain paracetamol. Which is the best one you have to take? High dose or low dose? You don't get headache. Maybe after 15 minutes you will have it. It is always better to have low dose. Because once you take high dose, no, after that no high dose is available in the market. No, that's all gone. So that is called technically IC50. What should be the inhibitor concentration of that molecule, small molecule, which is going to bind with the molecule to reduce the activity by 50%. Okay? Then I have to answer the suspense also. The holy name is Venkatraman Ramakrishnan. The structure was ribosome structure. So he shared it with Stiles and Ada Yonath. Okay? Because he is an Indian, we don't want to give credential. Another thing is, October 8th, a famous scientist was born in India. He missed the Nobel Prize for his famous discovery about the structure in our body. Can you guess who is that? Very good. G.N. Ramachandran. Is there any connection between G.N. Ramachandran and a Bangalore professor who was a Nobel laureate? Who was in Bangalore, who was a Nobel laureate? Out of syllabus. Sir C.V. Raman. He was director of Indian of Science, no? G.N. Ramachandran was the student of Sir C.V. Raman. He found the triple helical structure of collagen or muscle fiber. For double helical structure, DNA, Nobel Prize was given. To whom? So many unanswered questions in your mind. What's the none? No biotech person is here, all engineering? Yeah, you are the only person who is answering. I'm asking, all others are other discipline? Biotech. But this is not in the syllabus. Very good. At least next time you include the syllabus. Nobel laureates of India or Nobel laureates who missed prize. Okay. So now people will be wondering why this fellow is crying without showing any stuff. Okay. So this introduction I given, what are the neurodegenerative diseases, Alzheimer, Parkinson, ADPD, and recently we found that there are something called small proteins called neurotrophic factors. There are 7 neurotrophic factors. They bind with some receptors and they start the signaling pathways which leads to neuronal death, causing these diseases. So now, we thought, can you bind those amino acids which bind with the receptor so that you can block the binding of that with the receptor and stop the neuronal death. So that's what we have done. We have taken about 47 chemicals from the natural products. And this is the reason of the Alzheimer and ADPD, major public health problem. AD is the most common form of dementia, accounting for about 50 to 70% of all cases. Then it is due to beta amyloid formation. Then you can compare left-hand side healthy brain, right-hand side Alzheimer and Parkinson's. Do you find any difference? Don't say no difference. Then eyesight is a problem. We got to go to Agarwal clinic, eye clinic. Is there any difference between left side and right side? Healthy and Alzheimer, Parkinson. What do you find difference? No difference. All are healthy. Hey, what is there? If you have good eyesight, you can answer, no? Left side, right side. No difference at all? Simple lunch only, no? If you have been given lunch which I serve in conference, that's all, you'll be sleeping. Huh? Karabat, that thing. What's that? Sundal, that's all. Channa, parota. With 3 dishes itself, you are sleeping. Huh? You come and attend my symposium. October 8th to 10th, Vienna symposium. Okay. Now, what are the various stages of the symptoms? If you have mild neurodegeneration, memory loss will be there, lingual problems, mood deviation, moderate, I given reason. Severe means motor impairment. You have to be bedridden only, no more walking. Then this is a list giving the people living with dementia around the world. Then this is about global deaths and prevalence for Alzheimer's disease and other dementias. This is about Parkinson's motor problem. Only 3 medicines are there. You got to change the doses, that's all. Not yet. So this gives the mechanism of this Parkinson. Then this is the earlier work I did. For Alzheimer's, these are the 2 protein targets. We got to try structure-based drug design. For Parkinson, this is the other target. This is the macromolecular structure I'm talking, proteins. 150,000 structures are deposited by crystallographers in a protein data bank. It is free to use, okay? Yeah, if you look at the center, there's a small molecule, right, with ball and stick. This controls the function, okay? This is for acetyl, then Alzheimer, butyl. So, based on our work, we have found left-hand side so many compounds from marine seaweeds, which acts against the targets in the second column to control diseases mentioned in the third column. So then we went to terrestrial compounds. From sea, we jumped to ground. So we talked about the receptor called P75NTR, neurotrophin receptor. Then also we talk about TRK, tyrosine kinase family. These are receptors which bind these neurotrophins and which initiate the signaling, causing neuronal death. So you have to avoid this binding, right? So this is the mechanism. This is neurotrophin signaling, explaining. So these are the structures. These are small proteins, structures available for this neurotrophin. 7 neurotrophins, okay? So this shows you how the neurotrophin 3 binds with the receptor. See, at the center in the blue, how comfortably it binds, it has interactions, so signaling process starts. So what are the interacting residues? That's what we are interested in. Sometime after binding, left-hand side, we have 2 molecules, so dimerization occurs with tyrosine K binding. These are not stories. You must know this is a science which you are not knowing at all. If you have free time to go to Google or something, everything will be there. You have free time, but you don't Google this, no? Some interested Googles are there. Huh? That's the problem. So now, these are the compounds I have taken. Everything is natural bioactive compounds. I subjected them molecular docking studies. Whenever you do the docking, there are 2 parameters. One is known as score, another is energy. Based on that, we select the compounds. So these are the best compounds we selected. 1, 2, 3, 4, 5. Among them, there are 2 techniques. One is known as virtual screening. Because whenever a small molecule binds with the active site, it produces changes in the rotation angles, that are conformation angles, the concept introduced with G. N. Ramachandran. So, if you want to take into the rotation of each bond, that will take a lot of time, 1 day like that, no? So, initially, even though we know that binding causes your conformation changes, we assume initially that receptor is rigid. And you give flexibility only to ligand. Okay? Then, from hundreds of compounds, you can filter to 10 compounds. Now you go to reality. You assume both receptor and ligands are flexible. That is known as IFD, induced fit docking. It takes 1.5 hours like that. So that's what we did. Okay. So you're asking if this thing goes like that, you will cross beyond 10 minutes. No. So this is known as Lick plot, showing the interaction of the small molecule ligand with the amino acids at the active site. So same stuff, what I'm explaining. So similar thing we did for all the 7 things. So since you are not interested, there is no meaning in showing you all. You want the end of the story, no? I know. Because teachers are not interested, first of all. They have timing, no. That's the problem. I never used to go to colleges, don't mistake me. That is the reason. Dance, time complete 11, 2, tea break, something, that's all. But people don't care whether you, when before attending conference, after attending conference, you got to do a scanning of MRI of your brain, no? Mostly it will be down nowadays. So don't mistake me, that's the reality. So now I will tell you the conclusion. So 7 neurotropic receptors binding I have taken. If you look at the natural compounds, best compounds, I have only 5. Rutin, piniculatin, salvianolic acid, naringin, and rosmarinic acid. So now we are thinking to have a formulation of these 5 and to give to patients. And we have a big project from Ayush. So we are planning for about 9 crores. So with Nimans, they have Parkinson mice. So it's equivalent to treating a Parkinson patient, no? So that's what I'm trying to do. So if you have listened to this, you could ask questions, but unfortunately, since you didn't listen, I don't expect any questions. I thank both Dr. Bhuvneshwari and my former PG student Vidya for all the way coming to Chennai for invitation. That's why I came all the way from Chennai, Bangalore to Chennai, Delhi, Bangalore. So thank you. So if you don't understand, you think that there is a field called X-ray crystallography, which has produced more than 30 Nobel Prize winners. Our Indian scientist, Sir C.V. Raman, was one among them. His student, Professor G. N. Ramachandran, was the one who narrowly missed the Nobel Prize. Okay, thank you. So if you have any questions, I can answer. Huh? Which plant? Yeah. That is secret, no. No, we did it. We did it.
It's an extracted compound.
No, actually we are going to do now, because we don't want to believe reported values. These are all from literature. You go to literature, natural compound, Alzheimer, natural compound, Parkinson, so many papers are there. I collected. Then from here, I go for PubChem like that, what are compound release, or GCMS result. Again, everything Google only. Scientific way, I utilize Google.
It's a database. If you put this, you will get the plants.
I didn't give any plant, no name. It may be one of them. I didn't give them. So I thank you. One question I received. So you are luckiest person to ask the question, no? Let us see what is the question.
You ask like that only, no problem.
Hello. Sir, I just had one doubt. So here, I think neurodegenerative diseases were used, but in certain neuropsychiatric diseases like autism spectrum disorder, VDNF plays a role in the development and the subsequent management. So can these compounds even be used to target neuropsychiatric diseases?
Yeah, but nobody is working on that, madam. That's the problem. Actually, why I was very much interested here is my own father-in-law, come brother-in-law, was suffering by Parkinson for 10 years. That's why I was very much interested. But nobody is there to work, no. Because the moment we work for 1 year, 2 years, they want publication. It's not impossible. This is not one man's work, right? So animal test, every ethics committee is there, human trial is there, which we cannot do. Luckily, Padmanabhan did his PhD in our department. He's head of biophysics in Nimans. So he said, we can do. So he's one of the collaborators. So that is the way. Everything is there because before allopathy came, people survived, no? Right? People were there in the globe with the diseases. So now, when I went to Ayush, you won't believe, such crores of rupees they are wasting. I do not waste, any politician is there? Okay, sorry about the word using. Crores are spent without any outcome in nature, or they want nature, no? How many papers publish in nature something? Even IIC doesn't publish nature regularly, publication. But the moment you get it, how many, show me the index, how many papers you publish in nature, PNA, something, without knowing. So that's the story, Indian story. In spite of that, India is doing science, good science. In NIRF, South Indian University has come to the top, no? NIRF. That's a good thing. Instead of the political suppression, we are coming up. So we have to thank our own scientists. And now there is a boom for AI, ML. So no more science. Basic science, I mean. Yeah, everything is fond of money, dollars. No. Yeah, I worked with the Nobel laureate. I got to stay there in US. I worked for 4 years. But had I stayed there, I would not have produced 44 PhDs in India. And those guys are very loyal to me. Until, wait, then clap. They're very loyal to me. Until they get the green card, they used to communicate with me regularly. They've got recommendation, that's all. 40 people stopped communicating with me. Because they're citizens of US. Thank you. I hope it's a lively talk which you people wanted, no? Audience.
Thank you, sir. It's really honored, especially for me. My professor has come and given a talk. And I just, as he told, he used to take quantum mechanics for us. That is called bioquantum mechanics those days when we are doing. He used to take the whole day, especially quantum mechanics. But we never had even 1 minute of boredom. It's not because of the subject. It is because of his humors in between. The science and humor going side by side is a very extraordinary character of Dr. Velmurugan. And I really big round of applause to Dr. Velmurugan. Next, I would like to call upon Dr. Shakila to take the stage.
Good afternoon to one and all present here, and I'm privileged to be here in this afternoon. And first of all, I would like to thank Sunita ma'am, Pankaj sir, and Vidya ma'am, Bhavana ma'am especially, and to my Brinda ma'am a special mention, without whom I wouldn't have been here for presenting my paper. And it's all about the topic, and it's quite interesting. All through my journey, I've been a teacher. More than a teacher, I worked in an IT industry for 7 long years. I know how hectic it is and how difficult to present something onto the screen. And after which, I joined in Ethiraj College. I've been there for 17 years. Now I've been promoted to associate professor. And my topic on this special forum is AI and ML assisted in gene therapy. It's a boon or bane. Anything comes more than a nature, it'll have its plus and minus. The creator and the maker will always have his own special quality. And he made us with all the possible things. And we are so special, and we are not simple as such. And the maker has made us so beautiful. And when he knitted us in the mother's womb, he has a plan and he got a purpose. The purpose and the plan is totally different. And we are all being here for one main thing, that's gene therapy, that into computers. And it's not that easy to do this, isn't it not? I have not seen my own gene. I know DNA, I know RNA, I know my cell, I know what my cell will do, the functions and etc. But I never knew my own gene, how it's structured and how it's been shaped. But the maker has made it so beautiful. And some alterations, if I do, there may be plus, there may be, the invention of the medicine is really good. Good in the sense, there are a lot of medicines which saves life. It has been privileged. And to say in my own family, we had so much of cancers, that 2 or 3 people had cancers. But the greater part of it, it's a blessing when somebody has a good treatment for it to have a quality life in them. But then I used to think, some says pharmaceuticals is a gambling-like and all. But is it all true? But it gave beautiful quality life to my own people. For that, I'm really happy. And then I chose a topic saying that AI and ML that is assisted in gene therapy, boon or bane. Can I have my slides, please? And here it goes. And my special thanks to Bhuneshwari ma'am, who did in the morning session about AI. I think she would have opened your eyes a little bit what it is all about. And now I'm going to relate it with the correlations. Next slide, please. I have it, please. And here's a slide that goes clinical correlation and regulatory affairs in gene therapy. All 3 are together. might be a clinical correlation and regulatory affairs and gene therapy. Without this combination, a drug could not be developed. And then, clinical correlation in gene therapy mainly focuses on genetic modification that translates the clinical outcomes. For example, a clinical diagnosis is very must when you do a finding. Then we need to go for imaging studies and then the laboratory work. We have 3 types of correlation. One is positive, the other one is negative, and there may be no correlation. First of all, we need to take all tests which is related to blood, where it will have a biochemical and enzymatic reactions. And then scan, X-ray or all other imaging techniques, where we go for the second one. And more than that, we have an extra test which is very, very specific to all the human beings. So what is the role of regulatory affairs? There are regulatory bodies which focus on laws. And before they go for the laws, there are too much of ethical issues into it. And then the regulations, they'll put forth some plus and minus regulations where we have to stick on to it and then work. We have a concrete bodies. We have been recognized by the private and the government sectors. They do a little bit of work. That is basic research. And then after which they'll move on to the development. From there to the approval applications. Then comes the manufacturing. Then post-marketing. And what could be the special function of a regulatory affairs, that is compliances? Product like life cycle they should see, how much they can do clinical trials and market surveillance. Suppose, for example, if it is COVID-19, there wasn't a clinical trial exactly for which we have been just subjected to the vaccine, and whatever is happening now, it has been recorded. But normally it will not go that way. A clinical trial is a must for it. And then the documentation. Then comes an interdisciplinary collaboration and how much it has been impacted in the globe. And gene therapy, right from 48 hours, we are just only hearing the term called as gene therapy. What is exactly gene therapy and why do we need it? Into a very local, common man, it is something to say, it's nothing to fix a gene that don't properly work. And then sometimes it replaces the gene that don't work properly. And sometimes make the immune system aware of the disease cells. So wherever I have highlighted in the red ones, to which is some disease, somewhere, if the gene is not properly working, the product which we are giving or the gene therapy, it will turn off. The other one, at the same time, it can even turn on. And to the cancers, especially that is P53 is a tumor growth which induces more into it. All that can be fixed. So this therapy is a need of the hour too. There are risks which is also involved in it. Normally, a healthy gene is taken, it has been subjected to the virus vector, and from there, with the virus vector, with a healthy gene, it will be incorporated to the human being to get the corrections. If it is done little away from it, because I already told you in my introduction itself, computer is not that good. Sometimes if some coding is missed, some assessment is missed, finished. It randomly works. And that will also be reflected. It's not a joke because we are just doing it with humans now. Unwanted human immune system reactions and targeting the wrong cells, then infections can be caused by the virus and possibility by the causing errors in these genes. All these are the risk factor which is involved in the gene therapy. Then comes artificial intelligence. What is artificial intelligence and genetic engineering in gene therapy research? It is more human brains work with the robots. That is all about it. They can think, they can work, they can do everything. And it's not a simple artificial intelligence has got these many things in it, and mission languages are the subset. These are all the subset in it. One is mission language, and the other one is natural language processing, expert system, vision, speech, planning, and robotics. All these are very clearly explained in this flow chart, so that it is one mega thing. And where comes machine language learning in the subset of AI? Because it's not that easy, I told you earlier. Machine language, it's a language part. Whereas artificial intelligence is an intelligence part where human can work. So here there will be an input, lot of gene expressions will be there. Along with that, the chemical composition, we need to have an algorithm. Algorithm is a very important thing where too many people will be involved into the research. For example, 500 or thousands of samples may be taken and all that being analyzed and put it into the algorithms. And then you will get a predictive medicines or a recommendation. And what is the major difference between AI and ML? AI is the mimic of human intelligence, whereas machine language, it will perform what it is needed. So accurate results can be given into it. And then I told you something about natural language processing. And this only enables the healthcare providers to automatically consolidate the medical data from the lab result, patient record, insurance claim, and other sources. If you go, they put everything in one packet and then they will give you. Okay, just because of the process called as NLP. And clinical correlation in AI, it's nothing but taking all the data and analyze them, that is separating them, that's what the common word we use to clean it up, and then you analyze and then the report. So this part is being done by the clinical correlation in AI. And this flowchart will explain you the general steps in gene therapy. I'm not going to go much into it, but my focus is on after gene therapy, it can tailored to an individual patient. It's not common to everybody. So for example, if you have fever, we know what drug, it's all paracetamol we are going to take. If you have gastritis, you either go to pantacids or gelisols. There are common medicines, but tailored medicines is mentioned for each and every individual. And this is all about the AI-assisted gene therapy. So I told you all clinical correlations and the reports, what you get, you put it into the high-dimensional data set. Then the learning methods, go for the data analysis, and personal medicines. As I told you, it's a tailored thing. And now comes artificial intelligence in gene therapy. First thing, we need to understand identifying the gene targets. Second, personalizing the treatment. Third, you go for designing, guide the RNAs. Select the delivery strategies, monitoring the patients, and design the proteins. Applications of AI in gene therapy development, you will personalize the medicine. Target identification is there, and validation is very, very important. Viral vector designing optimization is there, so that you'll design the own optimizations. Then manufacture them. Clinical predictions are there. Biomarker predictions is also there. It's now a very heated thing in the pharmaceutical sector. Clinical applications of gene therapy. These are the drugs. That is, Luxturna is for retinal dystrophy. And Imelgic is skin cancer called as melanoma. They're all been treated, and these drugs are made out of gene therapy. How is AI used in clinical decision making? That is very, very important. Suppose doctors are there, and never an AI cannot overrule a physician or an expert in whatever the field is. But they can assist, they can modulate, so that they can do more number of patients in a given time. So AI can assist health professionals. It can diagnose disease, planning treatment, predictions, and then managing population health. This is all about how it does. You got a variables, you got all set of things. I told you algorithm is very, very important, and after which it will go for each and every individual. What are all the advantages of AI? When we go for the advantages of AI, it is faster in time, cost efficiency, regulatory compliance is there, data can be analyzed and managed, personalized medicine, improved patient outcome. Then, what are all the challenges? Challenges is very important when you find out something new. So now, lack of high quality data privacy should be there, regulatory and ethical consideration. And best in AI is early detection, continuous monitoring, and flag potential health issues. Is it a boon or a bane? Here comes the end of my talk. So obesity, diabetes, mental illness, and cancer, all these been already there with 40 plus. Some have within 30 plus itself, but for which this gene therapy is really good. And you can also have a designer baby, so you are going to cut on what is very defective and make it all a good one. But what is the bane in it? Here you will have a designer babies. It's really, you cannot do it as such. We cannot have our own functionings. I already told you, the creator and maker had made you so beautiful. You are not ordinarily made. You are fearfully and wonderfully made. Right? We are all fearfully and wonderfully made by God Almighty. But now, this designer babies, you can edit and do these babies, right? Then biohackers can own all these things. So, pros of AI-assisted gene therapy, it can increase the efficiency, improved accuracy is there, cost saving is there, personalization is there, and improved safety is also there. At the same time, there are also cons. So, job loss and bias, it is bias and discrimination, lack of human touch is there, privacy and concern is there, dependency is also there. In all about my conclusion, I could say more of bones and less of brains, but still we need to think what we are going to do. Technology is useful to mankind, but it is dangerous as well. And then humans are subtle and extraordinary, but robots are not. We cannot tell a robot to be a creator. Thank you.
Thank you, ma'am. Indeed, it's a wonderful presentation. Now, I would like to call upon Dr. Mahesh.
So, good afternoon to all. I am Dr. Mahesh from belonging to INSTEM and NCBS. And today I am talking about the CRISPR genome editing, generating the mouse model for the human therapeutics. All your, my voice is reaching to all? Last, yeah, sure. So, madam already told how the designer babies and all. So, we are generating the designer mice for the various application of studying the human therapeutics and all, and that's we are routinely generating the 10 to 12 mice models every year, and that's we will learn here today how we are generating the mice models, right? Yeah. So we are getting around 300 crores of the base pairs, nucleotide base pair from our parents. Half is from father, half is from the mothers, and these are the genetic building material that is passed on from the genetics to genetics. So these are called like a chromosome, genes, and the proteins, all you know, the central dogma and all you are studying in the biotechnology, right? So these, our genome is constantly under the threat of the DNA damage, and every day a lot of DNA damage are happening in our body. And that is called the various, in the form of the double standard break or the various breaks, and these DNA damage can be repaired by our own body itself. We have the cellular machinery, and that is repairing that. That's the cell and making it functional. But those DNA damages are not repaired, will be accumulated, and that turns in the form of the impaired cell function, and that is result into diseases. So for example, earlier also people, the speaker has told about the sickle cell anemia. So it is a single base pair mutation. So these one base pair mutation are causing the entire blood cells in the form of the sickle shape and impairing the functions of the body. And there is no cure for such type of the diseases until now, after discovering the so many drugs and all these things. So to treat such type of diseases, researchers are in search of the new technology, and that's how the programmable nuclease come into picture. Right. So recently, like 3 programmable nucleases are come in the picture. The one is the ZAPN, another is TALEN, and the last one is the CRISPR. And due to its simplicity and the affordability, CRISPR is making the boom. And there is a lot of good products are coming to treat the diseases with the CRISPR-Cas system. So what is a CRISPR? CRISPR is the bacterial immune defense mechanism system that is adapted as a gene editing tool. So when the bacteria is attacked by the viruses, the the bacteria will procure some of the SNPs from the DNA, from that viruses and incorporate into their genome. And the next time the same virus is attacking on that bacteria, he will use as a reference sequence of that genome what is incorporated earlier, and it will create an immune system. So bacteria saving himself from the attack. So these tools has been developed as a this tool has been adopted from the bacterial system to use as a genetic editing tool, right? So only the Cas9 protein that we are not synthesized by the human body system has been procured from the bacteria, and with the help of that, the human genome or the any genome, including plant and any other species, can be manipulated, right? So this is like a 25-year of the research that the different scientists has done over the period of time, and the one element has been discovered, and finally the all pieces has been gathered in 2012. The Professor Jennifer Doudna and the Emmett Carpenter has shown that with the help of these guide RNA and the CRISPR-Cas system, we can create a double standard bake into the human genome. And that is what the great discovery they have shown. And that whatever the double standard bake is happening in the genome can be repaired by our system. So that's how we can correct the gene, right? So in the last 10 years, the lot of, 12 years, lot of research has been done, the lot of gene knockout and the plant edited crops or the different type of the base editors or the CRISPR editing system has been come. And the future is like that. It will be currently the sickle cell anemia, FDA has approved the sickle cell anemia therapy using the CRISPR, and with the help of the imaging or the artificial intelligence and the sequencing, the things are advancing a lot, and we are getting the cure for the diseases. Right. So currently, just I told now the sickle cell anemia, there is already FDA has approved the CRISPR therapy in the UK and US. And the other various diseases are under pipeline. So what is the use of these therapies? So earlier, managing the sickle cell anemia, patient has to take a lot of medicine and lot of pain. But by using these type of the therapies, we can, even if we go, the patient can walk to the clinic and within a one shot of injection, he can get a permanent cure. So that's how the quality of the patient's life as well as their near and dear ones' life or welfare will be happen. So in our lab, we are trying to develop the models, and the mice is used as models for studying the various genetic diseases, and it's the basic laboratory tools. So we are using the CRISPR-Cas technology, and we are generating the various model. So this is the CRISPR is very simple and the affordable, and the turnover is rapid. Hence, from 2012, the model generation using this technology has been increased a lot. And this we have adopted at our place, and we are generating the model for any type of these things. So what we need to create a double stranded bake at the genome, we need a guide RNA called 20 base pair recognition site that will search into the genome and find the target. And that's we need also the PAM sequence. These are the details maybe you can find and go by Google. So by using this thing, there are two types of the mechanism. One is the non-homology enjoining, and another one is the HDR mediated direct repair mechanism. So by using both by these two different mechanisms, we are generating two different type of the models. Like for knockout, we have to use this non-homology end joining mechanism, as well as for knock-in model or the point mutation, we have to use the homology directed repair mechanism model. And this is the workflow. With the help of the bioinformatics, we are designing the strategy, which gene at what location and what point we have to make the changes, and then the reagent will be synthesized in-house or procured from the companies, and then micro-injected into the 1-cell embryos or 2-cell embryos, and generate the models, and screen the models and screen the pups, whatever we are getting, and establish the lines for that. So how this work? Some scientists will find out, the researcher will find out some mutation in the patient that is novel mutation, and that's they want to correlate or study into the human, they validate into the model. So they are giving us that particular things, which type of the mutation they want to create into mouse model, and we will take over the project. So how difficult it is to generate a mice model? So it's very simple. With the help of some bioinformatics knowledge and the molecular biology tools, we can easily able to generate this thing. So first thing is we have to find out the target. Here I have given the example of the knockout animals, where I want to knock out this exon 2 or like a particular critical exon that is contributing for the protein expression or what? First, yeah. So with the help of that thing, we can just generate the design, and by using the free resources, we can find out the guide RNA that is essential for these type of the studies, and then we can synthesize the reagents, validate the reagents into the laboratory. And this is our 4-day protocol, where we are injecting the embryos and making the gene editing possible. Right, sure. So here is how we are injecting with the help of the micromanipulator. We are injecting the embryos with the particular cocktail of the solution, and the DNA edit will happen. And likewise, we have generated a various model, including some point mutation, and that are showing the phenotype every year. So due to lack of time, I have to go pretty much faster. So in the COVID, we have generated the model called ACE2 model, and the various laboratory has used this model for validating their vaccines, as well as some COVID-related molecules also. And we are also doing the assisted reproductive technology at our place, where we are cryopreserving the models, and we have the whole sorts of more than 700 mice model that different researcher can be used at different places. So we are combining these two facilities, animal research facility and the mouse genome engineering facility, where we are organizing all these things, and we have the very high barrier facility for animal model, and that's how we are conducting the workshop and seminars also. Those who are interested, they can join for such type of workshop and get trained with us. Yeah, thank you very much.
Thank you so much. In the interest of time, I would like to call upon Dr. Shubha to give her talk.
Hand mic, it should be there now.
Okay, good afternoon to everyone gathered here. I am Shubha, Head Department of Microbiology from Sona College of Arts and Science, Salem. So my topic is AIML in antimicrobial peptides, designing and its application in my biomedical field. So students, I want to give some idea about the novel techniques here. Okay, antimicrobial peptides, it's a small cationic molecules. It is a bioactive molecules. So it is present in all the living organisms. If any pathogen infect our body, so it provides the first line of defense against the pathogen. So it has a wide range of applications in different fields like medical, pharmaceutical. In different fields it has applications. So this antimicrobial peptides, it is synthesized by using 3 methods. One is a natural method. So I already told this antimicrobial peptides is present in all the living organism. So we are extracting this antimicrobial peptides from the living organism. Then the second method is we are inserting the antimicrobial gene into the vectors. Then this chimeric construct is transferred into the host cell. In the host cell, it undergoes expression and it produces more amount of antimicrobial peptides. Then the third method is an artificial method. So here we are using AIML techniques to produce the antimicrobial peptides. So this antimicrobial peptides, it has applications in a biomedical field. So it is used as an anti-cancer agent, antibacterial, antiviral. It has different functions, and also it is used as it is used to treat the eye infections. That is, in the inner surface of the contact lens, it is coated, and if you are using this, it prevents eye infections. So it is used as an immunomodulator. Then tissue repair, it is used as it is used for wound healing. Then important function is, if you are using regularly the antibiotics, it provides resistance to it generates resistance in the bacteria. So it is avoided with the help of this antimicrobial peptides. So it is used to overcome the antimicrobial resistance. So in the AIML, many website tools and databases are available to design the antimicrobial peptides. So this databases and website tools we are using to identify the antimicrobial peptides and also its physiochemical properties, functions, then AMP protein interactions. These are all studied with the help of web-based tools and the databases. So these are all the web-based tools. Then these 2 databases are used, mainly the machine learning algorithm. These 2 databases are used to designing the antimicrobial peptides. So this tool is also used to design the antimicrobial peptides. So antimicrobial peptides, the use of these peptides in gene therapy. So this antimicrobial peptides, it is mainly used for drug targeting. So along with the drug if we are inserting this antimicrobial peptides, it target the drugs in the particular target sites. So here we are using a cell-penetrating peptides and intracellular targeting peptides. So these two vehicles, it is used to target our AMP into the cells. So in some condition, if we are using any one, it has some barriers. So in that condition, we are using the fusion of these two targets. So in this antimicrobial peptides, it has the ability to cross the blood-brain barriers. So it is used to treat the brain-related diseases also. So different strategies to enhance the treatment. I already explained the recombinant DNA technology. Next one is a camel. So we are using the camel to transport the materials. So here it is used as a camel. So here we are hybriding two different antimicrobial peptides. For example, here we are hybriding secrofen and melatonin antimicrobial peptides. So if you are fusing and if you are using this hybrid antimicrobial peptides, it gives antimicrobial activity and also it provides anti-cancer effects. So this C18 camel, it is mainly used for cancer treatment. So this P53 plasmid, it is conjugated with the C18 camel and it is transferred into the cancer cells. So in the cancer cells, it undergoes expression and it prevents the cancer formation, tumor formation. Then this complex, it is coated with MD-M2 inhibitors. It enhances the cancer efficiency, cancer treatment efficiency. Then normally this antimicrobial peptides, it is a proteolytic enzyme. So it is easily degraded by the proteolytic enzymes. So here we are using some techniques to enhance its stability. So by incorporating alpha amino acids, by using a cyclation process, it's a one method. Then it is conjugated with some biomolecules, stabilizing molecules. We are able to enhance the stability of the antimicrobial peptides. Then AMPs in cancer therapy. So this, it plays an important role in cancer treatment. So traditional method, that is radiotherapy, chemotherapy, in this normally it is a cost-effective process and it is a costly process and also it causes some side effects. But here, if you are using antimicrobial peptides, it enhances the cancer treatment process. So here also we are using this DCT-PEP. So this databases, it is mainly used to design the anti-cancer peptides. So this anti-cancer peptides, it has a different functions in the cancer treatment. So it prevents the tumor cell proliferation, then apoptosis induction, angiogenesis, that is, it prevents the blood vessel formation in the cancerous cells, and also it inhibits the DNA replication, and it acts as an immunomodulator, then targeted specificity. So it is used as a drug conjugates. So it acts as a linking agent. So it links the chemotherapeutic agents or cytotoxic agents. So it is conjugated along with this, then it is transferred to the cancer cells. So it inhibits the cancer cells proliferation. Then next one is a PROTAC. So this PROTAC, it is a proteolysis targeting chimeras. So this PROTAC, it is also, we are combining this antimicrobial peptides with the protein of interest. Then it is transferred into the cancerous cells. In the cancerous cells, the efficiency is enhanced. So if we are giving the anti-cancer peptides alone, the efficiency is somewhat less. But if it is combined with protein of interest, then it is transferred into the cancer cells. It enhances its efficiency. So this is the method. So the peptide with some linking agent, it is conjugated either with the chemotherapy drugs or with the cytotoxic substance. Then it is transferred into the cells, cancerous cells. So it escapes from the endosomal lytic enzyme. Then it causes apoptosis in the cancerous cells. Then peptide-based A1 CAR designing. So the CAR designing, it's a chimeric antigen receptors. It contains single chain Fc domain, then T cell signaling activating signals. These two compounds present. So along with that, we are transferring our A1 peptides. So it enhances its efficiency. So this is a mechanism. So the stem cells, we are collecting the stem cells either from the umbilical cord blood or from the blood, adult blood. So we convert it, if we are using the adult stem cells, it will be converted into a induced pluripotent stem cells. Then it is used to produce the T cells. So from the T cells, the CAR, we are constructing CAR. Along with that, we are transferring our A1 peptides. Then it is introduced into the patient. It increases the cancer treatment. So it is used in breast cancer. So it is used to produce, the peptide is used for producing vaccine, vectors, then targeting materials. Then in the CRISPR also, we are using the antimicrobial peptides. So already many speakers, they discussed about this. Just 2 minutes. 2 minutes. So in that, by using a gene editing technology, we cut the double stranded DNA. So in that double stranded DNA, we are inserting our antimicrobial peptides. So it acts as a immunomodulators. It enhances the immune cell production and also the immune chemicals, that is cytokine and interleukins, and it prevents the cancer tumor formation. Then it is used to produce the chimeric E-caps. Chimeric E-caps, it is an engineered cationic AMP. So it is also a hybrid molecules. It is also used to enhance the cancer treatment. Then cell-based therapy. So in the cell-based therapy also we are using AMP. So this AMP, it enhances the immune cell formation and also it enhances the immune molecules formation. That is, it acts as a immunomodulators and it enhances the cancer therapy. So by using, I already told, AMP is designed with the help of AI, ML, that is machine learning, and DL, deep learning. In some condition, we use it in a hybrid method. DL and ML combinedly we are using. So this is the process how we are developing anti-cancer peptide. So in the antisense AMP, so it is also one of the method to prevent the expression of oncoproteins. So here this AMP molecules, it blocks the mRNA molecules and it prevents the unwanted protein synthesis, that is oncoprotein synthesis. So by using this method also, we are using, this method is used for cancer treatment. So these are all the applications of anti-cancer peptides. So it is used in qPNA PCR, PNA fish, biosensors. So it has a wide range of applications. Just two seconds. So in silico drug designing, so AMP, it is used in silico drug designing. So here also we are using, these are all the databases. It is used to design the novel drugs against some pathogenic diseases. Then it is also used in tissue engineering. So tissue engineering, we are developing the tissues in the laboratory condition. That is with the help of scaffold, we are developing the tissues. So that tissues, we are coating the AMP along with the tissues. It prevents the infections in the, during the time of transplantation. Then protein engineering. So we carry out some modifications in the AMP with the help of protein engineering. So with the help of that, we are increasing the stability and also the bioavailability of the AMPs. So conclusion, So this AMP, it has a wide range of applications in different fields. So it opened the new era in the biomedical field. So in future, we are using some molecular techniques along with this AMP to enhance the treatment process. Thank you.
Thank you very much, Dr. Shubha. That was wonderful 4 keynote lectures we had. Now we have the Young Scientist Award, and we have 10 contenders for that. So judges, this is a different session, a part of the same session, but different award. We have a Young Scientist Award for one among these 10, and we have one Best Paper of Session where the 4 keynote speakers have been attributed. Are we ready? Can we start with our free paper presentations? Yeah. Now, the rules of the game, children, who's starting first? Come.
The Proteor.net from Anaga Setlur.
Dr. Anaga.
And she is from AIML Biotechnology track.
And what's her paper?
It's Proteor.net, novel GNN-based ligand binding affinities prediction.
Anaga, ready? Okay.
Just 3 minutes.
Anaga, we are going to try and put up the clock timer over here. You're given only 3 minutes. I'm having Anirudh stand next to you. So he'll go tap, tap on your desk. Okay? Come. This session is actually meant for all you students, only so that you can learn to give the 30-second elevator speech. We've done too much work. There's too much work that you need to put into practice. You can't. It's not humanly possible to tell all your experiences, all your PhDs, all your startups. You need to be able to condense that. And that condensing is what we are trying to teach our young scientists. So they have time. Judges, time is of essence here. Yes, Anaga, 3 minutes, start.
Good afternoon, everyone. I am Anagha, and today I'll be talking about Proteor.net, which is a novel GNN-based ligand-binding affinity predictor architecture. And this is done via SMILES, and we've done this for 3 important organs of the human body: liver, kidney, and brain. So just to briefly talk about the background, so we know that traditional QSER models basically predict how the chemical structures relate to its biological activity. This basically helps in understanding and estimating protein-ligand interactions. But they, however, struggle to fully capture all of the interactions that are there, especially the ones that are complex. That is why there is a need for an integrated platform, and that is why the problem statement is to directly predict the ligand binding affinity to the 3 organs that we have selected using a structural approach. And the solution is to use graphical neural network based architectures. So the main objectives were, one is to generate and validate the QSER models, to design and evaluate the GNN architectures, and to develop a user-friendly real-time docking score or binding affinity predictor. So this slide talks about the overall methodology as well as the tools and techniques. So we have 3 methods, that is data collection, pre-processing, as well as building of the model. In data collection, we collect and select specific proteins from all the 3 organs. Then we have building the ligand library, docking, as well as performing QSER models. In our study, we performed 300 plus QSER models and built them. Then we pre-process the data by initial data set preparation, followed by model building. That is, we built 2 models. One is GCN, Graph Convolution Networks, and a hybrid version of the first one. Then we deployed it on Streamlit. So these are our results. This basically shows a sample representation of how our QSER plots look like, which is seen in the graph on top, as well as we have the distribution plot also. So these are the 2 models which we used. One is the simple GCN model, wherein the graphs are correlated to the target variable, and in this case, it is the docking score or the binding affinity. And we have model 2, wherein one additional attention mechanism is taken into consideration, which is the positional encoding. So this slide talks about our initial data set, what were the small molecules and how we kept it all and correlated them. Then we did a feature representation. So since this is a preliminary model, we took only 5 features to represent them, and these are the 5 features which were taken. And the Docking Score app was then deployed on Streamlit, and the application and link is also given here. So the application somewhat looks like that, and it gives users 3 options. One is to select the organ in the human body, then to also select the protein which they want to check their docking score against, and also to build a model. So they can select either model 1, which is the GCN, or model 2, which is a better version of GCN and gives a better accuracy. Yeah. So these are the 2 publications which we have got from this study. Thank you.
Thank you, Anika. Thank you. Thank you very much. I'm not going to ask questions now because you're running a little late. We'll go to the questions at the end of the session. Dr. Advait, please come. Anirudh, please read out what is
So we have the second poster presentation under AIML in biotechnology track by Advait V. The title is Transportation Networks Inspired by Growth and Evolution of Slime Molds.
The timer is on a PIP, so you can see it up there. Okay.
Yes, ma'am.
Thank you. Ready, Advait?
Okay, so I'm going to talk about bio-inspired transport network systems. So we, as Bangaloreans, have been stuck in traffic forever and ever, and we need to get around quick and fast. How many here have been stuck in traffic within the past 24 hours? Quite a few. There we go. So the problem statement is tackling this problem that we have. Okay, so introduction of so I have a solution to this problem that we have, that is to tackle the traffic problem and the roads of Bangalore or any other place in India. So before going to the solution, I'll just give you a brief history about all these, where we took the inspiration from. We took the inspiration from an organism called Fusarium polycephalum. That's basically a slime mold, and it has existed for about 600 million years, and we have existed for a very minute time according to that. And we honestly, something that has survived for the past 5 great extinctions ought to know something better than us, that is to survive. And the main objectives of this problem statement that we have is how to transport properly, how to distribute population, traffic management, and water management. So after that, these are the materials used, not that important though. Okay, so coming to the main problem, what are we using to solve the problem? The traveling salesperson problem is a very common thing in computer science, where we talk about how a traveling salesperson has to go from one house to another house constantly just to deliver substances such as newspaper or anything. And this slime mold encompasses all of that information and takes it and solves it within few seconds. Okay, this is the physical experimentation that we did in our RVC biotechnology lab with the help of Dr. Nagashri ma'am. At the end of day 7, there you can see, although the straight path would have been the most efficient, shortest path, it took this path, showing that it's the most efficient path because that would cause a lot less traffic for the slime mold to grow. Okay, so here are the basic applications of it. We'll have a better, efficient city designment, and then we can control movement, traffic organization, and coming to the medical part of it, we can integrate nanobiotechnology, AI, and this, so that we can solve problems such as catheterization that can be completely automated. We wouldn't need doctors to do that. We could have the slime mold grow within another place so that it can do catheterization right there. Okay, so this is the algorithm that we made based on the physical experimentation. So we have 2 algorithms. That's a map simulation at the first, where you can just pin out any points that you want, and then it'll give you the most efficient path to go from one place to another place. As you can see in those clustered areas, those are the population dense areas, it'll give you the most efficient path. The second one is a map finding path, where it'll give you the most efficient path to go from point A to point B. So if you scan that QR code, you can go to our website and try this out for yourself right now and anytime you want. So, okay, with that, this is the outcome, and thank you.
Good job, Advait. I'd like to call Falkia Taranum.
So now we're moving on to the second track, which is healthcare. So in that, as ma'am just called on, we have Falkia Taranum, and their title is Steady on Cytokine Production by T lymphocytes Triggered by Uroshiol from Rust-Tox Tinctures.
Superb job that you're keeping to your time. Excellent. Your stop clock is there. Okay?
A very good afternoon, everyone. I'm Falka Taranum. We recently conducted a study on cytokine productions by T lymphocytes triggered by urushiol from Rustock's tincture. Urushiol are basically...
What happened?
Can you please move to the next slide?
Please, yeah. Can we restart? Should
Timer
I
again.
Change or you guys?
Is this your talk?
Yeah.
Okay. So we'll start the timer again. Let's go.
A very good afternoon, everyone. I'm Falka Taranum. We recently conducted a study on cytokine production by T lymphocytes triggered by urushiols from Rustock's tincture. Urushiols are basically plant-derived catechols found in Anacardiaceae family. They are known for causing acute contact dermatitis, a type 4 hypersensitivity reaction. They are known for causing symptoms like skin lesions, inflammations, and swelling. The purpose of our study was to investigate the anti-inflammatory properties of rustocks and their impact on immune cells, and also their ability to modulate to modulate their cytokine production and reducing the inflammation. Moving on to methodology, we used two commercially available rustocks from Schwabe and SBL and cashew nut shell extract, which we used as a substitute for commercially available urushiols. We also did both qualitative and quantitative estimation of polyphenolics using several advanced techniques such as GCMS and TLC. We also did cytokine assay, and also we used blood samples from both naive individuals and those allergic to cashew nuts to check the CRP levels using turbidometric assay. We also developed a novel method of culturing T lymphocytes without the use of growth mediums like RPMI or fetal calf serum, therefore reducing the cost of cell culture. So these are the results. The phytochemical analysis and TLC showed several bioactive phytochemicals, specifically phenolics and flavonoids. So the GCMS analysis showed several compounds that can cause irritation and several compounds that can cause that showed anti-inflammatory properties. So the absence of urushiol, it also showed that there was no detection in urushiols because there might be some breakdown by an enzyme called lacase, which during the tincture preparation. We also saw there was no changes in CRP levels in both the blood cultures. It might be because the CRP might be CRP might not be the inflammatory marker in whole blood inflammatory marker, or it can be that anti-inflammatory compounds present in the tincture could have impact on them. So to conclude, our research has shed a light on phytochemical analysis of Rustock's tincture and their impact on immune cells. We also saw there was no role of CRP in CRP and yeah, next. Thank you. This project was funded by KSCST.
Thank you so much. So next, moving on to the second participant under healthcare, I invite Hima Bindu. Her title is Targeting Friedreich Ataxia, a Sustainable Path to Safer and Smarter Therapeutics Through Integrated Docking and Toxicology.
Good afternoon, everyone. So our study mainly focuses on Friedreich ataxia. So the topic is targeting the Friedreich ataxia, a sustainable path to safer and smarter therapeutics through integrated docking and toxicology. So now let me brief you about what is Friedreich ataxia disease. So it is the autosomal recessive neurodegenerative disease which is caused mainly in the age, like the symptoms are caused in the age of 5 to 10, and the disease can be caused in 25 years old. So in this disease, it is caused due to the mutation which occurs in the FXN gene because of GAA trinucleotide sequence which occurs in the intron of the FXN gene. which blocks the transcription process, due to which the frataxin protein is not being produced. If frataxin protein is not being produced, it causes a major problem for the body because it is a mitochondrial protein. So if no frataxin is being produced, it incorporates iron and sulfur ions in the mitochondria, due to which it causes free ionic iron accumulation, due to which the ATP production does not occur in the cell and the cell starts dying. The other way is because of the free ionic iron accumulation, the free radical formation takes place in the body, due to which oxidative damage occurs in the DNA and the proteins, due to which the cell dies. So because of this disease, we did the research. We found that till now, there is no such treatment which directly cures the root cause of the disease. That is, it doesn't solve the problem of epigenetic silencing of this gene. See, the treatments which have been done till now are only focusing on the symptoms which have been caused by the disease and reduce them. But we have found that the therapeutics which are found, one of the example is Omaveloxolone, which comes from the Skyclarys company, which mainly focuses on symptoms, but it has caused many side effects to the body, due to which it is not solving the problem of the disease. The second challenges which is faced by the drugs are because of the complexity of the mechanism of this disease, as it is a mitochondrial protein, many biochemical pathways have been affected to the body, due to which the person who is suffering from this can also have heart diseases and can die. And the third is the therapies which have been found have major toxic problems which have been causing a side effect to the body. So our team has focused on finding a drug which has more efficiency than the present drugs and has less toxicology. So the methods which we have used is finding a novel drug discovery using predictive modeling, that is computational modeling. So in this we have used a platform known as Galaxy EU, which gives a environment and which gives the environment to perform the docking. So here we have taken these several compounds.
Thank you. Okay, now we move to the second participant under genetics. So we have genome-wide identification, functional characterization of protein phosphatase 2A by Nitya Fernandez.
Good afternoon, one and all. I'm Sister Nitya Fernandez. I'm here to present my paper title, Genome-Wide Identification, Functional Characterization of PP2A gene family and its role under abiotic stress in Cicer-Aratinum. Well, before we go into the study, I would like to state my problem statement. Despite the role of Arabidopsis PP2A genes have been well studied, and it's well documented, but the role of PP2A genes in Cicer-Aratinum is not been well known. And hence, we have come with this objective saying that identification and functional characterization of PP2A genes and its role in abiotic stress in Cicer-Aratinum using genome-wide identification studies. So, well, here are a few of my findings of my studies. First one, to study about the evolutionary relationship of PP2A genes of chickpea, with different plant species containing these PP2A genes, a phylogenetic analysis was conducted using MEGA software. Furthermore, to evaluate the structural characterization and the diversity of PP2A in chickpea, a correlative study of motifs from PP2A peptides of Arabidopsis thaliana and Cicer-Aratinum was conducted using the MEME-SUIT software. There are 21 genes of PP2A chickpea genes which are widely distributed across the 7 chromosomes as it is represented here. Next. The cis, that is care, cis-acting regulatory elements analysis helps in defining the responses to various stress factors by the plant. Next. As we move further, the RNA data was analyzed from which the potential role of PP2A chickpea in response to drought and stress factors were obtained, and the heat map was generated using Heat Mapper server online. The green color represents the upregulation in the drought stress as well as in the salt stress. Next. The structures of PP2A proteins were obtained from TR rosetta, both Arabidopsis thaliana and Caesareatinum, which were superimposed by using Pymol, which was then, which showed that PP2A, that is the A subunit proteins of chickpea, showed similarity with the PP2A subunit of Arabidopsis thaliana. Next. To identify the structural characteristics of PP2As, the exon and the intron structure of the genes were analyzed using the gene structure display server. Next. Concluding to my research that I have done, firstly, we identified there are 21 PP2A genes in Caesareatinum. Expression analysis showed that 5 genes, both in drought and salt stress. Well, thank you.
Okay, now I'd like to call upon Mela Sudha with their title being Multi-Ligand Docking. This is also under genetics track.
Just tell next. Are you starting? Good afternoon, everyone. I'm Merla Sudha from Dayanand Sagar University. Today, I'm going to present my art study on multi-ligand simultaneous docking of benazepril and captopril reveals enhanced affinity and synergistic interactions with ACE. ACE is angiotensin converting enzyme which converts angiotensin 1 to angiotensin 2. And angiotensin 2 in turn induces constriction of the blood vessels, which will in turn it'll constrict the blood vessels, which will in turn increase the blood pressure. So now there are 2 inhibitors which are FDA approved, which are benazepril and captopril, which are often prescribed by doctors for patients who suffer from hypertension. So these inhibitors are known to inhibit ACE. Now we have performed single docking and multi-ligand simultaneous docking. So in our study, we have performed single docking and observed that in the 2D interactions, there are unfavorable bonds with these both inhibitors. And when we observe the binding affinities, the binding affinity of the single dockings are less compared to our MLSG study. So MLSG is multi-ligand simultaneous docking, where you dock 2 ligands at the same time. So in this case, we plan on introducing the concept of combination therapy, where we want to introduce both the ligands together. So here we observe an inter-ligand interaction, which conveys that both the ligands complement each other and show synergy. So along with introducing the concept of combination therapy, we also explored the influence of the polymorphisms associated with ACE. So why we are looking at polymorphisms is because the genetic variations in each protein might lead to a functional or a structural change, which might lead to the change in the affinity of the drug. So for example, if any individual with certain polymorphisms, if they have the polymorphism, they show a different response to the same drug. So in this way, if we understand the polymorphisms, we can tailor a specific personalized medicine so that even that individual can have a special treatment. So in the methodology, we have taken the amino acid sequence, homology modeled it, and then we have performed the single docking and multi-ligand simultaneous docking. So these are the results that we have after we have performed docking with a polymorphic sequence. So as you can see in our single docking, we observed unfavorable bond with the first inhibitor, and the figure 6 represents the multi-ligand simultaneous docking. Now, so to conclude, we want to propose the combination therapy with both the ligands, and also we want to study the polymorphisms in each individual so we can come up with a personalized therapy even for people who are not used to the standard medication available.
Thank you. Now we're going to move to the next track, that's nanobiotechnology. So I call upon Shruti A. Rao, with her title being Bacillus, Synthesized Silver Nanoparticles and its Efficacy in Controlling AIDS aegypti.
Good afternoon all, I'm Shruti A. Rao and I'm here today to present my team's work that basically deals with vector control, that is Aedes aegypti. We know there is a dengue outbreak every year and around 390 million infections are reported every year at a global scale, and it's very important that we come up with certain green methods that can be used for the control of vectors, especially mosquito vectors like Aedes aegypti, which transmit dengue virus. Me and my team, we came up with a green method that is the microbial synthesis of silver nanoparticles. They are highly efficient in vector control. They kill mosquitoes. So our methodology goes like this. We identified and isolated our bacteria. It is a soil bacterium. We uploaded on NCBI and we got the accession number. It is Bacillus welesensis. Following that, we optimized conditions for the growth of our bacteria and subjected them to, so the growth was going good, and then we subjected them to stress, the bacteria. So they started secreting secondary metabolites, which are very, very crucial for the reduction of metal ions to metal nanoparticles. We subjected the secondary metabolites that we got to, we treated it with silver nitrate solution. They got reduced to silver nanoparticles, and it was visually indicated through a deep red coloration. To further characterize our silver nanoparticles, we conducted, so this is our culture broth, and the accession number is given here. We conducted UV visible spectroscopy, and the peak showed that there was formation of silver nanoparticles, followed by Fourier transform infrared spectroscopy, that is FTIR tests, to know that it was an extracellular synthesis of silver nanoparticles. It was followed by XRD to determine the size of our silver nanoparticles. It was around 56 to 59 nanometer scale. It was followed by SEM imaging, that is scanning electron microscopy, and EDAX, energy dispersive X-ray. We found out that the metal is found in its elemental composition, and also that our silver nanoparticles are cylindrical in shape, and they have a smooth surface. Following that, for the application part, we conducted many lethal concentrations tests and mortality percentage tests using probit analysis, and we found out that the larvae of Aedes aegypti, which were rare, they were sourced from NCDC, National Center of Disease Control. They were very much susceptible to our product, that is silver nanoparticles, especially as you can see in the graph, the second instar larvae.
Time's up.
Yeah, they were, thank you.
Thank you so much. Now, moving on to the next person under nanobiotechnology, Farhana. Their title is A Comparative Study of Layered Double Hydroxide in Basic Nanoparticles as a Potential Catalyst in the Conversion of Microbial Lipids into Biodiesel.
Good afternoon, everyone. Myself, Farhana from MSc Biotech from NMKRV College, and I'm here to give a presentation on a topic, a comparative study of layered double hydroxide and basic nanoparticles as a potential catalyst in conversion of microbial lipids into biodiesel. In our research, our basically research consists of comparison of two types of nanocatalysts based upon their catalytic performance. Traditional biodiesel primarily utilizes vegetable oils, but recent studies have been shifted towards microbial oils. The transesterification process is carried out using catalysts that can be nanocatalyst or LDH. LDH have shown to be promising, and also with that of nanocatalysts have shown to be promising result by enhancing the efficiency, reducing the catalyst weight, lowering the reaction temperature, and also increasing the reaction speed. Moving on to the next one, that is materials and methods. extraction of, first one was isolation of the bacteria, and which was where the cultures were extracted using Bly-and-Dyer method, and estimation of the total lipids were done using TLC and SPV method. Then synthesis and characterization of nanocatalyst and LDH was simultaneously performed, where green synthesis of nanoparticles such as we have used zinc oxide and magnesium oxide using the extract of stem and leaf of Colocasia esculenta. Parallelly, chemical synthesis of LDH was also performed using co-precipitation method. As these nanocatalysts were synthesized along with the microbial lipids, transesterification was carried out, which was characterized using FTIR and GCMS. Moving on to the results, figure 1 shows the oil red O stain bacteria. The figure 2 shows the profiling of the lipid using TLC. Figure 3 shows the estimation of the lipids, that is by sulfophosphoalanine method. Figure 4 shows the XRD patterns of the crystal size of the ZNO, MgO and LDH. Then figure 5 is the SEM analysis, where we determine the structural morphology of these nanomaterials. Figure 6 is the EDAX. Moving on towards the figure 7, that is FTIR, where we can see the peaks which determine the fatty acid methyl esters in the biodiesel. Moving on towards the conclusion, our study identified and characterized the obtained fatty acid methyl esters with that of using catalyst was determined to be in zinc oxide it was 35, and in magnesium oxide it was 45, and with that of LDH it was 25. Hence the 21%. Hence the obtained FAMEs were comparatively high with magnesium oxide than that of zinc oxide and LDH. Since by comparing these emerging catalysts, we can, as you can say, this research contributes valuable insights in refining biodiesel production techniques, ultimately supporting the greener approach.
Thank you. Okay, now moving on to the next track, sustainable agriculture. So I would like to call upon Bharat Kumar. His title is Phenotypical and Genotypical Identification of Wood Decay Fungi from Shimoga Patches of Western Ghats, Karnataka.
Good afternoon, everyone. I am Bharat Kumar S., Research Scholar, St. Joseph University, Bangalore. So my topic is phenotypical and genotypical identification of wood decay fungi from Shimoga patches of Western Ghats, Karnataka, India. So here, this is my survey area, Shimoga. So here I have collected mainly 20 wood decay sample, and majorly I have selected sawmills and forest depot. So in the 20 samples, I come up with 2 novel species of Ganoderma. So along 20 species, majority of the most dominant species are Polyporaceae and Hymenocetaceae. Coming to the introduction, so there is a difference between mushrooms and fungi. So these wood decay fungi are conch, bracket-like structure compared to mushroom. So these mushrooms are smooth in structure. Coming to the color and texture, based on color and texture also, we can easily identify which is wood decay fungi and mushrooms. And based on the location also, we can identify. These are my objectives of research: documentation and diversity of wood degrading fungi and biocontrol. So this is the problem statement. These wood decay fungi, these are the major threats to the timber yielding plant or valuable plants. So I am going to control these fungi, and these are the tools and techniques which I have used: microscope, camera lucida, and Mega11 software to build the phylogenetic analysis. And this is the study area, Shimoga district. These are the collection. So I'll collect major sawmill and forest depots of the Chikmagalur district, Shimoga district. And these are the identification of wood decay fungi with the help of macroscopic and microscopic character. And this is the isolation technique. After the isolation, I have sent to the Rajiv Gandhi Biotechnology Lab for the molecular identification. So here result, most of the fungi I have found majority like a living tree compared to dead tree fungi. And these are the table which I have found the different type and host of the fungi. And this is the taxonomical work. So in that Ganoderma lucidum, Ganoderma sessile, and Adspermum, Astralis, Gibbosum, Xyleria, commonly called dead man's finger, Daldenia, Xyleria, Acrosporulus, and Daldenia, another species. And this is my novel species, Ganoderma tectopravinae. Ganoderma is the genus, Tecto is I have found associated with the Tectona grandis. Pravinae is my guide name. And this is the gene bank accession number. And this is the Ganoderma switanium. So these fungi I have found Switanium mahogany plant. And this is the gene bank accession number. And this is the phylogenetic tree.
Thank you.
So these are the different. Thank you.
Okay, next person in sustainable agriculture, I would like to call upon Leah Merin. The title is Valorization of Chicken Feather Waste Using Keratinolytic Bacteria for Sustainable Plant Growth and Waste Management.
What if one day you open the doors of your house and see a pile of chicken feather waste just in front of you? This situation is not far from a near future if in case we fail to manage the accumulation of chicken feather waste. I am Leah Merin Stanley, research scholar from Christ University. And with immense pleasure, I would like to introduce the title of today's presentation, which mainly includes valorization of chicken feather waste. So these are the screenshots representing how day-to-day activities of our life are being interrupted by accumulation of chicken feather waste. So the main protein demand of growing population is met by chicken meat. It is estimated that in the coming year, by 2050, the amount of poultry production will exceed 27 billion metric ton. This exceeding figure is of a great concern when it comes to the amount of waste which is getting released into the environment. But with the help of bacteria, we can degrade chicken feather and thereby we can reduce the load of chicken feather as well as create beneficial byproducts from chicken feather. So with this aim, I have formulated an experiment which mainly include isolation of feather degrading bacteria, which is called as keratinolytic bacteria. And these are the results: collection and degradation of chicken feather using bacteria, and finally taking it for plant growth studies. So for characterization, we have used several techniques to confirm feather degradation and find out the nutrient value of the solution. This is the result. The first picture represents the isolated bacteria, control media with the intact chicken feather, degraded feather, feather residue after degradation, and finally plant growth. So these are the main highlights of my research. So with this method, I was successful enough to isolate a keratinolytic bacteria named as Bacillus tropicus, which was able to degrade 40% of chicken feather and was efficient enough to support plant growth when compared to the control plant growth. Research articles I have published, and thank you so much for the patient listening. Thank you.
Okay, now moving on to regulatory affairs in biology, we have Divyani Chatterjee with her title being Organoid Technology, Pioneering Pathways in Biomedical Research.
Am I audible? Yeah. So, good evening, everyone. I'm Divyani Chatterjee. I'm doing my bachelor's in Manipal School of Life Sciences in Manipal. And today, I'm going to be presenting on the topic, Organoid Technology, Pioneering Pathways in Biomedical Research. Now, imagine this. I go up to someone and I tell them, each and every organ in your body can actually be created in the lab, and we can test things on it. A sane and sensible person will probably call me crazy. And you know, a decade and a half ago, they probably would have been right. But not any longer, because now we have organoids. So what are these organoids? They are three-dimensional tissue-engineered structures, which are mini organs, which are created in vitro and can be used for multiple testings. Now, the main thing of these organoids is that they can functionally mimic each and every organ, and also they can be used to observe all the developmental pathways in the lab. They are mostly derived from stem cells, and there are a variety of different types of organoids for different applications.
So the applications extend into regenerative medicine, into personalized and precision medicine, into drug testing, and even into phylogenetic studies. The list is endless. Now the main uniqueness about the organoids is that it can be very easily integrated with artificial intelligence. Now why is this so? Well, they have good potential and tests can be done very easily on them. Also, where there is a requirement to generate a large amount of data and analyze data in a short duration of time, especially in drug testing and precision medicine, where we require repetitive tests, it is very, very useful to have this tool. Now, all of this technology, such a great, vast technology, so one quote comes to mind: with great power comes great responsibility. And thus, the ethical and regulatory landscape should be observed with a lot of care and concern. So the main thing about organoids is, since they are very new, the main ethical issues and regulatory issues are subjected to informed consents of the donors and also the established production guidelines of the organoids. But there are a lot of issues when it comes to market use and commercialability. So which include the cost capping. How much should these cost? Also, what about the ownership of the organoids and the intellectual property rights? Who owns them? What about protecting privacy and anonymity? What about if I get a disease if an organoid is implanted into me? Now, all of this requires research. And the main proposed solutions which we have come up with is that we already improve upon the consent forms which is present and also educate the donors, tell them exactly what they're getting into. Next is, we can also have extensive therapeutic and diagnostic testing so that we can understand what are the long-term effects of the organoids. Plus, research. This is definitely the last but not the least, because research unearths a gold mine of data which help us in the future. So having all of this together, we can hope to see that we can reduce animal testing and also have a society where organoids can actually cure every disease. Thank you. Have a nice day.
Thank you.
Thank you very much. Thank you. I really like to congratulate first Dr. Vidya Niranjan for such wonderful presentations, and second, all of you guys who stuck to your time. Now you know how to do a 30-second elevator speech. Yes? So, guys, we have finished with our session and we are waiting for our judges to give us their results. Are you ready with your results, sir? Or I can have a dance performance before that? Okay, perfect. Can we have one dance performance, Anaga? My Kashmiri group is ready. Ajao. This is Pakiza from Kashmir, and the song she's singing to or dancing to is Roni Madan. Correct?
It's Kashmiri Roof Roni Daman Pyomai Shwani Gomai.
Roni Daman. Correct? Pakiza is from Kashmir. Talent search, please. Talent search. Where are the other talent search? Thank you. All ready, talent search? Ready, Roni Madan. Pakiza from Kashmir on Roni Madan. Start. Start the song from the beginning, guys. Sorry, Pakiza. Oi. Start the beginning. Aarti, one of you, can you try my Sorry, Pakiza. Sorry, audience. And the Young Scientist Award. But as soon as this dance finishes, you're ready with the tabulations now. Thank you, Pakiza. I request Dr. Vinod to give her a special award. Thank you very much, Pakiza. And I'd like you all to know we're all whole of India is in love with Srinagar and Kashmir. And if you're as old as I am, then in love with Hindi movies, then we have seen Srinagar and Kashmir to the best. Thank you so much for bringing out that Kashmir here for us. Thank you.
Thank you. I'm very thankful to Dr. Sunita Rana Agarwal ma'am. She gave me this opportunity to perform here my cultural dance performance. It's a Kashmiri cultural dance performance. Thank you very much. Thank you. Thank you all.
Thank you very much. Thank you. I'd like to request all our members of our judges, please come forward. They're going to announce. Can I have our keynote speakers, please? Dr. Shakila, Dr. Shubha, Dr. Mahesh. Who's the last one? Yes. Come, all of you come up. For the best paper of session. Once again, not you. Best paper of session. No, not yours. We want only best paper of session. Keynote. There's only one winner. What is the name of the winner? Keynote speaker. What is the name of the winner? Doc, there we go. Yeah, you play.
Okay, the
So we have our keynote speakers, Dr. Vail Morgan, Dr. Mahesh, Dr. Shakila, and Dr. Shubha. And now I request you to announce the winner.
Yeah, the best paper presentation for the keynote address of this session goes to Dr. Shakila.
Give it to Vinod. There we go. Camera. There we go. Thank you. Thank you very much, Dr. Shakila. Now, thank you. Now we have amongst our young scientists, we have another award to be given. Yeah. You announce, she'll give. Chala. Announce. Dr. Anaga.
Yeah, this is the first prize. The
Dr.
first prize goes to Dr. Anaga.
Wow. Congratulations. And Dr. Vidya would like to make an announcement here. Dr. Vidya. Your announcement.
Now mine. It's the task. It gives me great pleasure to present this award of meritorious student of cash prize rupees 10,000, generously sponsored by Campus Dreams.
Wonderful. Congratulations, Dr.
To
Anaga.
a truly deserving student. This award not only recognizes academic excellence, but also celebrates the hard work, dedication, and passion that this individual has demonstrated throughout her journey. Thank you.
Go. That is the Guru Shishya that continues in our country. And now I'd like everybody, come all, come into a line. We have, one second.
For each track.
Sorry, beta. We have some faculty awards to be given. Vidya Bhuvna, please come up. Yeah. Dr. Vinod, you will give one mark. Okay, go. Dr. Lavanya, Dr. Akshay. Sorry, I missed your name. Sorry. Dr. Chandrachud and Dr. Trilok Chandran. Sorry. Thank you. Thank you. Thank you so much. And anybody on this side that we have not given the faculty award? You. Two more, please. You got. Shakila, madam, you got? Okay. So, Dr. Vel Murugan. இப்போ நான் அங்கே பண்ணேன்னா நீ எங்கே பண்ணோம் என்ன பண்ணோம் that is the correct way. Thank you so much for being a part of the ISGCT, and we have a signature movement in ISGCT which I'd like you all to come forward. Wango, I'll give as soon as we finish. Please catch your hand, everybody. Children, make the line bigger. Come, four of you. Then only you can make the line there. Come. Is there a line? Come. Now is it okay? Is it a line now? And please, we have not finished. I'd like you to please sit down. Please sit down. We have not finished. I have Dr. Vel Murugan. I know. Students, don't go away. Please come back. Whom you want, Kondo? Please.
Okay, now these are all the finalists of the poster presentations which had taken place as part of the pre-conference which happened on 10th September in RV College of Engineering. And they were our finalists for each of the tracks. So now we are going to be announcing the winner and the runner-up for each of those 6 tracks. And as already awarded, Anaga Setlur was the best presenter amongst all of those tracks. So now let's go to each of those tracks. First track, AIML in Biotechnology. The winner was Dr. Anaga Setlur.
Come, Dr. Anaga.
A huge round of applause.
Okay.
And we have the runner-up, Advait.
He has awards. You only need this. 1 minute, beta.
Next track in genetics, we have the first place by Mela Sudha. And the runner-up, Nitya Q. Fernandez. Next, healthcare track. We have the first place won by Musret Mukhtar. And runner-up goes to Hima Bindu. The next track, Nanobiotechnology, it's won by Dr. Ginny George's team. And the runner-up is Pushpa Kumari and team. In regulatory affairs, we had Divyani who has won it. And now moving on to the last track, which actually saw the most number of poster registries during the initial pre-conference that happened. That's Sustainable Agriculture. It's won by Bharat Kumar S and team. And the runner-up is Leah Merrill. A huge round of applause to all the winners out here.
Thank you very much. Thank you, children. Now come, let's start the talent search. Can I have the judges of the talent search up on the dais, please? Please come. Anirudh, get me the names of the talent search people. Better हो. नहीं वो तो आएंगे, ये तो part of the talent search आ जाएंगे. आइए. Come. Please, yeah, get me talent search names. God, I'll tell you. Talent search judges, please come up on the dais. Dr. Brinda Devi, Dr. Bhuvneshwari, Dr. Shubha. Vanguma, 4 here, 4 there. Vanguma, come here. Okay, whom do you want to call? Tell Brinda Devi. Tell Vidya. Betona, come. Dr. Leelavati, where is she? Abadi. Thank you. Please sit down, Dr. Shakila. Dr. Navneet.
In nanobiotechnology track, we have one of the winners who wants to call her team members onto the stage.
Sinchana N. Prasad.
A huge round of applause.
Dr. Bhuvneshwari, can you come this side? So we'll have 5 people here and 4 there. Whom am I missing now? 2 more. 2 more judges. Who are the other judges? Sir, you are judge, please. 1 more person. आजो क्या बोल Okay, are we ready with the next talent search competition? Now this is talent search. Again, you take out your mark sheet and start giving marks. Yes, madam. Ready, madam? Ready, sir? Judges, are you all ready? Ready? Can we start? What's the next performance? Come on, forward. Tell me your name.
Surya.
And from where are you?
Sri Bhavani Group of Institutions.
Surya from Bhavani Group of Institutions. Please, can I have her presentation ready? Yeah.
தொட்டது ஏன் கருப்பாயி தொட 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 மாட்டேன் தொட்டா நானும் விட மாட்டேன்
சிறு 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 பழகா சிரித்தது என் சிறு பழகா விட்டு விட்ட விட மாட்டேன் நீ தொட்டாம நான் விட மாட்டேன் சுத்து சுத்து நெருப்பு போல ஒட்டிக்கலாமா
எங்கடா அந்த எல்லோ சாரி
மஞ்சச்சேலையோடு ஒரு மாசி கருவாடு பட்டம் போட்டு ஆடி இது வானவில் ரோடு கடிச்சா கசக்காத ஸ்வீட்டு விடானி குடிச்சா எப்போ வரும் கோலி சோடானி இடிச்சா உசுறு போகும் தண்ணி லாரி நீ அடிச்சா போத வரும் பாண்டிச்சேரி நீ ஓ சம்மதத்தை சொன்னா என் சம்பளத்தை தருவேன் நீ கை நடுவி போனா நான் கண்ணதியாடுவேன்
போறப்பதும் ஒரு நொடிதான் இறப்பதும் ஒரு நொடிதான் சொல்லடி ஞான பொண்ணு ரெண்டுக்கும் பாலம் எது பதையில முள்ளி இருக்கும்
Thank you, Surya. Thank you. Now we have Yashya. Please come. Come, beta. Go, go. Two of them. Please come, beta. Come on, dais. Not you. Sorry. 1 minute. Surya, just 1 second. Oh, Adit. Sir, presentation to her. इसको दे दो पहले. Thank you for participating. Thank you. One second. Let me start this group and I'll come back to you. Thank you, beta. Thank you. Come, Yashya. Come. What's your name?
My name is Jisya, Sri Bhavani Group of Institution.
Bhavani. Thank you. Come. Come on, beta. Name.
My name is Smiley. Sia. Sinu Sri Bhavani.
Kriba Sri.
Very nice.
Maria George.
Thank you.
And Clara.
Thank you.
Anjana Lakshmi.
Thank you.
Sanika.
Thank you. Are you ready? Yes, sir.
Judges for that conference, paper presentation.
Okay. They are here now. Okay. After this dance, I'll give you. Done.
கண்ணில் கனவாக நீ கலையாதடி மறையாமலே நீ நிற்பாயா கலையாதடி மறையாமலே உன்னுள் வழி சொல்லடி விடை சொல்லடி நெருங்காமலே நீ செல்வாயா விடை சொல்லடி நெருங்காமலே हाय चक्का चक है तू हाय चक्का चक हूँ मैं हाय चक्का चक है तू हाय चक्का चक हूँ मैं
பாட்டுனா சும்மா உச்சந்தலையில இருந்து உள்ளங்கால் வரைக்கும் சுருன்னு அப்படியே கரண்ட் பாயணும்
பங்கு
Put the song.
மாடு சுத்தா மனுஷன் நான் தோலை வச்சு மேல கட்டி அர்ற நாக்க முக்க மாடு சுத்தா மனுஷன் நான் தோலை வச்சு மேல கட்டி அர்ற நாக்க முக்க அர்ற நாக்க
டான்ஸ்
சேஞ்ச் த சாங்.
இப்ப பாரு.
ஒரு டான்ஸ் ஐயோ அத்தை நீங்க பாக்கணுமே. விஷயம் தெரியாதவன் கப்புன்னு பார்த்தானா கையில சாவி தூக்கி கொடுத்துருவான். காக்காவலி மாதிரி இருக்குது. அதுவும் அண்டங்காகா வலிப்பு.
மருதமலை மாமணியே முருகையா,
தேவரின் குலம் காக்கும் வேலையா ஐயா.
என்ன சாமி பாட்டு என்னாச்சு உனக்கு?
இன்னைக்கு தைப்பூசம் இல்ல.
இன்னைக்கு தைப்பூசம் இல்லையே.
அதத்தான் நானும் சொன்னேன். இன்னைக்கு தைப்பூசம் இல்ல. அதுக்காக முருகரை மறந்துட முடியுமா? நேவர். சட்டுக்கூடு சந்தனம் சட்டுக்கூடு சந்தனம் சக்கச்சி சக்கச்சி.
Thank you. 1 minute, 1 minute. We have our chief guest of the evening today with us, Asba. Vidya, one award. Adit. Take the, thank you. Asba, this is film star Asba, and she's going to give the award to Jessia. Please come forward, Jessia. Thank you so much, Jessia. Thank you. Like this week. Thank you, beta. Please, bet here now. So like this, we are going to give awards to another 9 performances, and then we'll start. Yes, Vidya. Dr. Vidya Niranjan wants to make an announcement. We'll go with that, and then we have puja. Come, Pooja.
Now I request Dr. Prashant Karunakaran and Dr. Mrinalini, who were the part of our panel discussion at RVC as pre-conference, to come to the dais.
Please come, madam. Please come. Commendable work, and thank you for helping our lovely teacher. Please. Ask him. Pooja, are you ready for Pooja, guys? Yeah, okay. Pooja is from RV College, and what are you going to do? Bharatanatyam, sorry. Thank you, got it.
భవ్యమైన ఆత్మభావం రమ్యమైన జీవరాగం నవ్యమైన నిత్యతాళం నిఖిల జగతి మూలం
Thank you so much, Pooja. I like Asba to give you a token of our appreciation. Please, please. Sorry, Asba, we are going to keep making you get up and down. Thank you very much, Pooja. Thank you. Prem Anshu. Prem Anshu, please. Prem Anshu is from... Come forward, give your name.
Prem Anshu Dolai, Sri Bhavani Group of Institution. Sri Bhavani Group of Institution.
Bhavani Group. And he's going to do a solo dance. Ready, music? Next. Who's next?
जवानी छुपाओगी रानी अरे कब तक जवानी छुपाओगी रानी कवारों को कितना सताओगी रानी कभी तक किसी की दुल्हनिया बनोगी मुझसे शादी करोगी, मुझसे शादी करोगी।
चल छैया 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 छैया, चल छैया 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 छैया, छैया 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 छैया, चल छैया 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 छैया, चल छैया 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 छैया, चल छैया 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 छैया, छैया 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 छैया, चल छैया छैया छैया। सारे इश्क की छाँव चल छैया छैया, सारे इश्क की छाँव चल छैया।
दिल धड़काए, सीटी बजाए, बीच सड़क पे नखरे दिखाए, सारे हो करके इशारे, हो लड़की हो लड़की हो लड़की आंख मारे हो लड़की आंख मारे हो लड़की आंख मारे।
Superb, superb, Premanshu. One sec, Premanshu, don't leave. Asbaji, please come and give us an award to Premanshu. That was wonderful, and you kept everybody's tempo high. Thank you so much. Now we have another Bharatanatyam from Aarti from RV College. Music, please. Thank you, Aarti. Please, can I have the award for her? Yes, Asba, please. Thank you so much, Aarti. And I just want you, all of you to know, Aarti has done wonderful work for us backstage as well. At every point, everywhere, she has been helping me and being a little stronghold where we are concerned. Thank you very much, Aarti. Can I have Subham now, please? Come. Use this. Give me this key. Mike, it's not working. Hey, on this mic, catch it. Can I have this mic on, please? Hello?
Good afternoon, everyone. My name is Subham Pradhan. I'm from Darjeeling, and I'll be representing Sri Bhavani Group of Institution today. I'll be performing 3 songs for you all. The first is going to be English, and the other 2 will be Hindi following it. Hope you all enjoy. I found a love for me, darling, just dive right in and follow my lead. Well, I found a girl, beautiful and sweet. I never knew you were the someone waiting for me, 'cause we were just kids when we fell in love, not knowing what it was. I will not give you up this time, darling, just kiss me slow. Your heart is all I own, and in your eyes you're holding mine. Baby, I'm dancing in the dark with you between my arms and barefoot on the grass.
Just off the
We're listening to our favorite song. When you said you looked a mess, I whispered underneath my breath, but you heard it, darling. You look perfect now. Well, I found a woman stronger than anyone I know.
Camera, show this
She
visual.
says my dreams I
Camera.
hope that someday I'll share her home. Well, I found a girl to carry more than just my secrets, to carry love, to carry children of our own. We are still kids, but we're so in love, fighting against all odds. I know we'll be alright this time, darling, just hold my hand. Be my girl, I'll be your man. I see my future in your eyes. Baby, I'm dancing in the dark with you between my arms and barefoot on the grass. We're listening to our favorite song. When you said you looked a mess, I whispered underneath my breath, but you heard it, darling. You look perfect now.
That was
Okay. तू सफर मेरा है, तू ही मेरी मंजिल, तेरे बिना गुजारा है दिल मुश्किल। तू मेरा खुदा, तू ही दुआ में शामिल, तेरे बिना गुजारा है दिल मुश्किल। मुझे आज़माती है तेरी कमी, मेरी हर कमी को है तू लाज़मी। जुनून है मेरा, बनूं मैं तेरे काबिल, तेरे बिना गुजारा है दिल मुश्किल। ये रूह भी मेरी, ये जिस्म भी मेरा, ना मेरा नहीं ना हुआ तेरा। मैं दिया है जो वो दर्द ही सही, तुझसे मिला है तो ये नाम है मेरा। मेरा आसमां ढूंढे तेरी ज़मीन, मेरी हर कमी को है तू लाज़मी। जुनून है मेरा, बनूं मैं तेरे काबिल, तेरे बिना गुजारा है दिल मुश्किल। माना कि तेरी मौजूदगी से ये जिंदगानी महरूम है, जीने का कोई दूजा तरीका ना मेरे दिल को मालूम है। तुझको मैं कितनी शिद्दत से चाहूँ, चाहे तो रहना तू बेखबर, मोहताज मंजिल का तो नहीं है एक तरफा मेरा सफर। सफर खूबसूरत है मंजिल से भी, मेरी हर कमी को है तू लाज़मी। Last one. जब जब तेरे पास मैं आया, एक सुकून मिला, जिसे मैं था ढूंढता आया, वो वजूद मिला। जब आए मौसम गम के, तुझे याद किया हो, जब से मैं तन्हापन में तुझे याद किया। दिल समझ जा जरा, फिर मोहब्बत करने चला तू। यहीं रुक जा जरा, फिर
Wonderful, wonderful. Ryan, once again. Asba, please do us the honors. Thank you, Ryan. That was fantastic. No, the next. Sorry, Asba. I call now Epsiba. Please, come, girls. Come.
Yetsiba.
Ramba. Okay, once again. We have Mr. Leo from Bhavani Group of Institutions. Can I request Mr. Leo to please come up on the dais? Please, sir. Thank you. Thank you very much, Mr. Leo. Your participation with ISGCT is very well known, and we are very, very thankful to you, sir. Please. Are you ready, music? Audio.
Pange? Put the song. Check this out.
அம்சமா அழகா ஒரு பொண்ண பார்த்த உடனே பஞ்சரான. ஆமாப்பா, அம்சமா அழகா ஒரு பொண்ண பார்த்த உடனே பஞ்சரான. அது கண்ணா இல்ல கரண்டா கன்ஃபியூசன். அவ அழக பத்தி பாட இல்ல எஜுகேஷன். என் மனசு இனி உனக்கு ஒரு பிளே ஸ்டேஷன். நீ இருக்கும் இடம் எனக்கு ஒரு ஹில் ஸ்டேஷன். ஒரு சென்சேஷன். நீ வேணும் பண்ண போற டெலிடேஷன்.
இந்த ஏரியா அந்த ஏரியா அந்த இடம் இந்த இடம் எங்கேயுமே எனக்கு பயம் கிடையாது. ஏன்னா ஆல் ஏரியாவையும் ஐயா கில்லிட.
வந்தாலும் படோடா வந்தாலும் பங்கு போகாது தமிழ்நாட்டா பாட்டு. பொங்கல் இடுப்பு எலும்ப ஒடிச்சு வச்சு அடுத்தலாம் கொடிய பிச்சா போக்கிரி பொங்கல் பொங்கல்.
Thank you, Epsiba, and please, Asma, can we? Thank you so much, girls. Thank you very much. And thank you, audience, for such a lovely participation. I call upon the last talent search, Kishan. He's going to give us a solo song. Please come, Kishan. First, you have to introduce yourself.
Afternoon, everyone. Myself, Kishan, from AIML, MS Ramaya Institute of Technology. So I'll be singing. It was a last moment preparation for me. So I don't know how would it come up. I'll be starting with something classical and then followed by some film songs, which are basically my favorite. करधृत वीणा पुस्तक वरमणिमालालङ्कृत करधृत वीणा पुस्तक वरमणिमालालङ्कृत सुरसेवित पदयुगले
सुरसेवितपद युगले सुरसेवितपद युगले सुधाकर समदवले सुधाकर समदवले सरस्वती नमोस्तुते शारदे विद्याप्रदे सरस्वती. Coming up to the film song. तुम्हारी तस्वीर के सहारे मौसम कई गुजारे मौसमी ना समझो पर इश्क को हमारे नजरों के सामने में आता नहीं तुम्हारे मगर रहते हो हर पल मंजर में तुम हमारे गर इश्क से है मिला फिर दर्द से क्या गिला इस दर्द में जिंदगी खुशहाल है ये दूरियां फिलहाल है हो खैरियत पूछो कभी तो कैफियत पूछो तुम्हारे बिन दीवाने का क्या हाल है दिल मेरा देखो न मेरी हैसियत पूछो तेरे बिन एक दिन जैसे 100 साल है. Next one. I hope you love this too. तलब 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 बस तेरी है मुझे नसों में तू नशा बन के घुलना यूं ही मेरी मोहब्बत का करना तू हक ये अदा मेरी हो के हमेशा ही रहना कभी न कहना अलविदा मेरी सुबह हो तुम ही और तुम ही शाम हो तुम दर्द हो तुम भी आराम हो मेरी दुआओं से आती है बस ये सदा मेरी हो के हमेशा ही रहना कभी न कहना अलविदा आहा ही रहना कभी न कहना अलविदा. And in Kannada, we are in Karnataka, so one Kannada song. Hope those who have got married might have heard in their marriage halls. I'm not sure about that. ಪ್ರೀತಿಯಂದರೇನು ಎಂದು ಈಗ ಅರಿತೆನು ಪ್ರೀತಿಯಂದರೇನು ಎಂದು ಈಗ ಅರಿತೆನು ಸವಿನುಡಿಯಲಿ ತನು ಅರಳಿತು ಸವಿಗನಸಲಿ ಮನ ಕುಣಿತು ಒಲವಿನಾಯಿ ಮಾತಿಗೆ ಕರಗಿ ಹೋದೆ ನೋಟಕ್ಕೆ ಕೊಡುವೆ ನಿನಗೆ ಬಾ ಪ್ರೀತಿ ಕಾಣಿಕೆ ಜೊತೆಯಲಿ ಜೊತೆ ಜೊತೆಯಲಿ ಇರುವೆನು ಹೀಗೆ ಎಂದು ಹೊಸ ಹರುಷವ ತರುವೆನು ಇಂದು ಎಂದು ಓ ಎಂಥ ಮಾತಾಡಿದೆ ಎಂದು ನೀ ಎಂಥ ಮಾತಾಡಿದೆ ನನ್ನ ಮನಸ್ಸಿನ ಭಾವನೆ ನೀನೆ ಹೇಳಿದೆ ಜೊತೆಯಲಿ ಜೊತೆ ಜೊತೆಯಲಿ ಇರುವೆನು ಹೀಗೆ ಎಂದು. Thank you so much.
Thank you very much, Kishan. Asba, once again, please. And as we are going to be waiting for the judges to give us their results, we start. Thank you, Kishan. We start with a valedictory function. I'd like to ask Bhuvna, Vijeta, and Vidya, please come up on stage. And I'd like Asba, we are starting our valedictory function. And today our winners for the ISGCT Leadership Award is Asba, who is the film star, and Mr. Leo from Bhavani Institutions. As we're starting the valedictory function, we have a very, very nice and special invocation from RV College. And all the volunteers who've done so much effort in making this convention a success. I request all the volunteers, please come up on stage. Volunteers. As you come, I'd like you to introduce yourself.
I'm Sukrit. My name is Anirudh Aris.
My name is Anaga.
My name is Adit.
My name is Risha.
Hi, my name is Ananya.
Hello everyone, I'm Aparna Kashyap. Hi everybody, I'm Anushri. Hi everyone, I'm Shriya. Hi everyone, I'm Aratrika.
Hi everyone, I'm Disha.
Hi everyone, I'm Shivakami. Hi everyone, I'm Nivriti. Hello everyone, I'm Arya.
Hi everyone, I'm Charan.
Hello everyone, I'm Kanva. Hello everyone, I'm Kanva.
Hi. Hello, I'm Prajakta. Hi everyone, I'm Archisha. Hello everyone, I'm Kavya.
Kavya and Arjita, all of you, please, a very big round of applause. These are our technical team. If you have seen this whole thing going with a wonderful audiovisual, it's these guys.
Hi, I'm Darshini.
Hi, I'm Srijita.
Come forward. We need to get the circle bigger.
Hi, I'm Bhargavi.
Hi, I'm Advait.
Hi, I'm Natasha. Hi, I'm Shreya.
Hi, I'm Harshit.
Okay. Come, now all of you come into this little area. Ready? Come front. Come as forward as possible. The girls in front, please. Come forward. Come. Little girls, more forward. Little girls, come forward. Where's the big flag? Very good point. Superb. Come on, come down. Take a knee. There we go. Okay, ready? Are you ready? Yeah. Hey, go. Come on, everybody who volunteered for this, come up on stage now. Ready? Where's your flag? A big flag. There we go. Sorry? Yes, I am here only. Start.
आओ बच्चों तुम्हें दिखाएं झांकी हिंदुस्तान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम।
आओ बच्चों तुम्हें दिखाएं झांकी हिंदुस्तान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
Sing along.
उत्तर में रखवाली करता पर्वतराज विराट है, दक्षिण में चरणों को धोता सागर का सम्राट है। जमुना जी के तट को देखो गंगा का ये घाट है, बात बात में हाट हाट में यहाँ निराला ठाट है। देखो ये तस्वीरें अपने गौरव की अभिमान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
Vande Mataram. You do me a favor, give her the flag. Now you see. Come. There you are.
ये है अपना राजपुताना, नाज इसे तलवारों पे, सारा जीवन काटा बर्ची तीर कटारों पे, ये प्रताप का वतन पला है आजादी के नारों पे। कूद पड़ी थी यहाँ हजारों पद्मिनियां अंगारों पे। बोल रही हैं कण कण से कुर्बानी राजस्थान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम।
देखो मुल्क मराठों का ये यहाँ शिवाजी डोला था, मुगलों की ताकत को जिसने तलवारों पे तोला था। हर पर्वत पर आग लगी थी, हर पत्थर एक शोला था, बोली हर हर महादेव की बच्चा बच्चा बोला था। शेर शिवाजी ने रखी थी लाज हमारी शान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम।
जलियांवाला बाग देखो यहीं चली थी गोलियां, ये मत पूछो किसने खेली यहाँ खून की होलियां। एक तरफ बंदूकें दंदन एक तरफ थी टोलियां, मरने वाले बोल रहे थे इनकलाब की बोलियां। यहाँ लगा दी बहनों ने भी बाजी अपनी जान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की।
वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम। Oh, there's one more stanza. Okay, continue. I didn't know if you guys want to hear it or not.
देखो बंगाल यहाँ का चप्पा हरियाणा है, यहाँ का बच्चा बच्चा अपने देश पे मरने वाला है। ढाला है इसको बिजली ने, भूचालों ने पाला है, मुट्ठी में तूफान बंधा है और प्राण में ज्वाला है। जन्मभूमि है यही हमारे वीर सुभाष महान की, इस मिट्टी से तिलक करो ये धरती है बलिदान की। वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम।
वंदे मातरम, वंदे मातरम, वंदे मातरम, वंदे मातरम।
And thank you for sharing with us this fervor of patriotism. Vande Mataram. Thank you. Aju. I will now request Vidya Niranjan and Bhuvna to I'd like to ask Asba to please come up on stage. And Bhuvna, please give her the Leadership Award with flowers and the bag, delegate kit bag. Tanjore one. Oh, Krishna. Thank you so much, Asba. Thank you so much. Yeah. You can put it all in your... we'd like you to share a few words with us. Pehle isko chhod do na. Ye le lo, Bhuvna. Yes, ma'am.
I want the Josh to be on. How's the Josh? Namaskara, Bengaluru. Hello, everyone. I'm deeply elated, honored, thrilled, and enjoyed to be here among such a fantastic, energetic crowd. I remember my college days, which were just fab as it is. Take the best advantage of this time. You will get never that back again. And thank you, Sunita ma'am, for having me here. Esteemed judges, the people who have coordinated today, Dr. Hajira, my very good friend. Well, if I have to say, let me quote, there's a saying by Nelson Mandela. She says, opportunities are never the end. They are the result of your talent, your hard work, and your dedication. We all have thriving to be something or the other in life, but trust me, without talent and that muska and chaska in life, nothing is possible. You need that kind of, fun element within yourself to gear you up to be a better person, to enjoy your life, because everything seems serious and we all have responsibilities at the end of the day. But trust me, all that makes you powerful and go ahead is just the wide smile that you keep on your face. I wish you all the best to all your endeavors coming ahead. And so since it's a talent show, I would like to sing a small song if you're okay.
Definitely, definitely.
पल भर ठहर जाओ, दिल ये सवर जाए, कैसे तुम्हें रोका करूं, मेरी तरफ आता हर गम फिसल जाए, आँखों में तुमको भरूं, बिन बोले बातें तुमसे करूं, गर तुम साथ हो। Come on, मेरी नजरों में तेरे सपने, तेरे सपनों में है नाराजी, मुझे लगता है कि बातें दिल की होती लफ्जों की धोखेबाजी, तुम साथ हो या ना हो क्या फर्क है, बेरंग थी जिंदगी बेरंग है, अगर तुम साथ हो। Thank you.
Wow, that was fantastic. You're not going off the stage right now. I'd like to ask Mr. Leo from Bhavani Institutions to please come here and take guys this. The leadership award. Asba. Thank you so much. You hold it and take it away also. Please, sir. Thank you very much, sir. So we'd like you to share a few words with us.
See, good evening, everyone. Good evening. Past three seminars, we have been participating along with Madam. Actually speaking, I no need to thank you all. I'll have to thank Madam only. Because looking at her energy, you all getting more energy. Am I right? That is the real fact. That is why me and my institution is always associated with Madam.
Thank you, sir. Thank you very much.
I thank for
Thank
that.
you, all of you. Thank you. Thank you.
And no more advices, because you had enough advices from me, specifically the Bhavani students. And I started reducing the advices. Thank you. Thanks a lot for the opportunity.
Thank you, sir. Please, I'd like Dr. Bhuvneshwari to please announce our talent search winner.
So, good evening, everyone. So, here are the winners. And from the morning, the talent search is going on. And to start with, the winner of the talent show, we have some prizes for all of them. Varshini, who performed Kuchipudi in this morning, who just began this event in today's morning. So, we need Varshini here. Okay. Then Aarti, who performed Bharatanatyam.
Who's here? Aarti. Yeah. Sir, come, sir. Please. Come, Vidya. Bhuvna. Please, all our judges, please come up on the stage. Talent search judges, please come up on stage. Okay. Come. Talent search judges, please come up on stage. Thank you, Aarti. That was a wonderful performance.
Thank you. And Rahil and team, the boys, trio boys. And for the dance, we have a consolation also for Epsiba group.
Yeah, are they there? Okay. No, only, right. Sir.
No, one for singing, no? That one. Shubham Pradhan. It's a pride moment for a principal. And for the song category, Shubham Pradhan, who mesmerized the whole auditorium, including the judges. Where is he? Shubham? Congrats. So keep rocking, students. All well done. Thank you very much.
Thank you, guys. Please, all our judges and our chief guest, please. This is our signature movement. So we do this. Vangu sir, you know our signature movement. Thank you. Don't go off. We are now going to play the national anthem. Flag. Huh? And after that, we have the Alap group who's going to give you music and entertain you with the band. Please play Janagana Mana. So we are going to all sing Janagana Mana. All in one line, please. Can I have Mr. Krish Velmurugan? Please come up, sir. All the faculty, please up on the dais for the national anthem. Mr. Akshay, Lavanya, please. Sir, Mal. Vango, sir. Photo le varom, sir. Please. Sir, inge vango, sir. Circle bigger, don't worry. All the faculty of the ISGCT, please up on the stage. Okay? Okay? All good? So now we're doing the national anthem. I need the flag at the back. National flag. Who has the national flag, please? Kanva. Adit. Can you please bring me the national flag, Anagya? Come back. Anirudh, please wait at the back. Everybody is going to sing. You are not patriotic by just standing. It is when you sing with your hearts. Yes? Ready? So we're all going to put our hands to our heart, pledging patriotism to this wonderful country. Ready?
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you. That was wonderful singing from all of you. It was a chorus movement. Once again, I would like to thank Mr. Leo, Asba, Bhuvna and Vidya who have made, please come forward. Vidya and Bhuvna who have made this convention possible. This whole thing is these two's hard work. So please a round of applause and I would like to give them, hey, award lao bhai. Dono ka award lao. Flowers, please. Give. Correct, come. Come forward. I cannot thank you both enough. Their words absolutely are insufficient for this whole exercise. Asba ji and Leo ji, please. And flowers. Thank you all very much. Thank you for being a wonderful audience. And thank you so much for being a part of ISGCT. I bid you farewell, but there's always tea and coffee. And we're having a very beautiful music band. They will set up in another 5 minutes and we are ready to go. Thank you. Thank you, sir. Thank you so much. RV College, I'm so indebted to you all. Thank you for filling all my gaps. Ayo, thank you, madam. Thank you. Thank you so much. Thank you. Are baap re baap. Thank you, Subha. Thank you.
Now I would like to call upon Alap, the band of RV College of Engineering.
Photo, please. Sumit, oh, there. Okay. Come. Thank you. Yes, certainly. Thank you. Alap band. Tea, coffee is ready. You can have that by the time they'll set up the band. Huh? Raining, okay, better. I'll get it here.
Please wait for the Alap performance. They'll be setting up in another 5 minutes. Thank you.
Guys, just 2 minutes for Alap to set up. Kindly sit back.
Pros wakened up here. No, you're all well settled. Okay, no problem. Kavish, can you do me a favor? First, introduce your band. Thank you, Alap Band. Thank you, everybody. And I'm off. Thank you.
Thank you very much, ma'am. I hope you guys had a great time today. And to end it off on a good note, we have come from all the way from the RV College of Engineering to give you guys a wonderful experience of what we basically are. So we have a couple of bands within Alap, and one of the famous ones are Project Mishram, which was also nominated for the Grammy Awards. And this is an Eastern band, and we'll be performing a couple of multilingual songs. So we have Hindi, we have Kannada. So I hope you guys enjoy. Just give us a couple of minutes. We have some sound checks to do, and then we'll soon start with the performance. Thank you very much. Sir, could you increase the volume of this key on the monitor, please? Just a couple of minutes, guys. The guitar is being set. I promise you guys will have a good time.
Hello, hello.
Check.
Hello. Hello, hello.
So I hope you guys have a great time. If you know the song, please sing along, and you can you just have a great time.
Thank
के हमने सारी पी दी हंसी। शकों पे पत्ते गा रहे हैं, फूलों पे भंवरे गा रहे, दीवाने किरणें गा रहे हैं, ये पंछी गा रहे। ओ बगिया में दो फूलों की हो रही है गुफ्तगू, जैसा फिल्मों में होता है हो रहा है हूबहू। आए 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 सुरगुल सुरगुल पापा, सुरगुल पापा, सुरगुल सुरगुल जान रे इन सांसों में बस जा जान रे तू तड़पाना तू मुझको चाहत है अगर आके मिल जा मुझसे तू या फिर ऐसा कर आजा धरती मिला दे मुझको तू ही रे तेरे बिना मैं कैसे जीऊं आजा रे आजा रे सांसों में बस जा
ये हौसला कैसे झुके ये आरज़ू कैसे रुके मंजिल मुश्किल तो क्या धुंधला साहिल तो क्या तन्हा ये दिल तो क्या
So the next song is a crowd favorite. This is the last song we'll be doing tonight. I hope you guys love this one.
मेरे ही घेरे में घूमेगी हर पल तो ऐसे सूरज की घेरे में रहती है धरती ये जैसे आएगी तू खुद को ना मुझसे जुदा तू है मेरा आधा सा हिस्सा सदा टुकड़े करता है ख्वाबों के तू मेरे टूटेंगे भी तो रहने हैं वो तेरे मुझको भी तो ये इल्हाम है ऐलान है जब तक जहां में सुबह शाम तब तक तेरे नाम लो जब तक जहां में मेरा नाम है तब तक तेरे नाम लो जब तक जहां में सुबह शाम है तब तक तेरे नाम लो जब तक जहां में मेरा नाम है तब तक मेरे नाम लो
Okay, I hope you guys are wonderful. You guys are absolutely wonderful. Okay. Yeah. So we have a special request. We have a special request, and I'm sure that's one of the most loved songs in this country. I hope you guys enjoy this and sing it along, okay? It will be just me and this is all of a sudden, so I hope you guys have fun.
तेरे बिना क्या वजूद मेरा तुझसे जुदा कर हो जाएंगे तो खुद से हो जाएंगे जुदा क्योंकि तुम ही हो अब तुम ही हो जिंदगी अब तुम ही हो चैन भी मेरा दर्द भी आशिकी अब तुम ही हो क्योंकि तुम ही हो अब तुम ही हो जिंदगी अब तुम ही हो चैन भी मेरा दर्द भी मेरी आशिकी अब तुम ही हो
Thank you guys.
That's wonderful. I'm not sure whether Alap knows I am lovely guys, and I hope you would not have got such a crowd, beautiful crowd. But we need to stop our conference call. Okay, thank you so much, and I would like to give you an award on behalf of.
Let him take it. Let them.
We'll have a big pose. All of you.
Are the girls there?
Yes, ಅಲ್ಲ ಅದು ಬಿಡು ಇರುತ್ತಲ್ಲ ಮ್ಯಾಗ ಕರ್ಕೊಂಡ್ಬಿಟ್ಟು
Though this is the end, we have one more track. We will have a song played. Whoever is interested to come and dance on the stage. Open floor. After we take up these things, we will put songs. Whoever wants to dance can come on the floor. Open floor.